right, we're live. Welcome back, everybody. Good morning, good afternoon, and good evening from wherever you could be tuning in from. We're super excited to have you back. ROV SuBastian, after like a month-long break or so, is back in the water, and we're really excited to be diving here. Today, we're off the coast of northern Chile, and we're super duper excited to see what kinds of things we're going to find today. We're just doing a quick white balance on our main science camera, which is the feed that you're watching right now. And I guess I'll introduce myself. I'm Sophia. I'm the multimedia technician. I'm really excited to be bringing you the broadcast this expedition. And in the room with me, I've got Dr. Lauren, who's going to introduce herself in just a second. We've got Andy and Chris as our pilots today. And behind me, I also have a special guest, Hannah Nolan, who is joining us for this expedition as well. And we've got a really cool team of scientists on board with us this expedition. We've got microbiologists, and we've got astrobiologists, and chemists, and physicists, and just a really, really cool bunch of people. And we're really excited because we're going to get to know them a little bit better throughout this week. And yeah, thank you so much for your patience in waiting for this broadcast, and we're really excited to get this expedition going. We're going to be investigating primarily a lot of microorganisms that live here, and right now we've just touched on a xenophyophore garden, which is also really exciting. Also, here's a little secret. We saw a Dumbo octopus on our way down here. So a little bit more promising midwater content for you all, and we're gonna be broadcasting hopefully some more descents and ascents throughout this expedition, so stay tuned. But without further ado, I'm gonna pass it over to Dr. Lauren, who's gonna introduce herself and the expedition and get this dive started for us. Over to you, Lauren.
Thank you, Sophia. My name is Dr. Lauren Seiler. I come from Stockton University in New Jersey in the United States. And we are here off the coast of northern Chile at the Atacama Trench. We're about to pick up a sea cucumber here for one of our biologists. There's a lot of sediment on the bottom here. It's very, very fluffy. So we are here because we want to explore the ocean bottom off the coast of the Atacama Desert, which is the oldest and driest desert in the world. And the seafloor at this particular part of the world is not very well known. And we're hoping to find some incredible new sea creatures here, and also to look at the microbiology in the benthos at the bottom of the ocean and find out what microbes are living in the sediment. That's my particular area of expertise. I'm a microbiologist. But as Sophia said, we have a whole array of people here with us, biologists, geologists, chemists, so that we can better characterize this area and understand it better. So we're about to pick up this little sea cucumber off the floor here with our pinchers, carefully.
Okay.
You can imagine how difficult it might be to control a robotic arm and pick up something as squishy as a sea cucumber carefully. So we have to be very, very gentle and move very slowly. You can also see there's lots of particulate matter in this water. The bottom is very, very loose and fluffy. And as we were descending, we also saw a lot of particulate matter in the water column, and this is because we are under a highly productive part of the ocean. So there's lots and lots of productivity in the surface ocean, and all of that biological activity means that it all sinks down in the water column and then comes down to the bottom here. So because we're in such a highly productive area, we're seeing like lots and lots of little bits of stuff in the water column, and then that all sinks to the bottom as this fluffy particulate matter. So if we have kind of a cloudy resolution, that is why. And I think we're about to get him here. There he goes. And we're going to put him into the bio box and bring him up on board so we can take a look. Very, very cool. This is the very first dive of this expedition, so we're super excited to finally get down here and have a look. We've been chomping at the bit to get here, and this is really exciting. Even on the way down, we saw a Dumbo octopus. We saw a really beautiful ctenophore, otherwise known as a comb jelly. It was bright red. And Nick, one of our biologists, informed me that it's very common for deep sea organisms to be red because it camouflages them in the water. So a lot of deep sea organisms are red for that reason. I didn't know that. Alright, and we're just very gently gonna lower that in there. Perfect. Nice. Super cool.
Our next recommendation for samples.
What would we like next?
Second, can I get
sorry.
a confirmation of which biobox that was?
That was Biobox A, 1A.
1A.
Yes.
Thanks.
We have Drew here doing all of our sea logging so that we can log all of our samples. And what would we like to take next?
Our next recommendation for samples are some gray structures that are probably over sponges covered with sediment.
Okay.
And we like 2 specimens.
Okay.
You can see them on the left side of the ROV.
All right. So these
2 of those.
gray structures, possibly sponges covered in sediment.
Yeah.
2 of them.
You get one? Those.
Yes.
Okay.
Okay. Let's pick up 2 of those.
Yeah. So you think they're sponges? Okay.
Hey, Lauren.
Mhm.
Once more, that was biobox 1A.
That was Biobox 1A for the sea cucumber. Do we want to put the sponges in a separate box? Can they go in the same one? All right, if you're just...
Right here?
Yeah. If you're just joining us now, we are on the bottom of the seafloor off the coast of northern Chile and the Atacama Desert, near the Atacama Trench. And we're about to pick up some sponges off the seafloor.
You want to put this in a quiver or
No, one.
I'll ask, would you like it in a quiver or a bio box?
Pero, biobugs.
Bio box, please.
2 specimens of those gray structures.
So these gray structures down at the bottom that look like sponges, we're going to get 2 of those.
Just sponge covered with the same.
Okay.
Xenophytophores?
Should I just write that as unknown organism?
Xenophytophores? I believe that's what they are.
Ctenophytophores?
Xenophytophores.
Xenophytophores. Sorry.
Xenophytophores. These are foraminifera.
Foraminifera?
Yeah, these are huge, like
Oh.
foraminifera.
Oh, I always thought of foraminifera as being like teeny.
No, they're massive. They can get really big.
Oh, wow.
You think? Okay.
Okay.
We've seen them before.
They're very common, yeah, but.
Okay, so apparently foraminifera can get way bigger than I realized. This is what happens when you let the microbiologist talk about animals.
No, I've seen them. I know. I've seen them. Yeah.
No, I know, it's not an animal, it's a protist. But anything bigger than microscopic.
It's a lot of xenophytophores.
It's okay because the fauna, these structures that we can find many associated fauna.
Yes.
Yes.
So, yeah.
All right, so we're going to pick these up and hopefully find lots of little animals inside of them.
Yeah.
So these are
Of course,
technically not
especially
animals,
if
they
you're
are
going to the museum.
protists.
I mean, some very specific things like.
So a protist is an organism that has a nucleus in its cells, like plants and animals and fungi do. But it doesn't really fit in any of those categories.
For both.
And in school you might have learned that protists are single-celled eukaryotes, that's what I learned, but actually, protists can be multicellular as well. Most seaweeds are not plants, they are protists, in fact. So we're going to collect a couple of these xenophyophores, and maybe find some little fauna inside of them. And that is going into BioBox 1B. D? Sorry, BioBox 1D.
There's no getting half in there. Do you care if it goes over his cap?
Dos de ellos, okay? No es small. She's right.
Yeah, D is fine.
Big for a mammifer.
Okay.
We have
Biobox 1D is in David.
Yes, D is in Delta.
I didn't know that there is a few days and I started looking at videos.
All right, so we're going to collect one more of those.
Yes.
All right. 2 xenophyophores. I'm just going to very carefully pick that up.
But is it xenophyophores or from?
Oh,
No, xenophyophores are for mammifer.
hi Keith. Keith is my husband. He's in the chat. And my daughter Eleanor says, I love you, Mommy. Hi. She's 5. Hi, Ellie. Thank you for tuning in and watching us work. I'm so excited that you guys get to see this. Eleanor really loves octonauts, so, you know, she knows all about this stuff. I was showing her how deep we were going to go with the ROV, and she said, that's the dark ocean. And I said, yeah, that's right. So she's already a budding oceanographer.
Always better to be on the side.
Just a pole.
And that's also going in D.
Yes, is that okay?
Yeah, remember me?
All right. So 2 of those xenophyophores in BioBox 1D.
Perfect.
Very cool.
There is one structure above core 7 and 8 that got the attention of Armando, and maybe
A structure above core 7 and 8. So this thing over here?
Yeah.
Okay.
Yeah, well, find that. We have to go up a little bit to get that a little bit easier. Would you want that the same
My girlfriend
spot, or
Jax
you want
says,
it
join the
somewhere
chat. Hi, Jax.
Uh,
Most stressful claw game
we don't
ever.
have a preference for this.
Okay.
Yes.
So true.
No, brachiopods are not exhausted. It could be a bivalve. It could be a bivalve.
Yeah. Okay, so that we're looking at this structure right here between the 2 green lasers, trying to determine what it is. Maybe it's a bivalve. Yeah, looks like a scallop or something.
Yeah, it's probably a clam or something.
Or clam.
Yeah, it's a bivalve. With a few bullets on the
Ooh,
base.
those are the eyes on the outside there? There's a brittle star there too. Look at the little pink
Couple
brittle
of
star.
bullets, yeah.
Right now we are
Yeah, so this is bivalve.
At
Mollusks.
2,900 meters.
Brachiopods need hard substrates. They cement over substrates or they have a better
Of depth, and we are looking at this bivalve here with a brittle star next to it. It's a really pretty pink color.
sponge.
My friend Chris is in the chat. Hi, Chris. Yes, the robot is very gentle. It has to be gentle. Otherwise, we'll squish all of our specimens. Is that a shrimp over there on the? There's a shrimp over there on the right, kind of going along the bottom there. Okay, so what would you guys like next?
We would like to collect that yellowish specimen, which could be a bivalve or it could be a sponge, depending. I mean, we cannot tell right now.
Let's collect that specimen there that looks like a bivalve, please.
But yeah.
Or it could be a sponge. We're not sure yet. Ooh, what's that in the water there? On the left.
Yep.
Worm?
Polychaete, polychaete.
Polychaete worm. Hey, buddy. How you doing? Hi, polychaete.
They are benthic, but they have to swim. They're predators, so they hit the water column to eat prey, and then they're gonna.
Oh, wow. So he's gone hunting. Or she. They're going hunting. That is super cool. Yeah, there's a ton of marine snow down here.
The shrimp?
All right, so we definitely want to get what appears to be a bivalve in the middle. Can we take that, please? There's also a shrimp there. All right. We would like to take that. Yep.
Alright.
Take that. Get that bivalve. Get that bivalve.
But there's something definitely there. But let's concentrate on that bivalve.
All right, we're gonna head over there and grab that bivalve.
My neck is coming down. I cannot do this. Still here? Yes. No, you're fine. Actually,
Definitely seeing like some worm casings down here at the bottom too, like little burrows and things. All right, and we're gonna very gently grab that bivalve and put it in a bio box so that we can take a closer look. There's just so much down here. I think there's another Dumbo octopus over in the upper left too. I can go say hello to that. Is anybody interested in that polychaete worm too? Confer with the biologists and see what they would like to take next.
Yeah. Freeze it, look at it real fast.
Very cool looking polychaete worm. And also
Is that the shadow that
something shiny on the bottom. Looks like a little round globe. I don't know what that is. Interesting. Yeah, what's that?
looks like it has eyes? I feel like it's looking at me back.
It's the marble.
Like, what is that?
Oh, it could be, well, it could be many things. It could be like the very smooth valve of a bivalve.
Oh, okay.
We're not gonna know until we dig it out.
Okay.
But let's concentrate on that.
Yeah, let's get this guy first, for sure.
No, bivalvia.
Yeah, look at that. Look at all the little eyes.
Yeah, all can
So
see from there.
cool.
I don't know.
Okay, we're just gonna very gently pick that up. There's also a brittle star
No.
kind of hanging around there, and as you can see, there's a little shrimp kind of coming to check out the claw.
Some kind of a no sponge.
All right, maybe we'll. There we go. Oh, and we're getting the brittle star as a bonus, maybe.
Very careful, very careful. So
Yeah.
as well. Just hang in there.
Yeah. I think we're getting the brittle star too.
Bye, bye,
Oh,
brittle
nope,
star.
there he goes.
Bye,
Yeah.
bye.
Bye, brittle star. Okay. Thought we were getting a 2-for-1 deal there, but I guess not.
There's no bivalve.
All right, we're gonna bring that over and pop it into the bio box. Which one are we aiming for?
Sure. Where is it?
I won't put it in, I'll put it in a different one so it's not to disturb the,
Yep.
No.
I'll put it in C1C.
Okay, this one's going in bio box 1C, Drew.
About 1C.
Correct. C is in Charlie. Brittle star as a bonus.
For
Yes,
Nick or
yes, you
Guillermo?
are, Jax. Yes, you
all,
are.
all these animals by default would be for Guillermo.
goes.
And
Yay.
yes.
But by default,
This is a stressful claw game.
The shrimp look like classified.
Stressful, but lots of fun. Okay, so what would we like to collect next, or would we like to move on?
We would like to collect the polychaete if we see it again.
The polychaete, okay.
Yes.
Alright, we get the, look at the,
If we see the polychaete, let's try to get the polychaete.
Otherwise, the shrimp.
Otherwise, the shrimp. Okay, so polychaete, otherwise shrimp. Got it. Is that an octopus on the bottom in front of us? I see the shrimp.
Shrimp may be pacified. Look like Cnidarians.
The shrimp. Can we get the shrimp with the? We gotta get the shrimp with the little suction. Yeah.
The
So, yeah.
suction tube is full of sediment. Huh?
Yeah, there's a bunch of
Yeah.
sediment inside the suction
Yeah,
tube
we'll
right
suck it up
now.
before we get anything
So
Ah.
just get that out of there.
Are we sectioning the shrimp?
Yes.
Ready?
So we're going to suction the shrimp because he's too small to get with the claws. So we're going to just very gently use our little suction sampler here to pull. First we have to get all this sediment out of there 'cause there's a bunch of fluffy stuff there. So we're going to get that out and then try to suction up this shrimp in our suction sampler.
alright, second pull out of that.
All right. Just a little shrimp. Look at that.
Yeah, we still seven with that.
Yeah, it's still set. Cnidarians.
Yeah, the bottom here is just incredibly fluffy. Okay, so just very gently gonna put this suction sampler down so we can try to get the shrimp and avoid getting all this fluffy material. It's gonna be pretty tricky, I think, but our pilots are really good at what they do, so no concerns there.
Is it maybe it's possible to get it to move on the water column so you don't have to deal with all that sediment?
Maybe
Goes in the water
kick him
column,
up
then
into the
we'll
water
get
column.
it.
No, he's like running away. I do not.
I'm gonna try to-
No, sir, I do not want to be abducted. I have to chase him. Alright. Make another go at it. But yeah, get the lights on there. Oh, it's a little. Ah! Oh, so close! Oh, no!
Oh, he doesn't.
Oh, he's putting up a fight. Look at him. I like his I like his spunk. I like his attitude. He's just like, no, not
Oh,
today.
he's up in the air. He doesn't want to.
Wow.
He's up by the.
Yeah.
Yeah,
All right.
you're
He's
right.
swimming now, so maybe we can avoid all that fluffy junk. My husband says, "No probe, please. Very busy."
Nope.
No.
No, we can't reach him. Wow, feisty. That he is a feisty shrimp. I admire that.
So it's good for picking up the
Tiny but mighty.
and not bad at 5 hours.
If we don't have another option here for now, I would suggest to get a sediment core of undisturbed sediment.
We have a request for an undisturbed sediment core.
Okay.
So we're gonna let that shrimp go on his merry way, I think.
And it's the shrimp that got away.
All right. So let's take a core. Chris says, absolute mad lad. Truth. So, if you are just joining us, we are off the coast of northern Chile,
Are we out all the way?
near the Atacama Trench. We are at 2,923 meters, and we've been taking some animal samples, and we're about to take our first sediment core in the hopes of capturing some biofauna.
Things too unwieldy, dude.
Become unabductable. Yes, good advice. All right, so we're gonna take our first sediment core here. We're gonna try to take it from an area that we have not kicked up any bottom sediment yet. So something undisturbed, if possible. My friend Pete is in the chat. He says, congrats, you've just doomed that shrimp to never being believed by other shrimp for the rest of his little
I might
life.
get it
Yeah.
from where we are, but how many just want one?
Oh, there's a polychaete worm over there in the water column. I know some folks were asking for worms. Oh, he's going way up in the water column though. Alright. Nick, do you have a preference for where you want to take that core, or just any spot that's not?
Any spot between the foraminifera,
Okay.
especially when it's has a little more color, a little more grayish.
A little more gray?
Yeah.
You got it. So we're going to take a sediment core here and take our first push core. That's sort of opposite of the very large telescope that is up at about 2,900 meters inland. Yeah. A lot of similarities, I think, between exploring the bottom of the ocean and exploring
Yeah, be
the
out here.
surface of
That'll
another
be, that'll
planet.
be just fine.
There we go. Yep.
We're gonna get it by pure chance.
Right about there.
Just kind of a big structure.
Looks like there's some more animals up ahead if we want to head in that direction after this. Wow. All right, then we're just going to push that down as far as we can. Take a nice core of sediment for Nick to look for myofauna. So he's going to take that sediment core and put it through a fine sieve and look for lots of tiny animals.
That's
In the
very
top.
soft and feely stuff.
Yeah, that is very squishy.
Yeah,
Here's hoping we're able to get
that's good. Myofauna
something
stuff.
out of
This
there.
is
Yeah,
really-
good spot for myofauna. Nice and squishy. All right.
Stay.
Yeah. Stay. Okay. Ooh.
Excellent.
Okay.
Yes,
That is a lot of fluff.
Beautiful.
That's a whole lot of fluff. Okay. Now I'm just gonna
That's great.
pop it in there. So that's core 1.
Okay, core 1.
Yep. Core 1 for Nick. Nick's core.
Nice. Perfect.
Okay.
Thank you.
Oh, you are most welcome. All right, that's our first push core. Yay! Very exciting. A first of many more to come. Let's push that guy down in there. Yeah.
There we go.
All right.
That's like a
Nice. Okay.
we call it a microscope.
So I'm definitely seeing some stuff further ahead. Is that the general direction we kind of want to move in? Or
What is this?
what is over here?
Uh-huh.
This looks like it could be another Dumbo octopus on the bottom, maybe? I don't know if they're down this deep. What is it?
Most likely a sea cucumber.
Sea cucumber and another brittle star over here to the left.
And it's definitely a different species than the first sea cucumber.
Would we like to grab this sea cucumber since it's a different one from the first one? Okay.
Yes, please.
Okay. Fetch the science tweezers. No, the other science tweezers. All right. Okay, let's go get that sea cucumber. What's that to the left of that sea cucumber, that long looking thing? Can we zoom in on that too? Ooh, what's that?
That's an acorn worm.
Acorn worm.
Yes.
Pretty. I like the color. That's my favorite color.
That's beautiful. That's beautiful.
Would we like to get that as well?
They're pretty fresh. But
Or we can try?
It's just, they're pretty fresh.
Okay, so they're pretty fragile, so we can
We
make
can try.
an attempt. All right, well let's aim for the sea cucumber first.
We're cousins. We need to give it a try.
Yeah. All right. Okay, sea cucumber and then maybe the acorn worm. Pretty please.
Hemicordata.
All right.
Hemicordata.
Let us move forward. Wow. I don't mind telling y'all this is pretty incredible. It is so amazing to be able to see all of this in real time. I hope we can see some good cephalopods too. We saw a really great Dumbo octopus on the way down, and I want to see more of those for sure.
I saw a photo. Yeah.
I love that sea cucumber. Oh my gosh. What's that
That is
below the acorn worm?
It's no,
Oh, looks like there's a bunch more of those sea cucumbers up ahead. So maybe we can grab a couple of them. I would like to take an extra one to look at the microbiome if possible. So if we can get a few of those sea cucumbers, that would be awesome. Horror Movies 101 as a bio undergrad prereq. That's my astrobiology class.
that was one.
And the first movie we watch is Alien. So the moral of Alien is do not let the face hugger on your ship. Oh, there's a polychaete down there.
Yep.
Do we want to try to get it?
Have
Nick?
to get the, it's.
Or it might be tough.
Yeah, I think, look, they got the.
Okay. Well, yeah. Let's.
Pretty plentiful though, so I think if.
Yeah,
if
I think we
wanna
can.
Set up for it, like have the.
Yeah.
And then kind of keep an eye out for them.
Yeah. Let's keep an eye out for those, so we can grab a worm or two.
All right, so for the sea cucumber, do you want that in the section set, or do you want it in biobox?
Let's ask the biologist. Where would you
Or
like
put- yeah, or
the
in
sea
a quiver.
cucumber? In a bio box?
Yes, because they want to capture more than one.
Okay, yeah. Can
One
we
for
get a
you,
couple?
No, the one for you for microbiology.
Yes, one for me, too.
Guillermo, and one for.
Okay, yeah. Can we put those in a bio box and we'll collect a couple if we can?
Sí.
Because I want one too, if possible. Squeeze. Yeah, that worm was out of here. He was like, I'm not I saw what you did to that other guy. I'm sticking around for this.
Are we sure we're gonna find enough of these that I don't need to worry about assigning one?
Yes,
Any one person?
There's a bunch of them out in front of the ROV, so we should be able to get a few. The Gloctopus was meant as a cautionary tale, but it is just too rad. Now I'm gonna have to explain what the Gloctopus is. Oh wow! Yay! Okay, careful! Look at all those little appendages there around the mouth, anus. Mouth and anus.
2?
Yeah.
Biobox
So
2A.
you're gonna try to swim, see if that one goes. So.
Yeah.
Yeah. Oh, sorry, little dude.
Biobox 2A.
Suction sample.
Try to use the suction sample.
Top air, reach it 'cause it's back.
Right, Biobox 2A, and then if we find more of these, which we should, because it looked like there was a whole bunch of them on the floor there. Oh, we will put them all into the same box. Okay, wow, look at all that fluffy stuff. That's incredible. So pretty. I love that color.
Yeah.
Sea cucumber is offended. It's like, how dare you? Unhand me, sir. All right, very gently. There we go, little
Alright,
dude.
go closer.
Okay. And let's see if we can find a couple more of those up ahead. Grab a few.
Okay. We're gonna swap out.
Okay. Then,
Okay.
all right, we're swapping out pilots here. So stand by for just a second.
First things first. Slice through the bungee on. Scroll over and grab the. That thing is super, like it's really get up really delicate.
This is definitely going to become the Keith Jackson Pete chat. Good morning, everyone. Good morning. I'm so glad that a ton of my friends and loved ones are in the chat this morning on my very first dive. I feel super loved by all of you. You are wonderful. Shout out to my gorgeous daughter, Eleanor, who is also in the chat with her dad. She's 5. Hi, Ellie Belly. How are you? I miss you a lot. I love you.
When you're on the bottom, you can actually
Yes. And so it's very irritated in box stage. So.
All right, so we're going to try to get a couple more of those sea cucumbers. Looks like there's a bunch of them down here.
Yeah, they're gonna want.
That way we can share them. I would like to study the microbes that live in these sea cucumbers, in their guts. So I'm going to take a little sample for myself. Hi, Shoa. Josephina's here too. Josephina is my 7-month-old baby. Hi, little Joe. Miss you. Oh boy. Okay. Jax is gonna post this on Facebook, and a whole bunch more people are likely to join the chat. So I'll be here until 11 a.m. So have people come in and say hi to me. Miss everybody. All right, so we wanted to get that acorn worm and then some more of those sea cucumbers, correct? Is that the plan? Yes. Okay, let's grab that purple worm here, and then we'll try to get some of these sea cucumbers that are up ahead. And if anybody wants a core, just let me know. We'll take another core. But we are certainly finding some really gorgeous animals. That looks like a sea star up ahead, too. There's a brittle star down at the bottom. So yeah, if anybody wants anything, just shout it out.
Alright, so I'm gonna grab that worm, and where would you like it to go?
Yeah. Where should we put the worm? Bio box?
Yeah.
No será más fácil chuparlo. No te lo vas a quitar con la pinza. ¿Crees? Sí, se ve tan alargado la punta. Así de larga porque va a la vía.
Maybe with suction?
Right, we're trying to decide how to collect this particularly long worm that we have found.
You wanna try and suction it?
Long
Just grab
boy.
it, Put it in somewhere?
Yeah.
Suction?
Suction.
Suction.
Okay. Looks like we have everything except for number 1 open, is that right?
Yeah,
Looks like we're on 8. We're on 8 now.
yeah.
All right. Good morning to you. I am also excited.
Might have to push out.
So nice to see so many folks joining us. All right, we're gonna try to collect this really gorgeous looking worm that's directly in front of the RV. Ooh, fish! What kind of fish is that? Does anyone know? Good morning, Liz. Hi, Liz. Liz is another friend of mine. And there's my dad. Hi, Dad. Brian Seiler is my dad. Hi, Dad.
My recommendation, after we collect the acorn worm and the sea cucumber, to move on to another location.
Okay, so worm and sea cucumber, and then we move on?
Yes.
Okay.
Sea cucumbers too.
Sea cucumbers. So two more sea
Two
cucumbers. Got it. 1 worm, 2 sea cucumbers, and then we forge ahead.
And then we'll move on, yes.
Yeah, there's probably lots more to see. We could probably stay in this one spot all day and find beautiful things. So. Well, we are over the moon to be here and to have you. The fish is a rat tail. Says in the chat.
Nice.
Hi, Willa. Willa is my friend, and Virgil is her baby's son. They are also in the chat. Looks like we just got that worm we were eyeing. Very good. So many friends saying hello.
I think it's inside.
Hi, Willa. Hi, Virgil. This is also on Twitch. Yes, we are on Twitch, and we're on Facebook too, for folks who use that. So, yeah, we are live in multiple places for those who want to join us. All right, so we collected that worm. We're going to get 2 more sea cucumbers, please.
So, and the acorn worm is where?
That was in the suction
Jar 8.
sampler. Jar 8?
Jar 8.
Jar 8 for the worm.
Is that
This,
suction sampler 8 then?
Yep. Okay. And this sea cucumbers, you want to suction them as well, or grab them? Or
We grabbed them before and put them in bio box 2A.
Okay.
So if they can all go in the same box.
This one right
Oh.
in front, right?
Yeah. Yep. This guy, pink guy. And then there's another one, I think, a little further along that we can also grab, so that we have 3 of those.
And the other 2 red cucumbers are going to go in bio box 2A?
Yes, they're all gonna go in bio box 2A.
Gotcha.
Nice little sea star over there on the bottom. These sea cucumbers are really pretty. I like the color.
Not too far. Oh.
I think you're probably good. Oh, it's moving. It's on the move.
Okay.
There we go. Oh.
Hey.
Whoa.
Perfect.
Okay. It's probably like
I got one.
So
no,
squished.
the other way.
Ooh,
I tried not to
squish.
squish him too much. But
Yeah.
I got him a little bit.
It's all good.
Stop you as soon as I got it.
So this was
Science.
in box
Yeah.
2 if we open it.
So Biobox 2A, we already have one in there, so they can all go in the same box
Just
together.
open a little bit, look down with this camera a little bit, and then if we see him starting to escape, just close. You don't have to like that.
Yeah, these guys are feisty, so
As soon as we see it, if he starts escaping, just close.
all right.
That's good for you.
Alright, creeping in.
Put that in as CQ number 2 of 3.
Yeah, 2 of 3. Yes, we're gonna get one more.
I don't know what this guy's gonna do either, once I let him go.
Is that your wife, Drew?
It's gonna wiggle. It's gonna wiggle. It's gonna go in.
What's my, who is it?
Heather?
Here we go.
Heather is my, well, my wife's name is Heather. Hopefully it's the same Heather.
Well, I saw Heather Palmer, and I thought, what are the
That would
odds?
be my wife.
Okay. Hi, Drew's wife. I figured, I wanted to make sure, you know.
Alright, we got 2.
There's another shrimp.
That was close.
All right, now we need one more sea cucumber in the same box, please.
Even though I see a starfish immediately up front, I suggest to collect the third sea cucumber and move on.
Okay, third sea cucumber and move on. We will not be collecting the starfish. We will just say hello. Yes, the sea star we will not collect. We've seen a couple of really good brittle stars. Tons of these foraminifera.
Yeah, the seafood are very common.
Yeah, okay. Well, that's cool that we're getting a bunch of these. There's another acorn worm over there too, if we want to grab another one.
Where?
To the right. Over here. If we're interested in getting more than one specimen, which is probably...
That would be.
Let's grab this sea cucumber. If we want another acorn worm, we can
I
definitely discuss
don't mind
it.
if we collect another one. Anybody wants? I support that idea.
I'll take it.
Okay.
Can you
Watch it.
out or push in a little bit?
Okay, so we're gonna take another sea cucumber, pop it into the same box, so we have 3 now. These guys are feisty. Not that I blame them. I don't want a robot arm picking me up off the floor when I'm hanging out, sticking me in a box.
That one guy swimming there. See him? You have to be really careful with this.
Oh, look at him wiggling.
Okay, open it up.
All right, just gonna pop that in the box there. Yeah, we've got 3 sea cucumbers of this type that we're collecting in this one box. These pink ones. We also collected another one that was a different species earlier in the dive. We only have one of those.
Get him open, see what happens.
All right.
Sorry. He's in. And now.
He's in.
Yeah.
There.
And confirming 2A. So I got all 3 red sea cucumbers in 2A.
Okay. Do we? Let's try to get that worm there. Can we put it in the same suction jar in 8, or do we need?
Are those things very mobile or not really?
No.
No? Yeah, if they're not mobile, yeah, we can.
Okay.
But if you, the chance you would take is if you go back to the same jar, they can go out, you know?
Right. Okay.
As long as it's not real mobile, it's okay.
They didn't seem to be particularly mobile, so not like the sea cucumbers that are super wiggly. All right, so we're gonna collect this, what we think is an acorn worm. So now we'll have 2 of those. As you can see, the bottom here is incredibly fluffy. Every time we land, we kick up a whole bunch of this particulate matter, and that's because we are under a huge oxygen minimum zone. It's very, very productive here, so there's lots and lots of sinking biological material. As we were coming down, we saw lots of what's called marine snow, which is just like clumps of biological material. It literally looks like snow falling. And the floor of the ocean here is just covered with it. So the tops of our push cores are going to be super fluffy. All right, we're going to get
Coming out?
this
We're in the
worm.
same jar, right? 8 or whatever.
Yeah, let's put them in the same one, I think. All right, so another
If you can't see, we can just.
acorn worm is going in suction sampler 8.
Suction sampler, so we'll have 2 in there.
We'll have 2.
Did we do 7? Because right now we can't see it to change it.
Okay, let's do it in 7 then.
Coming out?
7, Drew. Suction sampler 7.
Got it.
Thank you, thank you. All right, and let us move on, I think, from this spot. Yes?
Yes.
All right, let's move ahead.
Let's explore really people.
Yeah, let's keep going.
So you're going down the line, right?
Yeah, we have a waypoint
Good point.
that we are heading towards, I believe.
This way.
We're sort of following a little ridge here, kind of going up and around the ridge.
domes that you have at the left?
Oh, these little mounds here?
Yes.
Yeah, let's
What they
go
are?
over there and see what that is. Hi, Ian. So many of my friends jumping into this chat. It's very exciting. It's nice to feel so supported. You're all here for the love of science. It's great. If you're just joining us, we are off the coast of northern Chile. On land, we have the Atacama Desert, the oldest and driest desert in the world. And we are close to the Atacama Trench, which is over 8,000 meters deep. Right now, we are at around 2,900 meters, 2,922.
In the month, that would be very interesting.
Temperatures 1.81 degrees Celsius, so just a little bit above freezing. Nice and cold.
That would be good.
Lawrence?
Yes.
Can you get that?
What are we looking at?
We are suggesting a core in one of those mounds.
Okay, we would like a core in one of those mounds, please. Push core.
Nay, bother. Looked like that core earlier came out really good.
It did. I was a little nervous about all this fluffy material, but we did it.
Almost ideal for coring, you know?
Yeah, good for looking for lots of stuff. Dr. Palmer, you have a fan tuning in from Florida.
A what?
A fan in the chat. It says, "Go, Dr. Palmer."
Oh, okay, cool.
Who is it?
Somebody from Melbourne, Florida.
Melbourne,
Okay.
Florida. Maybe you want to introduce you? Kevin.
Who's saying?
Oh, okay. Hello.
All right, let's get a core from this mound. See a brittle star down on the bottom, another shrimp. Lots of these foraminifera that look kind of like lettuce on the bottom. We've already grabbed 2 of those. Is that an anemone up ahead?
Do you guys care what core I use?
You could do 2.
2 if you could.
Yeah, let's do 2. Yeah.
For you? No.
Oh, Jax. Okay.
All right, and where exactly? Right in the middle.
In the mound, right in the middle? Okay.
Kind of right where I am.
Yep, like on the
All right, we're taking another push core. That is definitely a sea anemone back there.
I see.
Upper left. if anybody wants it.
Okay, right here?
Yeah, that's fine.
Looks
That's fine.
good.
Yes, this is just fine.
All right. So we're literally just going to take this plastic tube and push it down into the bottom of the ocean, into the seafloor, and pull up a big plug of sediment for sampling. There it goes. This is our second push core on the dive.
Easy?
It is metal. The claw body is metal. It's titanium steel,
Yeah,
so
this
it can
is
withstand
perfect
the
setting.
pressure.
Yeah, this is quite good.
And you see how it gets a little anoxic, huh?
Yeah, look at that. That's really cool.
Also, the upper 5, 7, whatever centimeters are nice and
Nice and fluffy for you.
yeah.
Yeah. So Nick is hoping for lots of fluffy material to look for myofauna.
Good now?
Yep, perfect.
Thank you so much.
I'm gonna put it partway in and then let
Hi,
go and let it fall to
Chris.
the bottom. Is that
Another one of
okay?
my friends. Hi, Chris. How
that
are
way
you?
it's self-aligned
It's true. Ain't no
with the
laws
seal?
when you're using claws.
Okay. All right, I'm gonna let go.
Alrighty. Oh! Oh!
That's okay, that's okay. We didn't lose nothing.
No, still good. Still good.
I commented that it was cut from a dome. But it's just to give you a little bit more frame of reference.
Good annotation.
All right. Drop that bad boy in there.
Everybody knows fire check.
So this is, I mean, we call this a bioturbation. That's the appropriate term. But here, biogenic dome is fine. But,
Claw in order. Oh my god.
Keep with these upgraded ones, you can, you obviously have to push it out now.
Oh, okay,
Because
that's
that's the
all.
way it's done.
Yeah, we think those are all xenobiophores. All this lettuce-looking stuff on the bottom here.
Okay.
We've collected two.
Yep.
All right, what is that directly in front of us? Can we zoom in? What is that? Sea urchin? Sponge?
it looks very similar to the specimen we collected before, a yellowish.
Do we want to get that one too?
Guillermo?
There's also a little pink something over here to the right under the xenophile. We are watching exactly what you were watching at home. So,
Oh, that's what it came
I mean, this has to be something.
yeah, what is that? A little pink thing.
I would say look at it. This is the look. After this, we should move on. I was told that you want to move on. After that, the sea urchin.
Okay, so we want to grab
You wanna grab the other sea urchin?
this sea urchin here? Or looks like a sea urchin.
Where's that gonna go?
You're gonna go.
Yep. Can we take that? Where would we like to put it?
Nick, ¿podría ir con foraminifera para ocupar espacio?
No. Excuse me, Armando, what do you
¿Puede
what was
ir
the
con
question?
los foraminifera para no ocupar nuestro espacio? ¿Ese aviso?
We can put it there, yeah. Or wherever we put the other. Yeah, we can put it there. No, that's a sea animal.
Do we want to take the sea anemone as well?
Guillermo, ¿quiere? The sea animal. No, no the sea animal.
No? Okay. We're not taking the sea anemone. We are taking
Nick, decide dónde quieres poner la...
this thing.
Where do you want
It
me
is,
to go?
yeah.
¿Lo puedes poner con los foraminifera
Yes, the
para
water
la-
temperature is only just above freezing. It's 1.8 degrees Celsius.
We can put
Pretty
the biomass
cold.
with the foraminifera.
More foraminifera?
The sea urchin, put it in the same box.
Oh, put it in the same
Yes.
box.
The foraminifera.
Okay, so this goes in the same box as the xenophiophores. Which box was that? Box 1?
Collection.
We have 1A, 1B, 1C, and 1D.
Okay, that was box 1A. A?
1A?
Okay.
Finished.
The xenophyophore, was that box 1A?
Yeah, no, I have a sea cucumber in 1A.
Oh, okay. Box 1D?
1D.
1D.
I think because we already have 2 urchins already.
Okay, going for it.
Okay, so yeah, we'll put that in the same box as foraminifera.
This guy was pretty easy to grab, was he? Just no special,
Yeah, I think we should be able to just pick it
not
up.
super duper delicate or anything?
Yeah. That anemone is really pretty. Okay. So bio box 1D.
But the
1D,
250
right?
don't,
1D. Yes. We have another shout out, Dr. Palmer, to you and to Katie.
Oh, hey, Emily. Emily is the undergrad who works in our lab with Katie.
Hi, Emily. Alright, I'm just gonna ease that on down in there. Cool. There's another acorn worm. Do we want to add that to the collection of acorn worms? So we have 3? Or no?
No.
Nope? Okay.
No.
Let's explore.
Alright.
Yeah, let's explore.
Alright, let's keep going. There's another sea cucumber up ahead. I don't know if it's the same species as the first one we found. If we want to take a look at it.
Yes.
You wanna go ahead?
Yeah, let's keep going.
I think we're supposed to be going this way. That's all.
You know, Lauren, it's okay that we're going away from the line, right?
Well,
Yes,
If we
as
wanna-
long as we're in the region,
well, I think we
This
should
way.
try to stick to the line if we're...
This is the line here.
Oh, okay. Why don't we...
let's go back to the.
Yeah, let's turn around and go...
Okay, I'll go ahead and put a ship move in.
Yeah.
Yeah. Just started seeing cool
Bridge,
things and
we are
just
gonna do
chasing them
waypoint.
down. So yeah, let's turn around and go towards that line. Thanks for keeping me on task.
Bridge, ROV. Bridge, ROV.
With bridge?
Hey, buddy, you got me?
I do, yeah.
We just put in a new waypoint for you.
New waypoint, thank you.
Thank you.
Okay, so while we're moving,
Yeah.
I would like
Where's
to
that
talk
big thing?
a little bit about. That's it. We have 3 of these.
Yeah, we have 3 of those. What about
Spa-
this big fat guy?
Space management. What's that?
Space management. Okay. No, another
So
fish.
we have not used at all the coral quivers,
Mhm.
the 8 spots.
Mhm.
We have used several of the suction samplers. I don't know how many. And 2 of the 8
You wanna see that
cores.
stuff?
Do we want to see that sea cucumber down there?
The squat lobster here. Did you see?
That's Guillermo's specialty, the squat lobster. So we should
Oh,
try to
okay.
get it.
Should we get that?
Is that the same thing?
The lobster? Where was it? We moved. Alright, we can get one of those for Guillermo if we can locate it again.
That's right. Yeah, yeah,
Ahí tiene sedimento usado para la roca.
Let's go to the rocks on the left, please.
Two
This one here?
There's rocks on the left side.
rocks on the left side.
Yeah.
Okay. That's a
Those are the
shrimp.
type of places where you may find brachiopods.
That's
I think we're really
Okay.
passing.
All right. Well, we're turning around, so we will head up towards that rocky part. Yeah. What's that? Over there.
That's probably a exact.
What?
Oh wow.
What is that?
Oh, that's a
Sponge?
Ooh, a sponge. Cool sponge. And another sea cucumber, different kind.
Let's collect that sponge.
Do we want to collect that sponge?
Please, please.
Let's collect that sponge.
I think he wants to collect this one.
I think he wants to collect that sponge. Let's get that sponge.
I want also to collect.
What's the bathymetry here? We're at kind of a flat part at the bottom of a sort of a cliff.
Like a piranha?
So,
With the,
Take it with the clams, please.
with the clam in the base.
with the clam in the base. Okay.
No, it's not. It's this.
All right. We're gonna try to get this. Wow! Look at that.
No.
Assyrian.
Acquire sponge.
Yes.
All right. We're gonna have to wait for the dust to clear a little bit.
Where did you all want this?
Where should we put it?
It's pretty big. So.
Yeah.
Biobox 3?
Biobox 3.
Yeah, we should follow that suggestion.
Yeah, Biobox 3 is, we got 2 empty ones there.
Okay. Biobox 3, A or B, either one. They are empty.
You want to list that as a sponge?
¿Qué es lo que es
Carnivore sponge.
Sponge.
Carnivorous.
There's a ton of particulate matter here because we are in a really productive zone, so all of that productivity is sinking in the water column and coming down to the bottom, creating all this fluffy material. So every time we land, we have to wait for it to settle so we can see again. All right. Okay, let's grab that for Guillermo and put it in Biobox 3. So many cool looking sea cucumbers down here. Just tons of them.
And this is the filtration where they get the food, maybe.
To the cata. Here.
All right, we're gonna grab this base. Ooh.
Oh, wow.
Yeah. It's rooted and it's
Okay. Very, yeah. Wow.
Cut that guy smooth off. Okay.
Yeah, wow.
That is-
Very
is
fragile.
delicate.
Yes. Yes. Very delicate.
Bag of water.
Tunicate. Big, big tunicate.
Let's go here.
Biobox 3A.
Box 3A.
huge. My gosh. See if we can like fold them up in there.
Yeah, I do. I have to.
Yeah.
Literally.
Wow. Woo. Okay.
All right. Get out of there.
Thank you, Zach.
Okay.
Oh, 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 oh.
It's okay. Don't let him get out. Don't let him get
Oh.
out. Close. Oh. And get repositioned, and I'll grab, push him in there.
Ooh. You like push it back in there.
Just a little more.
Yeah.
A little better angle. Okay. I'm ready.
Little nudge.
You go what?
Push in. You hit in the top.
Oh. Floating out. There we go, yeah.
Try that.
Yeah. Woo! Nicely done.
Will you try to get the hold as well?
The what?
The foot, so to speak.
Oh,
the sound?
the base of it?
The base.
So how to pull on now?
You could try. Do you want to try to grab the
Sure.
little
I don't know
chunk
what
that
happened.
broke
I'll
off?
go on.
It's
Yeah,
gonna- it's gonna
we
break
can
it.
try that. Cool.
If you can, you can. No?
Yeah. There's a little octopus hanging out on the bottom there. There's another one.
Could you use the scoop better?
Give me a little flip. The suction.
It's a suture from the red one.
Okay, we're gonna see if we can get the base of this that broke off. I don't know how deep down it goes. Yeah, nice.
With us.
Fantastic. Excellent job.
In the same place?
Yeah, let's put them all in the same
The
box.
sediment is okay too. But
Yeah, that's fine, I think. Okay, bio box 3A.
Box 3A.
And then we can continue to
To the
turn
box.
around
Yes.
to the track. Sounds good.
This guy trying to creep out again. All right.
All right. Okay.
Let's invent the rock now, Lauren.
Yep. All right. Gonna keep turning. Trying to get over by the rocks. And keep them to our right. Is that a cephalopod on the bottom there, or is that another worm?
But right in front of us is
Yeah.
another acorn worm.
Another acorn worm. Cool.
There's something off to the left of us.
Yeah, can we zoom in on that real quick?
We're gonna go there.
Right there? On the left?
Looks like there's something sticking out of it.
Yeah.
What if my rock move?
Oh, that's one of those big sea cukes like we saw earlier.
Oh,
I wonder what that spike is. Wow.
that's
There's
cool.
a white one to the right as well.
Yeah, very cool. Very cool. That is a big sea cucumber. Dang.
Can you say something? No, I'm saying.
Pretty color, too.
This is
Purple's my favorite color.
the pit of the mother.
See.
Wow, look at the.
Oh, look at that white one. Super pretty.
That looks like one of the first ones we grabbed, I think.
Steel, yeah, beautiful.
Very pretty. Yeah.
Lauren, sorry, I will go
Yep.
in the direction of the rocks.
We are. All right. Let's keep turning and go towards the cliff on our right hand side.
All right, moving along towards the vessel.
Yep.
You start drawing you in.
Okay, if you see anything interesting, you can consider this desolate for an ocean floor. Not for where we are, considering how deep it is. There's a lot of stuff down here.
This is a lot of life.
Yeah.
For that depth.
Quite a bit of life considering, yeah.
I'll stop in a minute. Fish coming up.
Well, I'm guessing because this is such a productive area.
See it?
Yeah.
What's that?
Oh, big fish on the left over
Yeah.
here.
See it?
Zoom in on that real quick. Get some footage of it.
Oh, that's another one like we just got. That's another one.
Oh, it's another one of those
Tunicate.
tunicates. Jeez. Okay.
Josh is playing heck with our focus. Sorry.
Oh, okay. Oof. Okay.
There's another one back there. These things are all over it.
Yeah, wow. Hmm?
Sea star.
Sea star. What's that down there? Rock?
Yeah, we can examine those rocks a bit more carefully.
Okay. Let's take a look at the rocks.
See that?
Just to see anyway.
Stuff in here?
Yeah, can we take a look at the rocks? Cool. Thank you.
Light rocks.
You can see some sea stars on these rocks here for sure.
And then we could get a piece of rock for the geologist.
The camera keeps working. There we go. Ooh, pretty. We do have geologists on board. We have a few geologists on board who are interested in rocks and sediment cores.
Can we zoom in to the whitish,
Yes, please.
bivalve looking?
Right there. This thing?
Yeah.
Okay. All right, we are zooming
Okay,
in.
right.
I'm gonna have to go mess with this, I think.
That one.
What's up a little bit?
Is that a brachiopods?
Seeing some little worms and stuff. What is? Ooh.
What's that?
A sea pen, a really tiny sea pen there. And do we want to try to grab that guy? What is it?
Is that right?
Small sponge. Okay.
If you could get that piece,
Do you want to take it?
so to get that,
Sure.
and the little tube one you have, whatever that
Yeah,
is.
this like sea pen looking thing. Sure.
If you could get that full
Okay,
piece
can
of
we-
rock,
can we
so
grab
the
that?
rock can be checked with the geologist, and
Yeah,
then
look at the.
and then you have a something else.
Oh, there's
Yeah,
little
also-
shrimp on there too.
Yes,
Okay, can we grab that piece of rock, please?
So, yeah, I'll give it a tap. It may be a bit hard to.
Okay,
not
let's
gonna work out.
give it a shot.
Yeah. That's pretty close to this.
Alright, we're gonna try to grab this piece of rock with It looks like a little sponge on it. And a teeny sea pen. Lots of good brittle stars down here. And anemones. Oh, it's breaking apart pretty easily. There. Okay. All right.
Nice.
Rock box. Where would we like to put this?
That's for the geologists.
Rock box.
No, because I had a lot of animals as well.
Sure.
Can go in a rock box, I think.
I think it's so small, honestly, it might fall through the rock box.
Oh, okay.
Okay.
I would put it somewhere else, probably.
Coral quiver?
Here. You want a coral quiver?
Yeah, let's put it in the coral quiver.
Happen to have one open over here.
All right. Which one is it? 3?
Yeah.
Coral quiver 3.
Yes.
All right, coral quiver 3. All right.
Okay.
And then they said the lid for that, they cut the rope on that, right?
Yeah, that isn't. What's one?
The lid's in the bottom, so I'll just use a different lid then, this front lid. That's okay?
Sure.
I think so, because it's going to be number 3, but we have the lead of 1. Okay.
Oh, Pete. All right, let's continue
We have
to
a request for a scoop of sediment
scoop of sediment. I gotcha.
right next to the structure.
Somewhere you can see some loose rocks.
A scoop of sediment next to that rock
Or an area with loose rocks.
or an area with loose rocks. Okay.
What we want to look at. Where's this scoop gonna go?
Where would we like to put that?
We'll put it in the
Coral quiver? Yeah.
If it's possible.
Okay.
In the what?
In the coral quiver?
Okay.
Let me mark that real quick. We did that.
We will try to catch the shrimps.
The shrimp? Yeah. Okay. We also have a request to try to catch a shrimp. I'm guessing with the suction sampler for that.
Alright, and does it matter where this comes from, this scoop?
I guess it's close.
No, it doesn't.
To
It's for
the-
the geologists, yes.
Yep.
Just gonna set it down and regrab.
Yeah, that would be just fine. Anything with loose material,
Okay.
ideally.
All right, we're grabbing a scoop full of sediment here.
So dusty.
Yeah, there's a lot of fluffy organic material down here. We'll do the best we can.
Just want to clear a little bit and I'll scoop. Okay.
there any questions in the comments? Anything that we should maybe discuss?
Someone asked if there's a jelly floating on the left.
Okay,
It's kind of hard to tell.
probably.
I think I've seen some more ctenophores, but smaller than the one we saw on the way down. It's hard to tell with all of this fluffy stuff in the way.
Well, the fluffy stuff are the ones that will give us the highest number of species by
Oh
far.
yeah, for sure. Yeah.
Well,
That-
metazoans, because
That
the
that looks
microbes
like a ctenophore there, floating along on the left. Maybe.
Oh, that looks better.
Okay, we got a nice scoop of sediment here. That's going in a coral quiver?
Looks like it would be interesting.
Okay, that's going in quiver 1.
Quiver 1. We're gonna call this geological sample.
Yeah, I've already assigned it to the geologist.
And then Gil asked if we could try to get one of those shrimp,
Right.
if possible.
I have a request from Oscar for a sediment core between the two rocks.
A sediment core between the two rocks. Okay.
Two cores, yeah.
Thanks, Chris. Have fun at work.
Okay, no, that's okay.
We are not on the abyssal plain. We're actually on the slope down into the trench. So we're kind of on a little flat bit here, but if you look at the overall bathymetry, it's pretty steep going down into the trench. We're just happen to be at a spot that's a little leveled off right now.
All right, so let me put this away and then.
If you don't find the shrimp immediately,
Put the lid on it?
there is a request from geology, Oscar,
Yep.
to get a sediment core between the two pieces of rock that we have ahead of us.
Okay, so we want a shrimp and a sediment core from between the two pieces of rock up ahead for geology.
Nikolas, can I put this back in its
So I guess we grab whatever is easiest first. There's a shrimp up ahead here, I think. Kind of hanging out.
Yeah.
Where's the shrimp? Is that?
That was up over in this direction, I think. Right there. Is the shrimp? And then I think that's where Oscar wants to take a sediment core as well, in between the rocks over there.
Lauren,
Yes.
do you think that rock that is seems that is not a rock, but carbonate concretion? Could we put a push core there? Try it, at least.
A push core where exactly?
But the rock, it seems that is not rock. There are carbonate concretions.
Oh, okay.
So we could.
Can we try to get a core that
Yeah.
that captures that? Yeah, we could see.
Yeah.
Is that a second?
At least try it.
Okay, because it
So-
did come apart
Yes,
pretty
that's what I said.
pretty easily.
Just to clarify, is that the same core what we're gonna collect for Oscar, or is that a different core?
Yes.
The same one.
The same one.
Same.
Same one.
That's, we're just clarifying what the core's gonna be.
Okay.
Yeah, that's the same one.
Okay.
Bingo.
Now we have to wait for all this fluffy stuff to clear up.
I'm gonna run check that camera real
It's
quick.
like pea soup.
We're gonna lose it for like 10 seconds, okay?
We're gonna
Okay,
lose.
sounds good.
We're gonna lose a camera for a few seconds while we're checking on something. Let the dust settle here.
Lauren, I'm gonna
Sure. Yeah, let's take a look at the bottom. Show everybody what it looks like while we're waiting. So last night, before we did our first dive, we did some mapping of the seafloor using our sonar. And you can see where we're at right now is kind of on this slope down towards the trench at the base of a little cliff. So it looks like we're at a really flat spot, but we're actually not. We are on a slope here. We're going to try to climb upwards a little bit where the slope is not quite so steep, if we can get that far. See how far we get today. That is where we are. So depth is about 2,923 meters. So not as deep as Sebastian can go, but pretty deep. Temperature 1.18 degrees Celsius, so just a little bit above freezing. So we're going to try to make our way towards that steeper spot up ahead in red.
Ctenophores could be high priority because there's so much interest in animal phylogeny. So a lot of people would pay attention to new species. There's a debate right now on, well, it's been going on for 10 years now, if ctenophores or sponges are at the base of the metazoan tree. And there are conflicting views with a lot of data too, from each side. So I would recommend that ctenophores, if we see them, we could go after them.
Okay. Yeah. Keep an eye out for ctenophores, for sure. Dust is starting to settle a little bit now. Fluffy stuff that we keep kicking up every time we move. We are in a really, really productive place in the world. There's a lot of upwelling, which means that cold, nutrient-rich water from the bottom is brought up to the surface.
Lauren,
Yeah?
a school friend of mine is sending me a hi.
Say hi. Yeah. An Aparna is saying hi to our Aparna. Hello. So yeah, love shout outs. Yes, we are in the bathypelagic, we're on the bottom, and the trench gets to more than 8,000 meters deep. The deepest that Sebastian can go is 4,500 meters. So we're going to do at least one dive at the maximum, at least that's the plan right now. But currently, we're only at 2,900. All right. So we're going to try to take a core here. The shrimp may have fled the scene in the confusion.
push core first.
But if we see another one, we'll try to get it.
Yeah. Until then, it was
It's a little tricky. A little tricky to get.
They want to push core between these rocks.
I think we're gonna try to capture, because we're not sure if it's rock or just carbonate, because it came apart so easily. So if we can get a core on top of the rock and capture that rock in the top layer for geology.
Through that rock, I'll try, but.
Yeah, I don't know. I don't know if we'll be able to get it or not.
The request is to try, as we said, and then if it doesn't work, just on the side of it.
Yeah, just to the side of it if we can't. And that would be core 3. Push core 3.
That's for Oscar.
I'd like
That's
to put
for
a lid
Oscar.
on that
Yes.
one,
Yeah.
coral clip,
Yes.
I'll never get a chance to, but it's a bit murky right now.
Oh yeah. All right. This is our third push cord.
Which arm is better for, or they have different functions?
Well, they're both good. This one doesn't have all that stuff on the end,
Okay.
so it's a little better for dealing with rocks or push cores. You can do either.
Probably equally expensive, huh?
Oh, yeah. No discounts.
Yep.
Okay.
All right, we're gonna try to get on top of that rock.
Yeah, I don't think it's gonna go well.
We'll see. If we can't, we'll just get as close to it as we can.
Like on this rock right here.
Yes.
Yeah, sounds good.
Push them a little bit.
Is it the rock or is it the rock? No?
Yeah, I don't know.
Okay.
It's like a bad game show. Rock or not rock?
Ah, okay.
Not decide.
Wasn't a rock.
Perfect.
It was a pretend
Wasn't
rock.
rock a rock?
Is that going in at an angle or?
Yeah, it kind of made me slide a little
Yeah.
because there's rocks that's hitting all the way in.
Right.
I'll make a note.
Nick, that to be full of animals, right?
That should be full of animals, but this is dedicated for the geology. I'll look at it, of course.
Horse?
Yeah.
Animals for you, rocks for them.
Yeah.
What do we got? What do we
Yeah, it's
got?
semi.
I think we just have a big rock in there. And not
Nice.
much of a core.
Nice.
That's what you want?
Yes.
Okay.
Yeah, I think this is good.
You do want that?
That's for
Okay. So you would want to use the centimeters or the.
We're expecting to be down for about 8 hours,
Yes, you guys.
depending on what we find.
We've got the extra ones.
And depending on how quickly we fill up all of our sample spots. So could be longer, could be shorter. O2 seems pretty constant down here as far as I can tell.
Lauren,
Yes?
after this, just keep exploring,
Okay.
trying to see new things.
Yep. Yeah, O2 has been around 155 micromolar. Haven't really noticed it changing much. It changed pretty drastically in the water column when we sent down the CTD, but down here I haven't noticed a huge amount of variation, although we're certainly keeping an eye on that. All right, shall we continue on our track?
while you,
Sure.
And we have been asked to keep an eye out for comb jellies, ctenophores. We can try to get them. Another sea star on the bottom, big purple one. What is that? What's that white thing down the bottom there?
Sea anemone.
Sea anemone. Oh yeah, yeah, big.
you want to stop?
No, we can keep going.
Guillermo, ¿quiere el sea anemone? No. No.
Con
Keep going,
los
I think.
associates, camarones. There was another big sea anemone.
Keep going along here. Now we're seeing some rocky stuff. When we first landed, it was all fluffy.
Ooh.
Now we're seeing some more rocks.
That's
If you wanna,
What is that?
Do you wanna-
Look like a sea pan.
do we want to sit down and take a look?
Yeah. Just, also there's a lot of rocks to
Where? Anywhere here?
Yeah.
Just below us, yeah.
Yeah, just below. Let's take a look at all this rocks. Yeah, we didn't see any rock before when we landed. It was all fluffy. Unfortunately, we did not get to extend the expedition because of the delays. So we are still ending the expedition at the scheduled time. So we're just going to make the most of the time that we have. We have a luck dragon on our side, so I'm feeling pretty optimistic about it. Tina Force. That's great. I love that. That's a really good stage name. All right. Let's have a look around. See what's on these rocks. Can we zoom in on this round?
The white thing in the corner.
Looks like a sponge.
On the left.
Here?
Above.
The bowl, yeah.
Just a little bit above.
A little bit higher than the
Brittle star.
little star.
So, to the left.
No, that's the
This guy?
foraminifera.
That's a foraminifera?
Yes.
No, I mean this one.
That's the
Oh, up here?
Upper.
This
Yeah,
here.
Yes, you got it.
Yes.
All right.
I don't see that. I see this.
We can zoom in on that guy. Yes, 7 days of diving is better than 0 days of diving. That is so true. All right, just gonna wait here for things to clear up. Any idea what we're looking at here?
Looks kind of like the thing we saw on that other rock we got.
Maybe. Good morning to you too.
Beautiful picture now.
It's very pretty.
This?
Yeah, what is that?
Has a right sayer, but I cannot tell yet.
Would we like to try
This is
to?
how you expect to find brachiopods, but I don't think that's a brachiopod.
Yeah,
No.
That's what I'm trying to do.
yes.
Anything
I know.
round?
Okay. Do we want to keep going?
We should look at these
White dots?
White dots?
Yeah, there were some white
Okay.
dots on the left.
All right, we can. Yeah, let's continue to examine these rocks. We're hoping to find lots of brachiopods on this dive.
Also, there is an interest to collect one of those probably sea pans.
Sea pans?
Those, white sticks, as we
Yep.
see them.
Okay.
sea log?
Well, I've had it sitting there for like 3 inches now because every job we do, like the first day it does this.
While we're waiting for the dust to settle, I think we're going to switch over to the next dive watch so I can take a break. But I'll probably be hanging out in the room looking for cool stuff, so I'm going to hand it over to the next person.
Trying to associate the number on the core too, but that's more important because it's sort of core fibers.
The one that the fiber comes out
This
this
comes on
time.
which one no water yet.
I think it's the HDMI connection.
Biobox,
You even go
push
back there.
cores, and then suction sample.
You go to the for sure.
Out.
Antonio, you want to switch over? Were you next or was it Parna next? Who was next?
I'm next on sea log.
Oh, you're next on sea log. You're next on narration. Okay. There you go. Probably pop back in after I've had a chance to take a short break.
Maybe. Because he knows overseeing the organized
That's the schedule that we
Oh,
do.
no, you didn't come at
Next year, so everybody can hear you.
Fire.
You're good. I'll be back in a little bit.
Hi, I'm Aparna Banerjee and I hope I'm audible to you all. So I work in Chile. It's the capital city is Santiago. So where I'm working as professor is somewhat like 300 kilometers south to this city, Santiago. And I'm in Chile for last 6 years. So I'm basically from India and I'm from West Bengal, from a city called Asansol. So I'm going to talk a little more in my native language, Bengali, so that part of the world can understand what we are going to do.
If it
Tami
takes
Aparna,
a while for the sediment to settle, perhaps we move around a little bit.
No, we're fine to move on. You want to move on?
আমরা আতাকামা ট্রেঞ্চ এক্সপিডিশনেতে আছি। আতাকামা ট্রেঞ্চ নিয়ে একটু বলতে গেলে যেটা বলা যেতে পারে যে আতাকামা ট্রেঞ্চ ফর্ম হয়েছিল 150 কোটি বছর আগে। 150 কোটি বছর আগে যখন ফর্ম হয়েছিল তখন নাজকা প্লেট আর সাউথ আমেরিকান প্লেট, এই দুটো প্লেটের মধ্যে একটা সংঘর্ষের জন্য এই আতাকামা ট্রেঞ্চ তৈরি হয়েছিল। 150 কোটি বছরের পুরনো আতাকামা মরুভূমি, আর 15 কোটি বছর ধরে আতাকামা মরুভূমি পৃথিবীর সব থেকে শুষ্ক মরুভূমি। পৃথিবীর সব থেকে শুষ্ক যে পয়েন্ট সেটা আমরা আতাকামা তে দেখতে পাই। তো আতাকামা যেটা মরুভূমি, সেখান থেকে আমরা মোটামুটি এখন প্রায় 300-400 কিলোমিটার মতো দূরেতে সমুদ্রের মাঝখানে, যেখানে আমরা এখন দেখতে পাচ্ছি আমরা এখন 2924 मीटर डेप्थ आছি। हम लोग 2.9 किलोमीटर डेप्थ में है और टेम्परेचर जो है वो है लगभग 2 डिग्री सेल्सियस के बराबर।
Armando is going to get
Thank you.
And you can see that the salinity is like 34.7 PSU. So
nobody else wanted cores? Just us?
the salinity is also very high deep down the sea.
Of course, yeah.
সমুদ্রের এতটা গভীর, we can see a very pretty animal over there, and I think my colleagues have interest on that.
That's,
Esa cosita
we
blanca,
know
Nick.
that species.
Nick, those
And we can see
this
a
one
fish
to the right.
down there
The white,
which
are you
is
talking about the white
moving and
spot?
looks
Check it out.
pretty interesting. সমুদ্রের এত গভীরে এত কিছু প্রাণ আমরা দেখতে পাচ্ছি। আমরা সামনে আরো অনেকগুলো প্রাণী দেখতে পাচ্ছি যেগুলো
for a suction. Get bad. No.
আমার কলিগদের ইন্টারেস্ট আছে এই স্যাম্পলগুলোকে এখান থেকে কালেক্ট করার।
So we could collect that with a clamp, please. The white round.
And now we are going to collect that sample over there.
Sure, you want to collect that and put it where?
That's a hexactinellid
In
sponge.
in a bio box.
Firebox?
আমার কলিগ বলছে দিস ইজ আ টাইপ অফ স্পঞ্জ।
Probably
আর এখন যেটা বায়োলজিক্যাল বক্স সেটা ওই স্পঞ্জটাকে কালেক্ট করতে যাচ্ছে। There we go.
B1,
Yes. প্রেসিশন আর রেজলিউশন যদি আমরা দেখতে পাই তাহলে আমরা স্পঞ্জটাকে কত ক্লিয়ারলি দেখতে পাচ্ছি। উই ক্যান সি ইট প্রিটি ক্লিয়ারলি and now
Try to roll
we are
down
going
with
to
the clamp into the sediment. There.
there we go. And we have got it. চারপাশের যে আমরা ম্যাটার্সগুলোকে দেখতে পাচ্ছি হিউজ অ্যামাউন্ট অফ অর্গানিক ম্যাটার্স এবং মোরাললেস এই অর্গানিক ম্যাটারগুলো বছরের পর বছর ধরে অর্গানিক প্রেসিপিটেশনের ফলে তৈরি হয়েছে।
This video is so sharp.
Yeah, it is. It's ridiculous.
¿Qué no está tanto? Ya tú bien, perdón.
চারপাশে আমরা যে পার্টিকেলগুলো দেখতে পাচ্ছি ঘুরে বেড়াচ্ছে এই পার্টিকেলগুলো সবই অর্গানিক পার্টিকেল এবং সমুদ্রের তলাতে এখানে অক্সিজেনের কনসেন্ট্রেশন এতটাই কম তার ফলে আমরা দেখতে পাচ্ছি পার্টিকেলগুলো এভাবে ভেসে বেড়াচ্ছে। সবই অর্গানিক ম্যাটার, জৈব পদার্থ।
BB1B.
Yeah.
Is that how we got this?
BB1B.
BB1B.
Okay.
And someone in YouTube we can see saying that the sponge is a Hyalonema species. So now we have it in the bio box. এই যে এই বক্সগুলো এগুলো হচ্ছে বায়োলজিক্যাল অর্গানিজমকে ক্যারি করার জন্য। সুবাস্তিয়ান যে রোভার, এই রোভারের ক্যাপাসিটি হিউজ। we can get biological samples, we can get rock samples for our geologist colleagues, we can get some sediment cores, আমরা এটাতে জলও কালেকশন করতে পারি সমুদ্রের তলার এত গভীর থেকে।
There's a piece right here.
And we can see the temperature is really low here, 1.8 degrees Celsius.
Did you notice?
Hi.
What's that?
It was literally like
Oh no.
30 seconds. They just
So I can see there is my husband and who is saying me a hi from Talca.
That's what
And
happens
please
on boats.
send my greetings to my 3 babies in the home, my 3 cats, and I miss them.
I'm just gonna double check real quick. Can we just do it one more time? Resetting now?
Sure.
And যখন এই রোভার ওর গ্রিপ দিয়ে সমুদ্রের তলা থেকে স্যাম্পল তুলছে তখন যেহেতু একটা হিউজ অ্যামাউন্টের জৈব পদার্থ আছে, এই জৈব পদার্থগুলো আমরা দেখতে পাচ্ছি ভেসে বেড়াচ্ছে। তো এটা সমুদ্রের তলায় মানে লো I can see my students also saying me hi. Hi Cynthia. Las personas observando el live desde Chile. Mando todos mis
Guillermo,
saludos
the broken pieces of the
desde
restaurant is still there.
nuestro barco. Nuestro ROV ya está en 3 kilómetros de profundidad de océano. I can see more of my students to say me hi. Hi, Catherine. Probablemente muchos de ustedes no pueden entender el idioma que estoy hablando a veces. Estoy tratando de comunicar la ciencia en los idiomas que he conocido para mí. Bueno, yo hablo bengalí y aparte de eso estoy tratando también de hablar un poquito en hindi, en inglés y español. I'm just trying to speak in 4 different languages. My mother language, we can see some nice animal over there. It's floating in the right, something over there.
I think that's somebody committee wants to look at the rat. Whoa.
And it goes away.
Seems like that's maybe
Yeah, that's, yeah, I think you're.
We were seeing a sea cucumber which is saying hello near our sediment course. একটা সমুদ্র শসা, এটা যেটা আমরা দেখতে পাচ্ছি ভেসে বেড়াচ্ছে, এটা একটা সমুদ্র শসা।
just lost all our HV cameras, so let's go check out that shit. I don't know why.
You see this white stuff? This white stuff here?
Yep.
You want to just pick up that?
So now we are going to pick up
This is connected.
a white stuff that is connected with the
That's like a sponge connected. And then like a together, and this is just a piece of it.
So now the gripper is going to connect the sponge that is connected with the sediment and the rocks over there. So we are going to collect that.
The stock is very tough and he may
এটা কালেক্ট করা একটু শক্ত হবে, তার কারণ
may
প্রথমত
need to be twisted.
এই স্পঞ্জগুলোর যেটা স্ট্রাকচার খুব সফট, আর আমরা দেখতে পাচ্ছি এখানে কিভাবে কানেক্টেড আছে একটা স্মল সরু একটা সুতোর মতো একটা স্ট্রাকচার দিয়ে। তো সেইটাকে এখন গ্রিপ করার চেষ্টা চলছে। এবং যেটা বোঝা যাচ্ছে এই স্ট্রাকচারটা অনেকটা ডিপলি বাউন্ড সেডিমেন্টের সাথে।
But your arm just like some sticky jaws into the tooth.
Yeah, good space, the base.
Yeah.
Perfect.
No! Refuses to live.
We can see some interesting structure over there, and what can be possible this structure is. And we can see some interesting foraminifera in the right of the right of the dive stream. We can see that it's very, very deeply attached, the string of the sponge. স্কুপটা তোলার চেষ্টা করা হচ্ছে, কিন্তু এটা এতটাই ডিপলি বাউন্ড যেটাকে কালেক্ট করাও ডিফিকাল্ট হচ্ছে। কিন্তু আমরা দেখতে পাচ্ছি যে এটাকে অ্যান্ড দেয়ার উই ক্যান মেক ইট, পাওয়া গেছে। নাও এবার এটা যেখানে যাবে
BB1B.
এটা BB1B বায়োবক্সের মধ্যে যাবে, and we can see how the robot is opening the biobox, and there it goes.
Don't worry, I will get your setting.
And of course the sponge comes up with a lot of sediments. So yes, there it goes, and you can see that the gripper is pretty perfect in taking the samples and dropping it in the exact location. So there it goes in BB1B box. So So we are now in 2,931 meter depth of
There's a white piece there.
Pacific Ocean in Atacama Trench. The temperature of the water is nearly 1.81 degrees Celsius. That is pretty low.
Wow. Yep.
And the leftover piece of sponge also, we are going to collect it in the bio box. And there it is collected. So the people who don't know me, to give an introduction of me, I basically
The
work on...
suggestion is to keep exploring for more rocky outcrops.
So my colleague's suggestion that we want to explore more of the animals. So where we were, to give an introduction of me, my work interest is extremophiles, particularly the microorganisms that are surviving in different extremities of life where we cannot think life can exist. We can get microorganisms over there and they are omnipresent. So like for example in the front we are seeing some kind of sediments. They are basically biofilm or microbiological mat of thousands of years. And in this expedition, that is something my interest is. So we are going to take the samples of the sediment and then we are going to make it grow in the laboratory. And what typical microorganisms or new microorganisms or difficult microorganisms we can grow in the lab, that's what our interest is. And how they survive in this kind of depth of almost 3 kilometers. When the depth is increasing, of course, the environmental pressure also gets increased. We can see that the salinity is high. We can see the oxygen saturation level is low. We can also see that the temperature is pretty low. So we can get a psychrophilic barophile probably in this kind of depth. And we can see some pretty nice sea cucumber in the front, and we are heading 242 degree.
You want to sit by this?
And it's nice to explore this stuff. So this time we are going to get some halophiles also. To give an introduction of me, আমার যেটা কাজের জায়গা সেটা হচ্ছে এক্সট্রিমোফাইলস। সমুদ্র হতে পারে, পৃথিবীর যে এনভায়রনমেন্টগুলোতে আমরা প্রাণ থাকে সেটা ভেবে উঠতে পারি না, যেমন উষ্ণ প্রস্রবণ, যেমন সমুদ্রের গভীরের যে পরিবেশ, যেমন প্রচণ্ড যেখানে লবণাক্ত পরিবেশ, যেখানে we can see some nice sediments here.
And then followed by one of those white hexactinelids.
So my colleague is saying that the geological course, they are going to collect it over here. We can see how the life is in this kind of environment. We can see also, I have a nice question in YouTube. Can extremophiles that live at this depth and pressure survive at the surface in order to be studied and replicated? That's a really extreme change.
Yes.
So yes, that's a challenge, but in the laboratory we have this kind of environment. We cannot exactly replicate the exact environment where the microorganisms are growing, but we can kind of mimic the temperature. We can kind of mimic the salt concentration, the micro and macronutrients that survive over there. And yes, we have earlier worked on Antarctica. So before this expedition, I have been just back from Antarctic expedition in Deception Island, and we have been working there in the geothermal samples. Deception Island is a active volcano, so we have got some pretty cool microorganisms from there who survive high temperature, low temperature, salinity. And so yes, it's difficult to make them grow, but we work on extremophiles and we have that kind of setup. And then I have another question, and it's like, what might cause the water to be so milky despite stirred up sediment? The water here, actually, it's We can see
Okay,
the
so the
sediment.
suggestion is, has changed, I'm sorry, so before the core or after the core, Armando?
Before, otherwise we're going to.
So we are having a discussion that we are going to collect the samples before collecting the core or after collecting the core samples, because when we are
So the suggestion is
taking.
to roll around this rachial crop.
With the camera.
So now the rover, we are taking the decision that now the rover will be moving around the field. So we were in the question that what is probably making up the field looking so milky. The first reason is we are in a minimum oxygen zone, so we can the oxygen saturation level we can see. So this is making up all this organic precipitation for millions of years. So the more deeper we go, the more these chances of organic precipitation. And also there is formation of marine snow, that is like some clogged up particles of the animal's poops, bacteria and some type of organic matters. So we can see sometimes them also. And that's a typical marine snow formation in the depth of ocean that we can see probably. So we can see some cool biofilm forming bacteria. That is our interest also. Can we share stream over there?
This stream.
Yes.
This if possible.
So we can see a pretty nice shrimp over there. আমরা ওখানে একটা চিংড়ি মাছ দেখতে পাচ্ছি, সামুদ্রিক চিংড়ি মাছ। And my geologist colleague have interest to collect this diced shrimp, and we are going for it.
Let's go for the two.
Yes, so we stay here because we do a core, a shrimp, and
The team first.
yes,
Yes. That's the one.
So we are going, we can see some.
Try to collect the red shrimp.
This is also to mention over here that this particular area of Atacama Trench have not before explored. So whatever we are seeing are pretty nice, pretty exciting, and all my colleagues and all the researchers in the team are pretty excited to see whatever we are seeing. আতাকামা ট্রেঞ্চের এই যে পয়েন্ট, এটা এর আগে এক্সপ্লোর করা হয়নি, তাই আমরা যেটাই দেখতে পাচ্ছি সেটাই আমাদের জন্য নতুন এবং ইন্টারেস্টিং। তাই আমার সমস্ত যারা কলিগ এখানে আছে ওরাও যে স্যাম্পেলই দেখতে চাইছে সেটাকেই কালেক্ট করতে চাইছে। जो भी सैंपल हम लोग यहाँ पे देख रहे हैं, जो भी एनवायरनमेंट हम यहाँ पे देख रहे हैं, अताकामा ट्रेंच की वो पहले दिखा हुआ नहीं है, पहले कभी एक्सप्लोर ही नहीं किया गया था। तो वो एक नया लाइन ऑफ रिसर्च जो है वो ओपन कर सकता है। लाइफ हाउ, कैसे ये जो जीवन है वो वहाँ की ऐसे एक्सट्रीम एनवायरनमेंट में सर्वाइव कर रहा है। सो दिस ब्रिंग्स अ लॉट ऑफ क्वेश्चंस ऑफ सर्वाइवल ऑफ animals and lives and its adaptation in this kind of extreme environments. And from wherever part of world you are seeing me, I'm sending you a hi.
Coming closer.
So this is like a common procedure that that the rover is going to fix it up, its position to collect that shrimp. রোভার কী করছে এখন, চেষ্টা করছে এখন যে জায়গাটাতে আছে সেই জায়গাটাকে ফিক্স করে ওখানটাতে বসার, যাতে পারফেক্টলি ওখান থেকে অর্গানিজমটাকে নিতে পারে। We have some question for you in the live. What was the animal-like organism that's off camera at the right? I think this one.
What do you think?
Look, the question that we are having in live.
I don't know.
Which animal? This is a foraminifera.
Okay. So my colleague Nick is saying it's a foraminifera that we are seeing in the right. Nick is a specialist in this kind of organisms.
You reach? Check on the closer.
No, it's like this. Okay. It is this right here. I think we're talking about the same thing. I'm just looking for this. What's the name?
have to find it.
Is it foraminifera?
এখন আমরা দেখতে পাচ্ছি যে কাইন্ড অফ রোবটের যে হ্যান্ডটা ওটা চেষ্টা করছে এসে এই চিংড়িটাকে ধরার। বেচারা চিংড়ি এতদিন ধরে এখানে বসেছিল, জানতও না একটা রোভার এসে দুম করে ওকে তুলে নিয়ে চলে যাবে। আর একটা দল সায়েন্টিস্টদের যারা ওকে ধরার জন্য এখানে বসে আছে, ও মোটেই ধরা দিতে চায় না, ও পালাচ্ছে, কিন্তু আমাদের রোভারও ওকে ধরতে চায়। রোভার ওর পেছনেই পড়ে আছে। Poor baby chingri. The visibility is pretty low for the organic particles over here. Oh. It is. The shrimp is pretty fast. So my colleagues have interest in getting up that. Sometimes the visibility is getting really low because as we can see the organic amount is so high that it's getting that visibility lower.
If we cannot get the shrimp, we suggest to do the geology core.
As we couldn't collect the shrimp ran away. It's the runaway shrimp. So we are going to collect the geological samples from this location. যে কোনো ধরনের এক্সট্রিম এনভায়রনমেন্টের দিকে কাজ করতে গিয়ে আমরা দেখেছি we came across a lot of difficulties. প্রথমত we have to have এই ধরনের একটা সেট আপ যেখানে আমরা স্যাম্পল কালেক্ট করতে, এখানেই যেমন এই রোভার তার পারফেকশন কতটা, যে 3 কিলোমিটার অফ ডেপথে সমুদ্রের তলদেশ থেকে সেখানে শুধুমাত্র স্যাম্পল যে কোনো জায়গাতে রয়, স্পেসিফিক্যালি বায়োবক্সের মধ্যে সেডিমেন্ট আলাদা করে, রক স্যাম্পল আলাদা করে, জল আলাদা করে কালেক্ট করতে পারছে। সো এখানে আর্টিফিশিয়াল ইন্টেলিজেন্সের একটা বিশাল বড় রোল থাকতে পারে, যে আমরা কোথা থেকে কীভাবে স্যাম্পল কালেক্ট করবো, দ্য ওয়ে দ্য ওয়ার্ল্ড ইজ হেডিং নাও। আর দ্বিতীয়ত হচ্ছে এই ধরনের জায়গা থেকে স্যাম্পল কালেক্ট করার সময় একটা ডিফিকাল্টি হচ্ছে, উই কালেক্ট দ্য স্যাম্পলস বাট when you go to make them grow in the laboratory, that's a kind of another difficulty. তাই আমরা যখন স্যাম্পল কালেক্ট করছি এটাই শুধু চ্যালেঞ্জ তা নয়, আফটার দ্যাট ওটাকে গ্রো করানো ইজ another challenge. So as we can see that now we are going to open the 7th number of box and tubes where we are going to collect the geological sediments.
So core number 4 goes to geology.
So this core number 4 that we are going to take is for geology. So I have a question in YouTube I can see from Shrobona. Hi. She has interest to know that this extreme condition under sea, less availability of light as well as oxygen, so what kind of microbes we can find? That's an interesting question. The
Yes,
kind
good.
of
Yeah, wherever you feel is safe.
Yes.
For the Cusco?
It's just for the geological samples.
Yes.
Yes. তো এইখানেতে আমরা the kind of samples that we are getting, the first challenge is of course light, because the light that is more impactful probably sunlight, because then we are getting the solar radiation. So the first option of making them grow will be definitely making them grow in absence of solar radiation, and also we can see that the oxygen saturation is pretty low. So we can make up a kind of anaerobic condition in the laboratory, or where the oxygen concentration is low to make them grow. And of course we can get some facultative aerobic bacteria living over there who can have some light sensitivity. So yes, we can get a lot of facultative aerobic bacteria in this kind of environments. আমরা কিছু অলিগোট্রপিক আর হেটারোট্রপিক ব্যাকটেরিয়াও এখান থেকে পেতে পারি, যারা এই ধরনের হাই অর্গ্যানিক ম্যাটার্সের প্রেজেন্সেতে গ্রো করছে ইন কাইন্ড অফ অ্যাবসেন্স অফ অক্সিজেন। So we can get a variety of microorganisms from here, not only barophilic but psychrophilic, halophilic, facultative aerobic or anaerobic microorganisms that have the capacity to form biofilm.
To collect an exactinated sponge ahead of us. Guillermo, what do you think about the tube anemone?
Yeah. What's what you think?
Now that we cannot see ahead of us,
We can see
if
in
we
the
possibly try
right
to
sea
connect
anemone
the
here
tube
also.
anemone to the right.
In the right, so my colleagues have the interest to collect that sea anemone. So now the rover is going to collect that sea anemone that is sitting over there. From how long we don't know. We can see a lot of foraminiferous here also.
Más o menos aquí, so the tube of the sea anemone extends deep in it, so it is likely we're going to break the specimen, but there's not much room to move around there with those rocks. So whatever
Yes. So
you get.
after this, can we head for some other area?
Yeah, we have to get a hexagonal for the museum, which is straight ahead, and then we
We
can
can go and explore some other areas. Yes. We can see a lot of organic particles moving around when we are collecting the samples. এই ধরনের স্যাম্পল কালেক্ট করতে গিয়ে এখানে তো রোভার গিয়ে স্যাম্পল কালেক্ট করছে, কিন্তু এরকম অনেকবার হয়েছে যে স্যাম্পল কালেক্ট এক্সট্রিম এনভায়রনমেন্টেতে যারা কাজ করে উই কেম অ্যাক্রস আ লট অফ ডিফিকাল্টিস। তার সাথে অনেক সময় রিস্কও অ্যাসোসিয়েটেড থাকে। একটা দারুণ মজার গল্প আর ভয়েরও গল্প বলা যেতে পারে এই সময়তে, যতক্ষণ রোভার গিয়ে স্যাম্পল কালেক্ট করছে। সো লাস্ট ইয়ার আই হ্যাভ বিন into Antarctica, collecting samples for an Antarctic expedition that I got a fund from INACH project in Chile. তো ওখানে আন্টার্কটিকাতে when you think about Antarctica, what we can understand, it's like everywhere it's snow and white. তো যখন আমরা ওখান থেকে ফিরছি জোডিয়াকেতে করে, তখন কাইন্ড অফ একটা ওয়াটার কারেন্টের জন্য হাত একদম ভিজে গেছিল জল ঢুকে। সেই রকম একটা কন্ডিশনেতে প্রায় 1-2 ঘন্টা থাকার পরে আমি হাতের কোনো সেন্সই ফিল করতে পারছিলাম না। তখন আমার মনে হচ্ছিল আমি ফিরতে পারবো তো আদৌ, মানে স্যাম্পল কালেক্ট করে কি আমি আদৌ ফিরতে পারবো, এইরকম একটা কোশ্চেনের সময়তে মানে আমি বললাম যে না, আমি আর জোডিয়াকে আমরা সেই সময়তে যে my colleagues
inside the sediment.
are giving some ideas to how to collect the samples more accurately with
Yes.
the tip to collect this
And it's probably the tentacles will
sea animal.
and
তো যেখানে আমরা ছিলাম তো ওখানে তখন যখন আমার হাত প্রায় ঠান্ডা হয়ে এসেছে আমি স্প্যানিশ মিলিটারিদেরকে বললাম যে না এই সময়তে আমি আর সহ্য করতে পারছি না, এবারে আমি দড়ি যেটা ধরে আছি ওটা ছেড়ে দেবো, কারণ আমার হাতের ভিতরে জল ঢুকে গেছে। আই স্টিল রিমেম্বার দ্যাট মিলিটারি উনি ওনার নিজের গ্লাভস খুলে আমার হাতে পরিয়ে দিয়েছিলেন। তো দ্যাটস আ কাইন্ড অফ কোঅপারেশন অ্যান্ড কোলাবোরেশন উই ক্যান অলসো আন্ডারস্ট্যান্ড ওয়েন উই আর ওয়ার্কিং ইন দিস কাইন্ড অফ এনভায়রনমেন্টস উই ওয়ার্ক অ্যাজ আ টিম অল। সেটা সেম এখানেও। এখন আমরা যেখানে ওয়াচরুমেতে বসে আছি সেখানে মোর দ্যান কারুর এক্সপার্টাইজ জিওলজিক্যাল স্যাম্পল, কারুর এক্সপার্টাইজ বায়োলজিক্যাল স্যাম্পলস, কারুর এক্সপার্টাইজ সেডিমেন্ট, কারুর জল, ফর এক্সাম্পল আমার মাইক্রো অর্গানিজমস। আর আমাদের প্রত্যেকের একটাই ইন্টারেস্ট, জাস্ট লাইক উই ক্যান সি ইন দ্য ইউটিউব for the tour of the ocean bottom. We all are seeing it, few people are seeing it live there, and we are going to collect the samples back when the ROV comes up. The visibility is pretty low for all the movement of the sediments, but if you wait and it gets precipitated, we can kind of see the bottom. And after we are done with this bottom, we will be going to the next place to see some more of the sea bottoms. We can also see some echinodermata over there, a starfish sitting. Guillermo?
Me taking this because I
We
think
can
this
see
is
some
what
echinoderms over there.
Let's rest.
We can see some echinoderm over there.
Sono fiore,
Yeah, study. You want to try?
fioroidea. So the brittle star.
Brittle star.
Yeah.
Do you have interest to collect that? No.
No, really.
Okay.
But this is interesting because never seen the fauna here in Chile.
This fauna that we are seeing, my colleague Guillermo is saying that we have never seen it before.
Never seen.
Que él nunca vio este organismo antes y puede ser una nueva fauna que nunca fue descrito o reportado anteriormente, puede ser.
Claro, no
Sí.
conozco nada de eso. Interesting.
Y por eso estamos por tanto tiempo parado acá para recuperar este animal desde este sitio. And our world is, like Nikolas is saying, our world is pretty interesting. We see, we have lives that we have never seen. We have lives that have never been reported earlier. We have lives that grow in pretty extremity. And you can see with how much perfection. Wow.
Yeah. From the.
It got the animal. Yes, we have
Do
it.
you want this?
Do you want a picture?
Wow.
Yeah, that would be.
Wow.
What is this?
And we can see how the animal looks. It's a transparent. And yes, my animal colleagues are very interested in this animal.
BB1C. I cannot expect that's going to be successful. But, yeah, this is
We have some question in YouTube. Is there a style stud behind the brittle stud?
Which one is it going into?
See.
After this, can you please focus the echinoderm that was there, because we have some question that behind the brittle stud, that echinoderm, the starfish, there is some animal that someone have interest in the YouTube to want to see.
Thanks. That's okay. Ah, that's a very, very good catch.
It was a great catch. And my colleagues are saying probably it has been never reported before. Something like this we have never seen. সমুদ্রের এত গভীরে এতে প্রাণ যে থাকতে পারে এটাই একটা অদ্ভুত ব্যাপার, তাই না? We kind of get goosebumps thinking that. And there it goes in our bio box.
The next suggestion is to collect the white sponge hexactinellid right ahead of us. We can barely see.
And after that, can we please focus behind the brittle star, because someone in the YouTube want to see that organism behind the brittle star.
Okay. Behind, like this brittle star, between 4 and 5. Aparna. Which brittle star? The one between cores 4 and 5?
Yes, behind that. Yes, exactly this. Behind this.
Okay.
That one, someone have interest.
Where are we focusing?
I think that one.
Oh, the little. Okay.
Yes.
Actually, that little coral is for the museum. Yes, the white coral.
Wow. So that's a coral that someone had interest to see. Yes.
Okay.
And you can see a lot of starfish over here. A lot of echinoderms. It's beautiful.
So does this person knows the group that asked? Does the person
Yes,
that
but
asked the
no,
question?
no. Just wanted to see it.
Yeah. It looks like stylasteridae, but I don't know if it's that deep.
My colleague feels that it's a skylasteridae, but he's not sure about what it can be.
So this is going to be very, very, Guillermo, this is going to be very brittle. Very brittle.
This?
Yes.
And the nice thing are they are like almost collaborating and cooperating while living, because the crinoderm and the coral is almost co-living together.
They are living. If we can collect the white coral, that would be great. It's going to be very brittle.
So my colleague feel if we can collect that white small coral.
Like that coral, but it could be hydrozoan, but just coral.
Do you want me to mark it like this unknown or today?
I mean, it can be both. There is a family that's called stylasteridae, and I think they make it deep.
I can include the for the unknown.
Yeah, well, okay. But it's definitely Cnidaria.
So you can see a lot of different sea animals over there. A lot of foraminiferous also.
Yes.
We have another one.
Okay. Let's concentrate on the brittle coral.
It's a very small one. Wow.
That definitely we need to go for this guy too.
Wow.
It's a family premium.
That's going to be easier to collect.
And
than
that's
the small one.
such a nicely formed structure of coral we can see. It's beautiful. It's beautiful.
It's interesting. One of the branches is partially dead.
Yes. I think if we hold it tight, they can break into pieces. something.
Yes, please.
Antonio, can we point that up? There is some orange biofilm we can see.
Sure.
In the rock. We can see some biofilm. And the microbial biofilm, normally they always form in surface. It's not possible to collect this piece. No, I think it's very soft, the sediment is. Wow. Can we take?
Look at this. The giant foraminifer.
It's looking beautiful.
That is an isopod.
Where?
And my colleague have found some isopods. That
Amphipod,
one,
it has the hump.
Amphipod.
Amphipod.
Yeah.
that's an amphipod.
it?
Or
That one.
Maybe.
Yes.
It looks.
This one.
Ah.
Wow.
now I don't.
Yeah.
That's
Paracarid. Let's go back a little bit.
Your amphipod is living with my biofilm, Nick.
So what's the next suggestion, Aparna?
iceberg.
We need to.
I think we should go forward and discover some more sites. If we cannot collect the coral, are you planning to collect the coral?
We would like to collect the coral.
Okay,
Yes.
so let's collect.
If we
some.
cannot collect the brittle coral, we go for the larger one, a
Larger
little
one.
further out.
Yes. Let's try and then we go for the next one, next site.
So,
I think the small one will be even difficult to hold. For
the suggestion is to go for the bigger coral.
The
You wanted to go for the big ones then?
Yes, sir.
the bigger
Okay.
one, yes.
Yes.
And it's a variety of life down the sea. It's unimaginable to understand the kind of life we can see down the depth of ocean, and how co-living the lives are impossible to believe. So yes, our interest is to trying to collect that coral, and probably then we will be going to. It's beautiful.
The best place to for the stock. I think the whole thing is gonna come off. Also, you see the caprellid amphipod, right at the base. It just.
Look at this.
Oh,
Brachiopods.
we're gonna stay.
So
We
my
have,
colleagues
we have
found
a
some
couple of very interesting specimens around here. Let's try to concentrate on the coral first. And I don't know if the viewers can see it, but there's a caprellid amphipod hanging at the base of the coral. It's on the kind of a fin on the right side with long antennae.
that one.
Yeah, that's a highly modified phytal amphipod. So I cannot point that out, but I think that some viewers were able to see it.
Do you want this, Nick? This one?
We want the coral.
Okay. So now we are trying to collect this up, and it's a pretty sensitive sample, so we have to be very gentle with them. তো এখন আমরা যেটা চেষ্টা করছি ওটাকে ওখান থেকে তোলার, কিন্তু আমাদেরকে অনেক জেন্টলি ওটাকে ওখান থেকে তুলতে হবে, যাতে কোরালটা অনেকগুলো পিসেতে ভেঙে না যায়, সেটারই এখন চেষ্টা চলছে যে যাতে আমরা প্রচণ্ড জেন্টলি ওটাকে ওখান থেকে তুলতে পারি। Is that an acorn worm? Because someone in the YouTube has written it.
That is correct. That's a hemichordate acorn worm.
Yes.
Can it be this one vertical stalk?
I see. It's very difficult to go to the base of the stalk. Yes, even a branch would be okay. Yes, I understand now. Yeah.
Hmm.
More brachiopods.
I think even the bunch will be falling out of the. It's difficult.
Yeah, it's gonna be.
Yes, it's true that so many individuals are co-living together. It's very, very cool.
Let's see.
So what we are trying to do is getting a piece of that coral, and let's see if we can get it. But it's getting difficult to collect it.
We're just removing the polyps. Yeah, it's tough.
It's tough. I think it will be almost impossible to collect. I think T22 will be available more
If
enough.
okay, maybe one more time. Then I have another suggestion.
It's difficult to take. It's kind of a stubborn little kid that don't want to listen to the parents.
Yeah?
We got a piece.
Okay.
We got, yes. Yes.
It's possible to take a piece of this rock.
No, right now we're going to the right one. What is that dive?
So this is coral quiver number?
I have it.
Two.
Should I share?
2. Thank you.
It's gonna have the
Balloon?
number 4 on it. Just like,
No, we have to collect something kind of
Okay.
very important.
nothing.
After this, can we move forward, please?
No, we're not going to move forward. We have to do something very, very important right now.
Okay.
Just a minute apart, okay?
Mm-hmm.
Just,
Cnidaria?
Keep going. See it down there?
Yeah, right there.
See it?
So now my colleague, he want to collect some more samples from here. I understand that for the geologist, it's a kind of event to see that many of individuals in this little part. We have a question. What kind of coral it can be? The species someone is asking. The coral that we have collected.
right now, soft coral.
Soft
Coral because
Okay. So it can be.
Octocoral.
Yes.
Octocoral. Okay.
So it can be octocoral that my colleague is saying.
Yeah, one of the trips.
Okay, so the next suggestion is just on the right side of the coral, there is a brachiopod.
Yeah.
And that's one of the most important groups we want to collect. It looks like a bivalve.
So beside
It's
this brachiopod that my colleague have
yes.
interest to collect this.
That looks like a
These are probably cut. It's better than
Yes.
I don't know its texture.
There is no
I'm wondering if it is easier to take the whole piece of a rock, which is not very tough with it.
So what we are trying to, because now the brachiopod is a pretty small sample, what we are trying to think that we are going to collect this. Okay, yes. So we kind of get an octocoral that you are asking, that my colleague have told. It's a soft coral, of course. I hope I've given you the answer. So yes, my colleague have interest to collect this brachiopod. But I think it will be kind of difficult to collect it over there, such a specific one. So what we are trying to plan is we can collect that whole piece of rock that comes up with the brachiopod, and let's see what is the decision that we will take. So yes, now we are trying to get that whole piece of rock to collect the brachiopod. So what we are trying to, by the time we are going there, maybe I can share some experience of me of working in other different kind of environments. So other than this kind of bathypelagic environment of profound sea depth, I have majorly worked on extremophiles from geothermal environment. So yes, I have interest on how the microorganisms they grow and survive in environment like hot spring, geyser, fumaroles, hot soils. So I worked before in my doctorate in India on hot springs. So that hot springs are also have formed due to some tectonic activity in India. It was located in West Bengal in a coal mine region, and after that I came to Chile for my postdoctoral work, and there I've started working again on geothermal environment, hot springs. And as I was initially talking that this entire Atacama Trench has formed millions of years ago due to a convergence of Nazca plate and South American plate. So this is interesting. Now we are going to collect that brachiopod from there. So we have a lot of seismic activity in Chile. To give an example, the most horrendous volcanic activity and seismic activity that happened 1916 in Valdivia was 9.5 Richter scale earthquake that happened. So this zone is seismically active zone.
Yeah.
Plates.
Okay.
So
That's it.
we can see now the brachiopods in zoom and in focus. That's what we are going to collect. So we have a lot of hot springs in Chile, and I have particularly interest in the biogeochemical environment
on the
of
substrate,
this.
or it has a pedicle, and not very tough, and nowhere near as tough as the corals.
So that that hot springs, there are more than 400 hot springs in Chile. So we work in central Andean mountain region in the hot springs, in south and in north in the Atacama desert. We also have fundings of Chilean Antarctic Institute, Institute Antarctico Chilean.
Yes. Wow, great idea.
And
Yeah, thank you.
wow, and I've got it. So I've been into Deception Island, Antarctica also, which is active volcano, and we have collected samples, and we are working currently in these hot soils and fumaroles. And yes, now we are in this Atacama Trench expedition where we are trying to collect that.
All in the city. All in the city. So
So we are going to collect those two brachiopods also.
The suction.
There, by suction, we are going to collect this up. These two small little organisms. And it's unimaginable to believe the amount of life that is there in that profound depth of ocean. So we are now just kind of trying to fix the point up and collect the samples, and there we go. There we are. So now we are going to collect it using suction. Oh, we can get that. Now we can get that coral.
Plus
This coral.
the brachiopod, which is right next to it.
yeah, all together.
It looks like a styluster, like. It's gonna break apart. I expected it.
My colleagues feel it's a styluster like coral, so it's going
Yeah,
to
for
break
the
up
pilot,
into
please forget
pieces.
the coral, it just broke apart in pieces.
I think we got a little piece of it anyways. There.
Okay, great.
What's the next one?
There.
We have a question in life that the eggs that are visible are for the acorn worm. The eggs that we can see.
Yeah, it's almost like it. Come on. Oh.
We have a question in life feed that the eggs that we are seeing are of the acorn worms.
And then you have another one at the left, at the same height, more or less.
To the left, you say?
To the left, yes, at the other side of the worm.
Okay.
The eggs that are visible there are of
You have
acorn
one
worms.
down there, and then one to the left.
That would be.
This one?
I don't know what this is. You have two of
Because
them there.
look, the question is that we can.
Just lost. Yeah, stand by.
Okay. Can we close?
Stand by.
Can we close it up here because we have a question from the live feed here.
We lost control of our phones.
Oh, it just closed. Wow.
Here we have it
Beautiful.
set up specifically so that it refreshes things.
Wow. Wow.
There are a couple of pieces for you. Sorry.
I didn't express myself well. Could you come back to the same spot?
No, we've got problems.
Oh, you have a problem. Sorry.
Yeah, it's back in there.
Okay. So you've got control, check. Yeah. Arms down, keep the hand there for a sec. If you kept turning left, so I'm gonna turn right. Okay. So I'm gonna bring back you up here. Okay, so we have a delta from 2. I need to get my screen back up. There we go. Let's have a moment to review.
What are you going to collect now?
Yes, you have control? Yes. You have another? No. Yes. Turn right again. So, yeah, here's our back position. I think you were sitting there pretty close. You can see where you are. I'll zoom in. Okay. Just give us a couple minutes to. Let's see. It turns on 0 right now. I'm not really sure about the launch points. There's that coral up there.
So we can see a lot of beautiful corals over here and a lot of organic particles swimming, floating over there.
Should be somewhere on there it is.
Kind of some organic raining. It's nice. Very, very beautiful.
Yes. Is that the one? That was in it.
Could you show us where you were?
Yes.
Yeah.
After the war,
Sorry, do that.
Are they on? Powered on?
So, yes, the time that the rover is moving, it's kind of taking up the visibility, but we are like in a in sand dusts, and we cannot see anything. I think the light someone have interest to see that pink slime stir.
After the
Okay.
Give us a second.
Okay.
Let the water get clear a bit.
আমাদের এখন সামনে যেটা দেখতে পাচ্ছি মনে হচ্ছে সমুদ্রের তলায় যেন বৃষ্টি হচ্ছে, তাই না? মনে হচ্ছে যেন ধুলোর ঝড় উঠেছে, ঠিক ওই গরমকালের আগে কালবৈশাখী হলে যেরকম হয় ঠিক অনেকটা ওরকম মনে হচ্ছে। কালবৈশাখীর যখন ঝড় ওঠে তখন গাছ থেকে পাতা পড়া, ধুলোর আস্তরণ, কিরকম মিল পাওয়া যাচ্ছে তাই না সমুদ্রের তলার সাথেও? মনে হচ্ছে যেন কেউ একটা সমুদ্রের তলায় কালবৈশাখী ঝড় তুলেছে।
Okay, this is right now we'll find a source.
এই ধরনের সমুদ্রের তলাতে যখন আমরা প্রাণ দেখতে পাই, যেখানে অক্সিজেনের কনসেন্ট্রেশন লিটারালি প্রায় কিছুই নয়, মিনিমাম অক্সিজেন
You find the
কনসেন্ট্রেশন,
one that you
সেখানে প্রাণের এত ডাইভার্সিটি অবাক করে দেওয়ার মতো। সত্যি আমাদের পৃথিবীতে কত কিছু জিনিস আছে যেটা আমরা কখনো দেখিনি, তাই না? যেটা আমাদের জানারও বাইরে। আমরা হয়তো ভাবি হ্যাঁ আবিষ্কার সমস্ত রকমভাবে শেষ হয়ে গেছে আর নতুন কোনো কিছু আবিষ্কার করার নেই। সেই রকম সময়তে যখন আমরা এরকম কিছু দেখি যেখানে সবটাই নতুন, আমরা জানি না ওখানে কি থাকতে পারে, দ্যাট কাইন্ড অফ স্কেপস আ চিলিং ফিলিং। যখন আমি অনেক ছোট ছিলাম তখন থেকে আমার মনে হতো এমন এমন একটা কিছু করবো যেখানে আমি জীবন পৃথিবীর যে যে জায়গাতে আছে সেখানে কি কি ধরনের জীবন আছে, নতুন কি আছে সেটাকে জানবো। বিজ্ঞান এমন একটা জিনিস যেটা আমাদেরকে কত নতুন জিনিসের টাচে নিয়ে আসে, তাই না? যেখানে আমরা অনেক কিছু নতুন জিনিস দেখতে পাই, আবিষ্কার করতে পারি, প্রত্যেকটা মুহূর্তকে আমরা বাঁচতে পারি আমাদের মতো করে। তাই প্রত্যেকটা মুহূর্ত আমাদের কাছে একটা নতুন চ্যালেঞ্জ নিয়ে আসে, নতুন করে বাঁচার স্বপ্ন দেখায়। যারা আমার মনে হয় যারা বিজ্ঞান নিয়ে গবেষণা করে, যারা বিজ্ঞান নিয়ে কাজ করে, তাদের কাছে তারা প্রত্যেকটা মোমেন্টকে বাঁচে, অদ্ভুতভাবে বাঁচে। এই যে সমুদ্রের তলায় যে পাথর, তার চারপাশের যে অর্গানিক সেডিমেন্ট, আর কত কত মিলিয়ন্স অফ ইয়ার্সের গল্প নিয়ে আছে আমরা জানি না, তাই না? এমন একটা সময়তে এখানে প্রাণ তৈরি হয়েছিল যে এই সময় পৃথিবীর মানে সব থেকে পুরনো প্রাণও হয়তো তৈরি হয়নি। তাই আমরা এমন অনেক কিছু হয়তো দেখতে পাচ্ছি এটা হাজার হাজার বছর ধরে ওখানেই আছে। আদিমতম কিছু প্রাণকে আমরা এখানে দেখতে পাচ্ছি হয়তো, বা আদিমতম কিছু শিলাকে আমরা এখানে দেখতে পাচ্ছি যার হয়তো বয়সের কুলপাথার নেই। So we can see a lot of orbs, we can see a lot of marine snow, we can see a lot of organic clouds precipitating over here. So we are kind of trying to collect some brachiopods over here, and they are like pretty small organisms, so we have to be looking up very minutely to get some brachiopods over here.
Yeah, from what I can tell, it can be
This is why it's taking some kind of time because
We're coming back to the same field because we're seeing that the pink stuff.
Right in the bottom, right?
Let me know if you let me know if you see something from there. Keep going left.
so yes, my you can see some pretty
The site is,
Pink.
the site is behind us because the big orange blob was kind of way ahead of us. So the acorn worm is behind us.
Behind us.
Yes.
so my colleagues want to collect some echine worms which is behind us, so now the ROV is going back.
Of course, it's full of acorn worms from our city.
So it's full of echine worms, as my colleague is saying, and their interest is to collect that. So we are coming a kind of back to collect those samples. আমরা এখান থেকে প্রচুর অ্যাকাইনোডার্মাটাস পেতে পারি। আমরা জানি যে অ্যাকাইনোডার্মাটা এমন একটা ক্লাস অফ অ্যানিমালস যেটা আমরা সমুদ্র থেকে পাই, সমুদ্রের গভীরেতেও অ্যাকাইনোডার্মাটা আছে। এখন আমার কলিগের কিছু অ্যাকাইনোডার্মাটার উপরে ইন্টারেস্ট, এবং আমরা এখন পেছনে এসে কিছু অ্যাকাইনোডার্মাটাকে খুঁজতে চাইছি। সেই কারণে আমরা চেষ্টা করছি যাতে ভিজিবিলিটিটাও বাড়ে এবং
Yeah, so we as soon as we let it settle, we can move up. Yeah.
আমরা অপেক্ষা করছি যাতে এটা সেটেল হয় কিছুক্ষণই অর্গানিক প্রেসিপিটেশনটা।
Yes, yes, please. Okay.
সো আমাদের কিছুক্ষণ অপেক্ষা করতে হবে যাতে এই অর্গানিক প্রেসিপিটেশনটা কম হয়, আর তারপরে আমরা স্যাম্পল কালেক্ট করতে পারবো।
I think it's to my right. It's to the right here.
আমি জানি না কারা কারা এখানে
I think it's
ইন্ডিয়া থেকে দেখছে, বাংলাদেশ থেকে দেখছে,
But we need to start.
কিন্তু
We should start going up.
আমি যদি আমার নিজের এক্সপিরিয়েন্স বলি যে এরকম একটা এক্সপিডিশনেতে প্রিন্সিপাল ইনভেস্টিগেটর হিসেবে ডাইভ স্ট্রিম লিড করা কাইন্ড অফ এটা আমার কাছে স্বপ্নের মতো। আমার মনে হয় আমি চলে যাচ্ছি 13-14 বছর বয়সেতে, যখন আমি স্বপ্ন দেখতাম কিছু করার, কিন্তু আমার চারপাশে কোনো মডেল আমি কাউকে পেতাম না যাকে দেখে আমি এগোতে পারি। এইরকম একটা সময়তে আজকে তার থেকে আমি 20 বছর পরে দাঁড়িয়ে 33-এতে যখন 13 বছরের ওই অপর্ণার দিকে তাকাই, আমার মনে হয় ইউ হ্যাভ মেড ইট। এটা নিজের কাছে নিজের কোথাও জিতে যাওয়া। পৃথিবীতে বিজ্ঞানের থেকে বড় সত্যি আর কিছু হয় না, তাই যেটাই আমরা দেখতে পাচ্ছি
No, that should be fine. Yeah, I have no problem. Yeah. Yes.
Okay, let's assume every mic and hearing's on.
Just assume. Okay. Yeah.
The white one?
আমরা অনেক অনেক কিছু পুরনো শিলা দেখতে পাচ্ছি, এখানে পাথর দেখতে পাচ্ছি। অবশ্যই এগুলো ভলক্যানিক অ্যাক্টিভিটি, সিসমিক অ্যাক্টিভিটির সময়তে যখন নাস্কার সাথে সাউথ আমেরিকান প্লেটের সংঘর্ষের ফলে আতাকামা ট্রেঞ্চ তৈরি হয়েছিল, সেই সময়কার পুরনো অ্যানশিয়েন্ট কিছু শিলা।
We were sitting here for a bit, right? But that's just now, maybe. Yeah. I think it's. Were you on station keeper when you were there?
No, it's not.
I suspect it's probably up to.
Yes, I'm sorry, he was on station.
So,
It's to the right. Us to the left. I can't go that way anymore.
That one, could we inspect this one?
Okay.
maybe with the whole rock.
Yes.
Let's look at the white thing.
So now after the sediments are almost getting precipitated, we can see some brachiopods, and they are kind of ancient forms of life. So our interest is to collect that brachiopods, but for that you have to kind of wait that the organic precipitation gets a little low so that the visibility increase and we can collect them. আমরা ছোটবেলাতে পড়েছি তাই না, পৃথিবীতে 3 ভাগ জল আর 1 ভাগ স্থল। এই 3 ভাগ জলেতে পৃথিবীর সব থেকে বড় অ্যামাউন্টের প্রাণ আছে। তো সেই প্রাণের সন্ধানেতে আমরা এখন কিছুটা ভিজিবিলিটি দেখতে পাচ্ছি। এইবার আমরা ব্রাকিওপডটাকে একদম কাছ থেকে দেখতে পাচ্ছি। এইটাই আমাদের কাইন্ড অফ ইন্টারেস্ট, এটাকে আমরা এখন ফোকাস করছি। নাও উই ক্যান সি দ্যাট pretty tiny brachiopods with a
Sponge.
wow.
No, sponge.
And it's a sponge.
Let's keep exploring other rocks, please.
I don't think it's like, I can turn you back right now. Just hard to see this.
This is about the distance we need to figure out if it looks like a brachiopod from far away, so we can zoom in.
So we were thinking it's a brachiopods, but
That'd be great.
it's basically a sponge.
Okay, moving. Is it gonna be right, Jim? Yeah, of course. I mean, if you want me to do it. Looking forward.
আমরা এখন অন্য জায়গাতে কিছু ব্রাকিওপড খোঁজার চেষ্টা করছি। Can be this one? Guillermo?
No. Yes, look. Hello.
Can we left somewhere there one white points? This? No? Maybe? This one?
Yeah, close up.
We can close it up here.
Yep.
Sí, ahí aquí porque está San Juanito el Villante. Yep, right there.
It's a
No?
No.
No. But
Keep going.
maybe zoom in a little bit on those rocks.
Yes, it's true that we can see some pretty pattern of geological features over here. The rocks and the formations are very, very nice, very beautiful. Wow, that's so nice. It's very beautiful. That's Nick. Look at those.
They are what, crinoids?
Let's come back here, please.
Crinoids.
And
This-
here, just nice. And then
That
we
also.
go there.
That also. Look.
Una line.
Okay.
So first this,
Yeah.
and then this.
Keep your eye on it.
Yes.
Yeah, here we go. Moving forward. Keep your eye on it.
Yes. So we are near to see a brachiopod there.
Okay.
And the kind of group are trying to go near at that to see if it's really a brachiopod or again the sponge makes some tricks with us.
Oh,
This one and this one.
those
So you got some.
those two specimens right on the just off the sand.
Those.
Those two, yeah.
I mean, use your best just to know how to get them.
They seem to be.
Stand by.
Esta roca es blanda. Se puede sacar todo el trozo.
Guillermo said that round rock should be easy to get.
The rock or the animal?
Rock with the animal.
Oh, this one, this whole thing. With the, with
Let's
the
try.
animal.
Let's try to get the animal. If not, you can take the pulsing because he said that it should be very breakable.
Okay. We could probably, if you want, we could probably take the rock. Oh, we'll let it decide.
Let's try the animal first. If it doesn't work, we can take the pulsing. But
Is geology interested for the for those layers of rocks?
Yes, it would be nice if we can retrieve some.
yeah.
Okay, let's try to get the Assyrians.
Okay, so that.
So we are kind of thinking to get the entire piece of
This?
rocks.
The brown
And
is
this.
for a piece.
Okay.
I mean, you get rock with it at some point or the other. That would be fantastic.
You could try and suck it, but they want the rock.
Let's
Yeah, so we'll just use start with animal ground with the suction software, and then we'll
So we
use the
So the geologist on board, they want
Yeah, the
that
way you
piece
did it
of
before
rocks
with
also.
by dislodging it with a tool.
Yeah.
with the lip of the
Exactly.
And,
Yeah, perfect.
I can't tell whether it's the same plate or not, same joint. I could probably see some of it, and I'm trying to index it.
So just what I would do is hold the index, you should be able to see it rotate and then hold it coming back, we know where the center is.
So we are kind of trying to
Okay, I'm gonna
collect
try now, okay?
the brachiopods not only over there but also the
Yeah,
piece
that's
of
separate.
rock
That's
that is
separate.
having some beautiful three-colored
Did you want it on
formation
jar 6 or jar 7?
of
You want to go back to the old jar?
yellow, beautiful
Yes,
maroon and black. So We will be first trying to get those brachiopods. If no, we will be collecting that entire piece of rock. আর পেছনে আমরা একটা বিউটিফুল কাইনোডার্মও দেখতে পাচ্ছি। আমরা এখন চেষ্টা করছি ওটাকে ওখানে ফিক্স করে যদি সাকশন পাম্প দিয়ে ব্রাকিওপডসটাকে আইসোলেট করা যায়।
Antonio, how many cores do you still have?
Just one.
Just one?
Just
Okay,
one.
same thing with me. Okay. I mean,
The
this is
thing
a,
is, I would like to have for geology a full core for sediments at some point, but we
So my geology friend wants a full core of sediment here also,
and many push cores available. So
yeah.
and they want the post-core samples also from here.
Let's see.
So in this huge zoom, we can see like the temperature is 1.82 degrees Celsius, and we are at a depth of 2,946 meter.
Ready?
So with the suction pump, we are going to catch up some stuffs of almost 3 kilometer of depth. It kind of takes time,
Yes.
Yeah.
but we are successful.
I don't know if you stick it in there, I think it's going to probably fall off and go inside.
So our target now is the one that is in the left, that another brachiopod.
Where we went. I don't remember which two we went.
yes, six.
So now we're going to collect this another one. That beautiful stuff over here. So with the suction pump we are going to take it up. আমরা এখানে দেখতে পাচ্ছি খুব সুন্দর একটা তিনটে লেয়ারের ফরমেশন, যেখানে মেরুন, ইয়েলো, ব্ল্যাক, বিভিন্ন
Excellent.
This is suction sample 6, right?
Yes.
The crinoids, yes.
Okay,
Beautiful. So let's
This
go
one?
to the crinoid. Yes.
The next suggestion
That's
is
this
collect one of the two crinoids ahead of us.
Okay.
Let's try that one.
Roger. Okay, I'm moving that way.
So now এখন আমরা যাচ্ছি ওই কাইনোডার্মটাকে কালেক্ট করতে। এখন যে কাইনোডার্মটাকে আমরা কালেক্ট করতে যাচ্ছি, এইটা কাইন্ড অফ আমাদের ইন্টারেস্ট অ্যানিম্যাল টিপের। তারপরে এখন চেষ্টা করছি ওটাকে সাকশন পাম্প দিয়ে ওখান থেকে নিয়ে আসার। It's a beautiful structure that we can see. It can be a delicate structure.
Yes. So we'll jump over to the second.
Guillermo, ambos, ¿eh? Ambos.
So my colleagues are planning to collect both the gynoderms. First we will be trying to collect this. And then probably we will be heading towards the right and collecting the another one.
they have the tendency to autotomize their arms. Let's see. To let their arms go.
And let's make sure this thing person.
I see the person is still swimming there, right?
So we kind of need some more
I'm gonna read next 4, right? Then I'm gonna come back.
to get that stuff up with more
I'm gonna come back.
perfection.
So that's 6. Let's get a little more.
No, I came back.
Okay, so you're happy there?
Yeah, see if it's okay.
Yeah, those pieces are straight and fell out, so. You get it on full?
I am now.
So now we are kind of near to it, and we are yes.
Oh, that's the swimming one.
Oh. Yeah.
Pobrecito.
Yes.
Pobrecito.
The next one.
So they're attached. They're not attached on the surface. So they're hanging on the surface of the rocks with their cirri. That's how we call these appendages.
So, suction sample 6.
Yes, also.
I did not know that they
So
had an
we
escape
are seeing that
response.
there is a hello from Pell School in Newport, and
So
our
expect
Falco
the same
2 will
here.
be talking to them on Monday. And now we are near another echinodon that we are also planning to collect.
Yeah, you see the cirri.
So I like going to get a little more near so that we can collect it up. And look at the precision of this.
Oh, too late,
Wow,
buddy.
wow, 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 wow. So we get that also.
Excellent.
It's still in there. It's still inside.
Yes. So we should keep exploring. Yes, absolutely.
Yes, yes.
But the geology wanted a full sediment core.
Yep. If it's possible, but maybe not now. I mean, at
Not
some
now.
point during the dive, I would like to have, because we just have 2 partial ones, so
Can
let's leave it on.
Can we just show that pretty nice, that animals over there
Of
too?
course.
And then we move to the another
Of
one?
course, yes, yes.
This in the close up, this two, if we can show in the.
Okay. See if I can,
So that's a no, that's a sea urchin.
That's a sea urchin, we can see. It's very, very beautiful. Wow.
Run, run.
You see that?
And look, the poor baby with its little tentacles.
And then let's move to the big block.
And there is a piece of coral also we can see there.
This one, the white one. You can get a little closer if you want. It's up to you.
There's a different kind of coral here also. Something interesting.
Is that interesting?
Yeah, what are those little yellow,
This?
2 yellow blobs
This
on
two.
the left side of the coral?
This too.
These two. These
Yeah,
two.
these 2, yeah.
Maybe you can zoom it up here and.
Okay, I'm gonna move closer. Yeah. Oh, sorry.
Yes, yes. Oh, which one?
Getting smaller and smaller now.
Guillermo, are you interested for the sea urchin?
I like it, but I need to see this.
That
Okay.
another one.
Okay.
This one.
But let's see the coral first.
See that two blobs beside.
And the two yellow blobs, this is what they are.
These two.
See those two? Says sea urchin. Oh, yeah.
These two.
Yeah, I cannot.
You want to try a sample?
Yes, yes.
Is jar 6 okay again, or do you want us index?
The jars were okay.
What?
Those yellow blobs. I don't.
Do you want that, Nick?
Yes.
Okay.
Nick, still 6?
Still
Still the texture sucker 6?
Still 6. Yes.
Okay, want some see?
No, that's fine.
Oh, this is an excellent photo.
How should I describe this?
It's going to be almost impossible.
Yellow ferrets?
Yeah.
Oh.
Let's get another one here.
তো আমরা এখন চেষ্টা করছি আমরা কিছু ইয়েলো দুটো ব্লাফ দেখতে পাচ্ছি ও দুটোকে ওখান থেকে কালেক্ট করার আমাদের আইডিয়া নেই তো ওটা কী হতে পারে সেই কারণে আমরা ওটাকে ওখান থেকে নিয়ে আমার কিছু অ্যানিম্যালের ওপরে কাজ করে যে কলিগরা তারা ওটাকে ওখান থেকে দেখতে চায় যে আমরা নতুন কী প্রাণ ওখান থেকে দেখতে পাবো সো সব pretty small stuff
Yeah, they're going.
but
Yeah, they have to be. Okay.
do you want to go to that one next?
Guillermo wants to explore the
That one.
orange.
Can we go to that orange stuff? Orange?
Yeah, did you want the...
No,
No, we're not gonna collect the sea urchin.
no. The another one Guillermo have interest. This one.
Okay.
And there is shrimp also there.
No, it looks like a sea anemone to me. I don't see anything. No?
This one, do you want to zoom?
Guillermo?
Guillermo, do
See anemone.
I mean, there's. I think so. Huh?
wow.
You know, literally.
Wow.
The sea cucumber.
No sé qué será esto. Bueno, puedes tomar el animalcito que está encima.
Let's try to capture the little white
That one.
animal that's on top of
That.
the big block. Okay, ya. Siguiente, el otro, ¿no?
And that's something here also. I don't know what.
I'm gonna move a little closer.
Alright.
Guillermo, do you have interest on this? That's
Una esponja.
okay.
Spongy.
That animal. So, my colleagues want to collect that little white baby. stuff in there.
Also, on the right side, it looks like we have a brachiopod. But
Give you the pointer.
okay.
So you can help us.
Yes.
What is your name?
Aparna.
Is that better?
Yeah, that's much better.
Better for us, Steve.
So we have a request for the white animal associated with the sea anemone.
Are you okay in jar sets with that?
Yes, that will be fine.
So my colleague have interest on that white animal
Number 7.
in collaboration with that sea animal.
6 if that's okay?
6.
Too many things.
Too many things. Let's try 7 if it is possible.
7 already have some.
I just kind of want to make sure.
We've got 3, 4, and 5.
There we go.
Doesn't make sense.
3, 4, and 5 are empty.
Yeah, we're on 7 now, if that's okay. I think 7 is empty as well.
Okay. 7 for this.
Okay.
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 So that animal that we want to collect, that white animal. Going towards it to collect that little white animal.
Maybe, yeah, if you're off, maybe I'm just trying to like pull him out of there.
এখন আমরা আমাদের ROV SuBastian যেটা ওই হোয়াইট অ্যানিমালটাকে কালেক্ট করতে যাচ্ছি। আমরা এখন একদম ওটার কাছে। তো আমরা দেখা যাক আমরা কালেক্ট করতে পারি কিনা যেটা এখানে আছে। Let's see. Wow.
I don't know. Let's see how it's going to react.
Wow.
Suction it, start slowly.
I see no segmentation. So even the
Oh.
antennas. So the tentacles are sticky. So it's going to be a little more difficult.
So they have some sticky tentacles.
When you get to it, I'll just turn it on.
Okay. Get closer to it.
So we are again trying to get the stuff up, but it looks like it has some difficult tentacles.
So the sea anemone reacts.
Yes, it is not liking that we are going to take her friend up from her. I don't know, but we in science we go for the things that are difficult. So we are, even if it doesn't want.
Trying to protect it. Huh?
Nick, why don't you get the whole thing to our sample?
The whole thing?
Yeah. Put it in a bio box.
Okay.
Yes, I think we are going to collect the whole thing up in the bio box.
Challenge.
You can do that.
And
Because if I pick him up, he's going to stick to me. If he came up from
we
underneath
are
it.
changing the plan, and we are going to trick that entire thing.
The old tentacles will be sticky, but underneath those tentacles there's nothing. There's nothing. There's like a base.
We cannot collect the entire animal in the bio box.
The whole animal.
The whole animal.
No, we're just talking about that the tentacles will tend to react and stick to the grab or to the.
Okay.
What if I just actually let him stick to us? I'll just use the yellow jaw.
So because the tentacle seems to be sticky.
I don't know how well attached it's to the rod. Yes.
But we can give a try to see how it will be difficult or.
You want pedal suction?
Yeah, I think you're right.
It's not.
Yeah, you can't give it.
Wow, wow, wow.
There's the base, there's the base.
Wow.
So he's gonna,
Ooh, 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 ooh,
well, that was that's a nice
I'm gonna porch out.
ooh, 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 ooh.
Maybe he's gonna leave it there and let's try to get the limestone.
Okay, okay, he's on it. He's on it.
It's a polychaete, yeah.
Okay,
Okay.
I'm porching out.
Just a really easy way to let researchers
Bio box 2 or 3. Yeah, preference.
Biobox 2 is fine.
So we're going to put it up in.
So it's a scale worm, it's a polychaete.
So yes, it's a polychaete that we are going to.
No, escaping.
Okay, wrong choice. Wrong choice.
Bio box 3.
The other one, that is.
We are going to
Which
collect
one?
it up
Biobox 3?
in biobox
What
3.
about 1B? 1B or 1D will be okay.
I don't remember anymore what we have in the box.
Can I open this? No.
Yes, nothing is moving there, yes.
Huh?
Yeah, it fell off.
So,
Okay, which one do you want?
so we're going to collect
We
it
can
in
go to
another biobox because otherwise the
BB1B or
little
BB1D. But it fell
animal
off.
was almost
Okay, you want that one?
going out
What do you want
of the hand. So
Yeah, it fell off.
so we're trying to go and collect it again.
I'm surprised that the disc was just loose from the substrate.
He's still down there.
Yep.
Okay, now you porch in a little bit, slow.
So yes, we are trying to collect it again and putting it in the biobox. Scientists, they are also stubborn. Like the things that they cannot collect, they are finding that to collect.
Okay, you want suction? So good. Okay, I'll stop.
Scale worm is back. It's there.
Okay, he's there. Slowly. Then come back in. Okay. Porching out. Right. It's gonna come back. There he is. It's coming back. Oh, it will. Come back.
So I'm going to change and I'm going to pass this to my colleague, Dr. Andrew
want
Palmer,
this?
next to me.
Yeah,
So stay tuned and
now we're
check it up some new, more cool things. See you guys.
There are other associated animals with it, but it's not much more.
Sí, pero estoy esperando para que manejar.
Bio box opening, bio box 1. Okay.
One B.
1B.
Yes.
You're going to have to shake it.
You got something right now?
No.
Yeah, they want me to point to next slide.
Okay, for now, need to point in the facility.
Sit.
You got no soccer, right?
No.
Okay, close a little bit and try to use the edge of the lid. Okay.
Can you open it again?
Yeah.
No. You should should fall over. Looks like he's about to fall over. Okay. He's dropped. Okay.
Okay. Yeah, no. Good great effort, different great effort. Yeah. There was a brachiopod right next to it, and I think it's still right there on the rock. Zoom in between. Yes. The white one is exactly a sponge. I'm talking about the one, just a small one, right in the middle. To the right.
That seems to be a scallop.
What is it?
It seems to be a scallop that is one here.
I cannot see yet.
All right. For everyone just joining us, I am taking over as dive lead. I am Dr. Drew Palmer from the Florida Institute of Technology. Typically, I am an astrobiologist, space biologist, but I have an opportunity here to explore the depths of the Atacama Trench with
zoom
the
in
team.
just to check it out?
Yeah, it seems to be a scallop.
Okay, let's. There are scallop-like brachiopods.
Can take it.
Can take it.
Yes, absolutely. Yes, not the sea urchin. This.
Okay. And 7's okay?
7's okay. Yes. 7.
And we added to the 6 with the reservoir.
Better.
We're not going to confuse them.
Thank you.
6. If it is possible, we can put.
Hello, Lisa. Hey, Harry. Good to see you. If you've been following for the last couple of hours, the marine biology team is going pretty crazy down here. Tons of new things they haven't seen before. Lots of interesting samples we've been collecting. We've actually moved over a very small amount of space so far. But there's just been so much interesting stuff to try and capture and take a look at. Right. And we're going to use the suction sampler.
That's the one?
That's the one.
I thought it was the big one.
Yes, I see it.
More. Got full sound. Is that right?
It could be a blind one, but it's okay.
Hey, Adam, it's good to see you. Thanks to everyone who's watching. It is absolutely amazing to watch the delicate level of work that the operators are capable of doing here. It's really impressive.
Which direction, Armando? Just.
Do you have any geologists here to suggest us anything? No?
Antonio left for. He wanted a full core. That was his requirement.
Full core where?
He didn't tell me exactly because the other cores were partial. So he's got one.
Maybe try right here then?
No,
It's too rocky, I
yeah,
think.
it's too rocky. Yeah,
Okay.
probably maybe
Down below.
down below, or if we're moving our way
So-
down, it's like a little flatter. We'll just maybe like.
Guys, I please remember that
Just,
we're...
following this way to also see the coral there.
Guys, if you can remember that requests for sampling is supposed to come to the dive lead.
Right before we.
So when you guys are requesting samples to be collected or cores to be collected, it's supposed to come through me
Guillermo,
and then whoever's
Look at
diving.
this.
That's Econ work. Oh, that's the Econ work. Yeah. But is that the same?
Was that one?
No, that's not the one. It's a different one. Yeah.
It's not the coral.
It's maybe another food.
He's pointing the same.
Yeah.
The same
I know.
let's see. So let's go towards that way, and then we can also get a core for geology.
Okay.
Yeah.
Hey, Nick, so requests for cores and stuff are supposed to come to the dive.
So what's our mission here?
So, well, I mean, getting a core link to the geologist is absolutely fine.
Okay.
Just, that was what we were told us to practice.
Yeah, yeah. We switched it up a little bit because we had to look back and forth, but yeah.
Gotcha.
All on the same page. Awesome.
Okay, we're gonna head that way.
Yes.
Get your source out there,
As we're slowly moving forward across this portion of the trench, we've really seen some amazing life today.
So you've got 2, 3, 4, 5 open.
And right now we're hoping to find a good geology core to sample for the geology team here. Personally, I'm looking for some cores that we think will have some good microbiology. Our research interests lie in biosignatures, so trying to identify markers for life.
Yeah.
It's
Possible to take it.
a request to collect the crinoid.
Okay. We have a request to collect a crinoid?
Yes.
Cool. That is a very interesting looking creature. I'm not sure what the best way to collect that would be.
I'm just going to pick it up.
Yeah,
Okay.
just grab it and then stick it in a bottle.
It's very reminiscent of like a pinwheel to me.
Oh no, that's all.
Okay.
So, for the marine biologists in the room, is this a common looking organism or is this one particularly unusual looking?
It's the crinoids are common as a benthic organism. So this one, I don't know.
Do you want the whole
species.
thing, or you want
Okay.
just the top, or do you want the...
So,
Probably from the middle of the stalk, if you can, just.
You can grab it from the bottom.
And then where do you want to put it? Bottle box 3?
My box trains an animal that's swimming around.
Maybe back to by box 1 or 2? 2. Right.
Let's grab from the bottom.
So, from the bottom? From the bottom will be fine. You want? Box 1 is good. Let's go where that's the ways. Maybe I won't try to cut it. Maybe I'll just try to pluck it off the roster.
so as a part-time plant biochemist, this looks like trying to pull up from roots, but I think these things don't have developed kind of root structures.
You got a good sit.
So, Nick, do you think there's this kind of a hair-like features coming off of that? Do you think that those are microbial associates or those symbionts, or is that part of the organism?
The majority, I mean, the organism, the most important part of the organism are the starting where the tentacles start.
Okay.
So this is just the attachment to substrates. Now, we know from studies that those animals are absolutely full of microbes. Now, if they're symbionts or not, we don't know, but there's definitely a very rich microbial community.
That'd be very interesting. So some of the wispy features that we're seeing towards the base of the stalk might actually be symbionts that are associated with it. So bacteria, maybe even some cool marine fungi. And after this, we plan on moving on to find a geology core. And I'm still looking for my cool micro core. It's been basically since we hit bottom this morning. It's just been kind of a non-stop treasure trove. So.
Yeah, that would be good. Also, Guillermo mentioned that this is a rock. That's a rock box. Okay, okay. For that.
Not this one.
That would be fine. That would be just fine.
What's in this one? This isn't going to swim away?
Tunicate. No, it's a very soft tunicate. It's not going to move away. Okay. Just we don't want to create any currents because it's going to open super. Yeah, nice and slow. Yeah.
Want to try 3 then?
Yeah.
I think it would be better because it's so long. At least it gets into the ball.
Biobox.
Yeah.
All right. So, Antonio, we're going to be moving it into the bio box. We have a new data logger. Our new sea logger is Miguel.
3A, yes. It has a
Say
very
hello,
soft
Miguel.
edge. It has collapsed anyway. I can see it from here. It has collapsed on the bottom. Yeah, so
Miguel does not want to say hello.
Are you happy to double up in that one?
that will be fine. I mean, we've got one rigid, one very, yeah, they're not going to interfere with both. Yeah, it's all right.
Yeah, maybe 3D then. Maybe.
Okay.
Yeah,
All right.
it's floating now. 3B. I thought it was 60. And it's not moving, it's just floating.
Yeah. Yeah.
All right, Miguel, we're going to be putting this in BioBox 3. I'm not sure that's going to bend.
It's okay to force it down because the most important part of the animal are the tentacles. Right?
So the stalk that supports this animal on the rock is actually not the most critical feature. That's just sort of giving it altitude, so to speak, if that's the appropriate word underwater.
Perfect. Wow. Okay, let's get some core now.
Right. We're moving on now to look for a geology core.
So let's move. The request is to move this direction.
You're going to do a geology core. You should tell me as the dive lead.
I would check it core in front of the storms to avoid maybe any critical ground.
So this spot right there. remounting the suction sampler.
Fecal matter from sea cucumbers. All right. Well, sea cucumbers are very important ecologically, especially the deep sea. They rework the sediment. So you can imagine all the sediment has gone through several times through the guts of sea cucumbers. So very important for the ecology, everything for the oxygenation, reworking of material.
So part of our mission down here, we actually have a mixed team of astrobiologists, marine biologists, geologists, chemists, and even geophysicists, all trying to explore this previously unexplored region of the ocean with Schmidt to try and identify a variety of things. So we're looking for living fossils, but then the astrobiologists like me were trying to learn about unusual life, weird life as we like to call it sometimes, and to see if that could teach us about life on other planets, Mars being one of my favorites. I know there's several of us on this team that are big fans of Mars. And a few days before this, we had the opportunity to go to the Atacama Desert on the land, which is the most Mars-like place on Earth. And now here we are in the trench, 3,000 meters underwater.
If geology gets a full core as Mars will, do my last one too. So 2 cores, one after the other. So we finish.
How's this area?
Looks very good.
Yeah. And we're going to do 2 push cores here, it looks like. Right, we're settling back down now. And our hope is that this area is a little less rocky. Okay.
Which one do you want?
I'm gonna try 5. I'm gonna try it with the starter. I'll just see how much John's gonna be able to switch it over.
5 in the front?
Yeah.
Okay, you want to push off? Or you want to try and grab it from here?
Hello to Heather Palmer, my wife. It's very nice to see you. Brian, it's good to see you too. I would agree Mars needs mollusks. One of the things that's been particularly interesting watching today when I was data logging earlier and just as a spectator in the room has been the amount of sediment that gets kicked up every time we land. Actually, this area is a little bit better than where we first started this morning. We had to wait several minutes for that to settle before we were able to do any collections.
Do you want to can we position it over one of these? Try to get like a 2-for-1.
Yep.
Excuse me.
Better picture of the
Oh, honestly, back to manual. Yeah.
Mm-hmm. And is this
I'm gonna zoom in until we get on top, then I'll zoom out. That's right. Okay, I'm gonna zoom out. No, drought. Yep. I need to go further forward and on.
this looks pretty soft, like soft material. Is this core for Oscar? Is for the geology team or is it for the biology team? Geology. Okay. All right. This is for our data logger. This is push core number 5. Miguel's on the case. Said you're on the case. That is, we have an excellent question in the chat regarding how much distance we've covered today. Actually, I don't know the answer to that.
This is a foraminifera. We have a category more specific than foraminifera. I'll keep the exact name.
Look at that. Very nice. So we managed to preserve the surface structure on that.
It's after this. That's 5. That's what's 5 hours
We can do the next core on same idea.
Yeah.
Yep.
Right.
I think we'll have to bump up a little bit. That's not
okay.
far of a reach, but do you want me to just grab
Right.
it?
We're gonna
Let me point you out a little bit.
move a little bit forward to collect the second core in the same area, but I think we want to displace some of the sediment.
You want them all? Yeah, this is. But then what happens is I realize that I don't want to get stuff out of another work on it because that's what I'm getting there.
And this is going to be push core number 6.
Tip.
And
I'm thinking how many projects I have already covered, so.
just for while we're getting push core number 6 going, I just want to clarify for our data logger, push core for number 5 goes to the geology team, and push core 6 will be going to Nick in the geology or in the biology team.
Because, this one, for example.
Move forward.
Nick,
Yes?
do they want to get the one with the glasses on so we can get it all seen?
With that file line.
Just a little bit.
Just a minute. Okay. We have a good suggestion. If you don't mind,
Yeah.
we can pour on top of
Sponge?
the sponge, yes, so I can also take the sponge. Sure.
We're going to do 2 for the price of 1 here. We're going to get some core and an organism again. So that's 2 in a row.
portion done, all geology portion done.
This is for
So now,
this is for around to see something really amazing. Otherwise, we keep going.
Yeah. So I think after this core, we're going to kind of move forward from this area a little bit where we've spent quite a bit of time. I know the micro team, because I'm one of the members, would very much wants to get a chance to see some other aspects of this part of the trench.
There you go.
But again, we've seen some really amazing things today already, so it wasn't hard for our macro biology folks to fill up on material pretty quickly.
Pushing down. Pushing down.
Looks like there's somebody living inside of there.
There's also the sun.
You can see somebody swimming around.
How, if I go too deep, I may crush that. I go a little deeper. It should punch up in the end. I'll try to leave my thing that much. Okay.
And for this core, we'll only be working on some fresh content. I know it's buried there, but I mean, I see it. That's fine.
So we seem to dig
Yes.
up on another tree as well.
That's right. Yes.
Okay, now let's move forward. We're looking for diversity of every type. Right? Just move and move along. Andrew, so now let's start looking for diversity of every kind. So
Yeah.
we stop only for the amazing.
Yes.
Okay, so just to have an idea.
I am the loose card.
So I actually was wondering if we had an idea of how much, what distance we've actually traveled over the last few hours.
Yeah, Carlos can find that out. Okay, each of this grid is 100 meters. So
Okay.
we started here, approximately 101 meters.
Okay, so we've only traveled about maybe 120, 130 meters, and we've already found a tremendous amount of things on the macro scale, but we're going to start moving forward looking for all types of features, both geological and biological. Very cool.
What's next?
Moving forward along this position.
Okay, I'm on a heading of 70 degrees right now. Keep on straight.
Yes, sounds great.
So I put wavelengths, so you should have them. So yes, the 9 is the next one.
Or we could just follow the line.
Yes. I'm sure.
So just follow the transit line.
Yes, please. Yeah, we're just going to move along the line we've already mapped along. We still have quite a number of cores and water samples to collect. Did anyone collect water samples while I was out? Did we ever collect water samples? We've collected no water samples yet? Okay, cool. So we've been remarking on the amount of snow here off and on during the day. It's been a really nice surface. Continue forward. So one of the more unusual things about this crew is the connections between all of us. I think is a really great opportunity in this expedition. We've learned a lot from each other already about geology and astrobiology and space biology, and now really getting an opportunity to learn a lot more marine biology, which for me is really exciting since I actually do work in a marine biology department, but I'm more of a biochemist and environmental scientist. So over to the right, another one of those pinwheel looking animals again. Really interesting terrain. I believe we're planning to be down in here until 5 or 6, but we still have quite a number of sample opportunities for geology and the microbiology course.
Hey Jason, I'm just gonna slowly book along this waypoint here.
What was it? Fantastic.
Oh, sorry, Ollie. Thank you, Ollie.
No worries. Thank you.
We're just going to move along this area that we've mapped last night a little more. I feel like we're coming up to an interesting area here. Can we take a look at that just real quick? Take a quick look at that guy?
Just to know what it is?
Yeah.
I don't know.
Yeah, that's what I asked.
Thank you.
Yeah, if we could just take a quick
Animal.
look. Yeah. Could you make, Miguel, could you make note of that just as a image? I think we're just looking at diversity here. Can we get a still shot of that? Fantastic. The resolution of the camera is amazing as well. Right. We have another one in the background there, but I think we're good. Wow. Fantastic. Cool. I think we have a nice picture of that. We can probably keep moving forward now. Yeah. We can keep moving forward. Move forward. Yes, please. Sorry.
Is that swimming
So remarkably, again, we've probably only moved about 200 meters total of the entire area that we're looking at today. There's actually quite a lot of life for this area, the bottom of the seafloor. Do geologists have any interest in the structures on the right, the features over there? Are you interested in any of this stuff here? Okay. You want to try and get a core?
Still looking.
Okay. Can we maybe move forward and zoom in a little bit on this feature on the right here, please?
So judged by class, we've been here before. We've been here before.
Oh.
This is our marks.
Oh.
I can see our old snail.
Oh, okay. Well, we were interested in it once before.
Okay. Yeah, that must have been earlier this morning, so I can see.
Okay. All right. Well, we can push forward then.
Okay.
Thank you. In some ways, if it wasn't for the water, it would feel almost like walking, like on like the moon or Mars. It's an amazing surface.
Maybe now if you want to go faster.
We can go a little faster, yeah.
We're just gonna let the ship catch
Ah.
up. Yeah, there's a lot over
I'm gonna give the ship a little bit of time to catch up with us before we move up a little bit faster. Could you possibly zoom in on that, please? Sorry. Oh, beautiful. Cool. Oh, it's a very nice starfish. Excellent. Okay. If whenever you guys are ready to move forward, we're good.
Okay. Moving forward.
You want to take it? Okay. We have a request actually to sample the starfish. Miguel, do we have any bio boxes left?
You could probably put it in 3B.
Okay.
Top of the runway there.
Okay. Let's go ahead and grab that then, please.
What do you need that for? Pick up. You're not suction. Right. Just pick up the suction and drop it. Go down to it. Didn't work. You can try those too.
Looks like we're going to use the suction sampler to grab onto it and then drop it into a bio box. We do plan on making dives for several more days. we go with the suction sampler. We're gonna see if we can grab it and put it into a bio box without any real other manipulation.
Okay, working out.
Nice.
It's working very well, actually. Okay.
Opening my box. I'm gonna take it closer.
All right.
Ready?
And all right. And we have a confirmation for BioBox 3. Yeah. So operators, if you're ready, we're ready to. We're kind of caught up back to our original position, so we're ready to move forward pretty quickly whenever you are. Next, we're going to try and move out of this area a little bit because we've sort of been moving around in a bit of a circle most of the morning looking at stuff. We're going to try and move forward a little bit. Okay. All right. Okay. All right. And now we finished collecting the starfish, and we are ready to go exploring for some interesting geology and microbiology sites.
Okay.
All right. If possible, we're we are ready to move forward. All right, here we have liftoff. All right. Is this a good pace for y'all? Okay.
Moving away from the low point.
Sorry, did you have a question?
We're moving south of the waypoint. Our next waypoint is the
Okay. Where do we need to go? We're moving up that way? In the right direction?
Yeah, yeah.
Okay.
Yeah, we should be going towards the waypoint that we left then.
And we're going to continue now. We've collected quite a lot of macro samples already today, so we're going to be focusing on geological and microbiological features that we think will be of interest. And obviously, if we see some cool stuff swimming around, we're not going to ignore it. But I think the boxes are pretty packed at this point. There's a very cool looking fish there. Hmm. Okay. And anyone on the team, if you see something of interest, of course, please let me know.
Slow down.
So in terms of microbiological samples, some of the things we're looking for on this expedition are methane seeps, which we think we'll find some interesting microbial diversity there. We also did a significant sampling of the water column on the way down this morning, looking at the oxygen minimal zone and more. And actually, several of our colleagues are still in the labs processing water samples from this morning. One of my graduate students who actually came with me, Katie Hubrick, is in there right now. All right, we're moving forward still. Gotcha. Very cool. One of the things that we down here as the scientists don't necessarily think about is that the ROV is actually connected to a ship above, and so we have to coordinate the activities of both. It's really interesting to watch. That can affect the rate at which we're allowed to move along the trench. It's very cool.
Right now, I'm 50 meters away from the ship.
Gotcha. And we see this structure that we picked up earlier today. It looks like a brittle star. It's been very nice. We haven't seen any signs of any methane seeps today. We're also looking for the potential of ice. We haven't seen any of that either in this dive so far. However, this is just actually the first of the sites that we're exploring on this expedition. And we move forward along the trench and continue to look at some of the neat features here. So while we're continuing forward, I was hoping one of my colleagues, Lauren, would you like to tell us something about the snow that we're seeing in the water column here?
Yeah, happy to. So we're in a really productive part of the ocean right now because there's a huge upwelling zone here, which means that very rich, nutrient-rich bottom water is being brought up to the surface, and that creates a whole lot of productivity in the surface ocean, and then all of that material that is made eventually sinks down to the bottom as marine snow. So it's actually like little tiny microenvironments, because each of those particles is colonized by bacteria that eat at the snow, and it's like a buffet where each type of bacteria gets a little bit of this, a little bit of that. Some break down certain parts of it, others break down others, and then all that marine snow sinks down to the bottom and creates this really like fluffy texture that we're seeing on the bottom.
So, and this now I'm asking the question because I don't know the answer. I really wasn't very aware of the oxygen minimal zone that's in this area. How does that affect the presence or absence of the marine snow?
Oh, so we see this huge oxygen minimum zone because there's so much productivity in the surface that it's like a smorgasbord for biological activities. So everything that eats all of that produced organic carbon sucks all of the oxygen out of the water as it's breathing, and that's what creates this huge oxygen minimum.
That's interesting. So it's very, in an extreme case of that, I think about like harmful algal blooms,
Exactly.
which where we are in Florida, we get tons of.
Yes.
Well, not tons of, but a lot more than we should.
Harmful algal blooms, fish kills, these kinds of things can happen when you have a ton of productivity in one place.
That's really interesting. Cool. Thank you very much.
Oh, my pleasure. My pleasure.
Cool. So the ship's caught up to us. So we're moving this way. The ship's moving. So we just had a really good question from one of our group about how far away we can move from the ship, and the ship actually moves with us to keep pace with us as we're going through this area, which is really fascinating to watch. So for this area, this is a question for the science crew. For this area, if we don't actually see any methane seeps, do we still want to collect water? And would just collecting water, including the snow, be worthwhile for your work? Could you benefit from just the water with the snow? Would you benefit from just water with snow?
I've taken a lot of water samples
Okay.
and one from the office as well.
And just continuing to move forward. This area seems particularly more rocky.
So we're hoping that our next waypoint on the map will bring us to like a kind of a wall of rock, a very steep drop. And then we're going to turn to the left and kind of follow along that wall. So right now we're at kind of like a flat place where a lot of sediment and marine snow is able to build up. Maybe once we hit that wall, things will
Get some
change.
differences.
Yeah, we'll see if, and we're just going to start like heading uphill on a less steep portion of the ridge. And we'll see if we see
Get some
changes.
different structural features,
Yeah.
biological features also.
Maybe the geologists will see something cool.
It's kind of short dip. You can see on sonar, and then it comes back.
So we've got a short dip coming up.
Should catch up with actual speed.
Are we going to go over the dip or do we go with the dip?
We're gonna. It's pretty short. We'll follow this thing and go. Yeah, that might be it already.
Oh.
Very small. It's just very pronounced on sonar.
Okay.
Flat spot.
Yeah.
You wave that face and then you're gonna say
Yeah. You didn't say
And again, we are moving towards a site where we expect to see kind of a wall, and then we're going to be going uphill a little bit, and we're hoping to find some different geological and microbiological features. For those of you who are just joining us, this is the first dive of the expedition, so we're looking at the first of several sites that we are exploring here in the Atacama Trench, and we are trying to get information on, we're trying to discover living fossils, so things that have been preserved here in the trench, which has been sort of geologically inactive for a very long period of time, basically mirroring the Atacama Desert, which is in Chile. And so we think this area might have a lot of interesting microbes and macrobiology that has basically been preserved here. We already this morning were collecting a number of samples of organisms that we haven't necessarily identified for sure in the past. Now we're trying to look and see if we can identify some interesting geological features and microbiological features that we want to take samples of. And again, this has been a really interesting opportunity to bring together researchers that might not always coordinate with each other. So we have astrobiologists and geophysicists and chemists and microbiologists all working together to explore this trench, which we think can tell us something about the ocean and about life on Earth, but also potentially the life on other planets, be those water planets or even planets that once had a lot of water, like Mars, which if you notice I talk about Mars quite a bit. That's my favorite. That's my planet of choice after Earth. Big fan of Earth. Mars is pretty cool. I can't tell if Miguel is thumbs downing Mars or Earth. Yeah, I've known him for just a couple of weeks now, and I'd say he's probably thumbs downing Earth.
Like on a leash.
Yeah. Unfortunately, we can only move so fast down here, so I say down here as if I was the one that's actually 3,000 meters down, but I swear the technology in this room is so good, it kind of feels like it. Like I'm fully able to just kind of immerse myself in this. I would need a pair of VR goggles and that would be done. So.
We have all the cameras up too. Because we're constantly going back and forth.
Yeah.
So it feels like we're down there.
The Schmidt virtual reality experience. One interesting thing to note is that a lot of the organisms we collected this morning, I'm actually seeing repeated here on the benthos. So I have a good feeling. So I have a great question. How fast is the ROV moving at the moment?
0.2 meters per second.
0.2 meters per second, Adam. Adam works with me at Florida Tech. Oh, nice. Can we get a picture of that, please?
We're going to set it. Yeah, I'm just going to find it.
So if you're watching the live feed, some of these organisms that you're seeing down at the bottom right, we collected a sample of that earlier today. Tried to escape one of the bio boxes, actually. I believe we have a sample of this guy in front of us as well. Beautiful. Oh yeah. We absolutely need to get a picture of that. Yeah. If you didn't hear that, somebody said it looks like a flower with wings. Yeah. There, and in the background, some of the members of our science crew are trying to coax the model to turn so that we can get a picture of it from a different side. Absolutely beautiful. Fantastic. All right. We're ready to move forward when you guys are.
Just tell me what camera.
Ready to move forward.
So for the uninitiated, these are commonly referred to as headless chicken monsters. Okay.
You want me to say that?
Chicken monster. You can. Yeah, but you just learned that.
I have just been informed that these can sometimes referred to as headless chicken monsters, and now that I've heard that, I can't unsee that or unhear that name. So that is going to be with me forever.
Wow.
My wife agrees that that's a good term. So. The top-down video, the top-down image is really nice too.
Someone once told me when a sea cucumber feeds, it's like sticking your arm out, grabbing food, and taking your arm and putting it all the way back to your elbow and your esophagus. Wow, it's like very different.
I think we're ready to move on. We have spent enough time with the headless chicken monster with wings.
Hi everyone. My name is Specialist at the Schmidt Ocean Institute, and I'm really honored to be joining this expedition. I'm usually on shore, but I'm out at sea this time, hanging out with our wonderful crew on Falcor 2 and the scientists. Soph has asked me to pop in and share my little fun fact about sea cucumbers, which is those feeding appendages that you see kind of around the mouth area. I was once told, sort of early on in my science communication days at the Seymour Marine Discovery Center at UC Santa Cruz, shout out to the banana slugs, that when a sea cucumber feeds, it's like reaching your arm out, grabbing some food, taking that food, putting it in your mouth, but then shoving your arm all the way down to about the elbow and then depositing the food into your stomach. So if that's an image you wanted to be left with for the rest of the day, awesome. I'm sorry if I've grossed you out. But yeah, that's my fun sea cucumber fact.
Thanks for your fun sea cucumber fact.
And so we are still moving at about 0.2 meters per second, headed inline towards this rock wall that we want to examine for some geological features.
First time.
Oh my god.
Feeling it.
What was the
Right.
first time for you?
Disco ball.
Disco mode. What's disco? You hit disco mode, on the street.
Yeah.
Oh yeah.
So out of curiosity, what's the fastest the ROV can go
I mean, we could probably,
that you can share?
realistically, depends on a couple of things, how deep we are, pulling in umbilicals.
0.5, 0.6, 0.7. Okay.
It's all about the leash holder.
Yeah.
See if all you can do a little bit more of it.
And the ship is.
Yeah, like, just wondering, are you able to bump it up 4.5? Thank you.
So we moved into a patch right now that's particularly barren compared to where we were this morning. I think we were pretty fortuitous. We touched down this morning and found a whole bunch of structures and features. By features, I mean biology, for us to take a look at and do some quick captures on. And then the quick captures turned into some longer capturing. So, but I think now we're going to kind of push forward. I'm getting really excited. I don't think I've ever been so excited to see a rock wall before.
Yeah, rock wall. Yeah, like just a little. Yeah, but yeah, maybe you're right. Just keep it stretched out, and then as it come up, then you guys can see it. Just like that.
So this expedition, I'll say, is of particular interest to me. As I mentioned earlier, I'm not typically a marine biologist, though I'm in the Department of Ocean Engineering and Marine Science, but I'm actually a biochemist turned environmental scientist and space biologist. But this opportunity was just to work with Schmidt and with all these fantastic researchers been just really incredible. So this opportunity is just too much to pass up on. So it's been really great. I've gotten a great crash course in oceanography and marine biology. So I have to give a big shout out to all of my fellow scientists on the team who taught me a bunch of stuff that I did not know. Turns out that sampling in the ocean and sampling in the land are different. It's almost like they're very different environments. But thinking about the tether between the ROV and the ship, and someone who thinks about experiments that we do in space, this aligns very well because we're very oftentimes limited by what's feasible in terms of mission control and capabilities. What can you do on the space station? What can't you do on a space station? And this feels like home to me. This part. And if you have not seen a YouTube video of the Falcor 2, I strongly recommend you take a look at it. So, as I was informed by several of my friends who are marine biologists, this vessel is really exceptional.
Hey Drew, can I ask you a question?
You can.
For those of us, everyone joining us from shore, can you maybe explain what astrobiology is?
Sure. So it's kind of a loaded question. So it depends on who you ask. So it turns out that astrobiology is actually astrobiology and space biology. And to keep it pretty simple, astrobiology is about what life might look like out there. And space biology is about what life like this, like ours, will do when it gets out there. So, like I, as a space biologist, really, when I have that hat on, I spend my time trying to figure out how we're going to grow food on the space station or on a Mars settlement or on the moon. when I put on my astrobiology hat, I'm thinking about things like origins of life, how microbes still be living on Mars, which I totally have my fingers crossed on. So, so there's kind of two different fields of study in there, but they overlap so much, because when we try to understand how life here might adapt to life out there, we think about things like extremophiles and some of the really weird life, quote unquote, that exists out there. That's some of the stuff that Lauren works on, and Armando works on. A bunch of us actually on the team work on. So it's a really interesting area, but yeah.
Thank you. That was really helpful. Follow-up question. Why should we study the deep sea to learn more about how life might look elsewhere in our universe?
Oh yeah, so that's for one, we actually are beginning to learn that there's a lot of ocean planets out there, and there's a high probability that there might be microorganisms. They may not look exactly the way we think of them here, that could be surviving there, and also, when we think about life in difficult environments, right? This is a very challenging place to live here at the bottom of the ocean. So much of the life that we understand on Earth depends on the sun and photosynthesis and all these other chemical processes. And so if life can survive pretty well down here, pretty plentifully, then maybe it can give us clues about where life could be hiding on other planets, right? So we, as we start to think about, we don't see living bacteria on the surface of Mars. That doesn't mean they're not there. Maybe that's not where they are. So maybe they're hiding out somewhere, right? So if we can figure where life is hiding on this planet, we can figure out where life is hiding on other planets.
Nice. That's awesome. Okay, I have another question. Sorry. You just are so great at answering my questions that I come up with more questions.
Questions are good.
Yeah. So why come here? Why come to the off the Atacama Desert to this particular spot?
So this spot's really interesting. So the Atacama Desert is the highest and driest desert on our planet. And so it actually, in part, has been that way because it's been untouched geologically for a very long period of time. And so that makes it a great, what we call analog site, essentially. It looks like what we think Mars looks like in many ways. And so, but it turns out that the Atacama Trench, where we are now, is basically kind of in the same plate. So it's also been generally, we think it's also been kind of undisturbed. So this is why we think we could find some living fossils here. So some things that, are probably extinct in other places, but because this area has been fairly undisturbed, we think some of these organisms could still be here. And if those if these larger, macroorganisms might be alive in this space, then there could be some really interesting microbes that we haven't seen before. I'm particularly interested in metabolisms or chemical processes that microbes do that maybe we don't see in other places.
Thank you.
Did that answer your question?
That answered my question.
Awesome. All right. So it looks like we're touching down when I found a site of interest for one of our geologists. Oscar is staring intently at the screen.
Yeah.
His brow is knit together.
It's something different for this entire sediment that we are looking for.
Mm-hmm.
Maybe the last 50 minutes.
Mm-hmm.
So it's interesting to go there.
All right. Do you want to try and take a core of this? Do you think, or is that solid? That looks like it might be solid to me. You think it is?
We can take that sample.
You-
It's possible.
No? Do you think it's possible for us to take a push core of this, or do you think it's too rocky?
Just depends.
Yeah.
We can test it. We can test it.
Maybe we can rust the surface, stretch the surface to know if the black is only an alteration about the rock.
Mm.
Yeah, we can test it. Are you gonna want in the rock itself?
Yeah,
Or
yeah.
just- You want the rock
Yeah,
itself as well?
yeah.
Do you want the...
We've received a request to zoom in on the fish more.
Okay. We will zoom in on the fish. I think once we get through some of this geology.
Just as we pass them.
Okay. We did stop by the headless chicken. So you want to try and get
No, the rocks. If we
this part?
Yeah.
Okay. You want to go for the rock itself. I'm not sure that will fit in a push core. You think?
No, at this core, no. Maybe to rush the superficie, to know what is the black color is alteration above the rocks.
Oh.
I need to know if the rocks have a different color to make a description of that, to know
Okay.
is something else. Maybe.
Is there a way for us
Would
to
be a
scratch the rock?
Yeah.
Okay.
Yeah, we can try to get a piece of the rock. We can also see if there's enough depth
Yeah,
to get
Yeah.
if you can pick out a piece of rock, it's great for me. Sure.
I think it would depend on how brittle it is. I think is that a brittle star there? And there's some cool biology to look at too. All right, we're trying to see if there's a way to.
Yeah, just underneath it. Yeah.
Is this a collection of are those brittle stars kind of all along the surface of this rock?
Yeah, it looks like it.
Yeah.
It's like another xenobiologist.
Maybe we can take a sample and go to one of the rock
Yeah. Do you want to try and take a sample and put it in a rock box?
boxes. Yeah, maybe the.
We have a rock box, right?
We have two empty for now.
Okay.
So we have 2 empty rock boxes. So I think the request has been made to try and get a rock sample. Is it? So while they're working on that, is it significant that there's more brittle stars on the surface of this rock? Does it tell us anything about the mineralogy? Or?
No, I don't think so. I think that it's a hard place to stay.
Okay.
So maybe it's easier for these organisms to live there.
So it's more that it's a stable substrate
Yeah,
in
yeah.
a very
the
fluffy
sediment.
area.
Yeah,
Okay.
Right.
Oh, cool. There you go, guys. We get to zoom in on some cool biology and try and get a rock sample. Oh,
Nice one.
all right.
Oh yeah.
The white is
Alright.
chiton.
Yeah. There's a chitin on the rock there.
Chitone, mollusca. Or white.
And,
Yeah,
okay, so it's
Suggest throwing it in a biobox. It's easy to distinguish just because it's through the interface, but we can just
Okay.
tiny little sediment.
yeah. All right, we have a
Get big
recommendation to move into a bio box instead. What do we have free for bio box?
Or something we could double it up with that obviously distinguish it from whatever else is in there.
Okay.
according to this, biobox 2B is empty, so
Okay. Let's move into bio box 2B, please.
for example,
Would it be possible to get that chitin on the rock there, white spot?
Which white spot? I'm gonna hand Lauren the pointer here.
Yeah, this little white spot here.
Okay. That'd probably be good for the sections.
it was a very recommendation.
Sorry, which one?
Which one for the bio box?
2B.
2B.
So that has all the.
I think there was something swimming in there that was.
According to this, it's empty. I don't know if the previous person did not record it.
Did you check the other sheet?
So it might be empty, but 2A has those 3 cucumbers. So
Oh,
there's a
okay.
chance if we open
And
it.
it's the same lid, right?
Okay.
Yeah.
Yeah.
Can we put it
That's
in a sort of thing or something similar? Or will it
Like I could.
is it going to wash out in the coral quiver? Because I mean, it looks like it's
It's pretty sedimenty, like it's
Yeah.
it was very soft. It
yeah.
didn't feel
I mean,
very hard.
I can see it kind of washing out as we hold this.
My suggestion would be like 1 egg, because it looks like there's something biological.
Hey, it's Soph. Just wanted to check back in while the pilots are sampling this foraminiferan. This is called a xenophyophore, which is a very fancy word for this type of protist. When I used to study microbes, forams were some of my favorite organisms, so I have a couple fun facts about them. Feel free to pitch in if you have any more. But this is actually all one single-celled organism that makes this skeleton, which we call a test. And so this test is now what we see. But foraminiferans are part of a group that we call SAR, S-A-R, which is all capital letters. And it's not an animal, but it belongs to this bigger group that includes different kinds of phytoplankton that you may have heard of, such as diatoms and dinoflagellates and other alveolates that the scientists on the previous cruise have studied, for example. So it's a very diverse group, SAR. And specifically about forams is they are the biggest makeup. So when they die and their skeletons or tests fall to the ocean floor, that's what contributes to the majority of the silica ooze that we find on the ocean floor. And that provides a base for the ecosystem that is here. So I just wanted to hop in and say that because I love these little guys. And I hope you do too.
They definitely, yeah, we have a strong request for.
Yeah. It's one of the living fossil species.
Yeah.
Oh, hey, it is a
These attached are pretty good. Do you think
Is
the
it
suction
chitin?
sample will get them off, or do we have to?
Oh, I'm not sure.
Is it
We'd be worried about damaging it if we try to suck it off.
bound?
Is that rock?
But yeah, it underneath is soft.
Maybe we can try to break the rock around.
Yeah, maybe.
With the,
They're pretty. They're
the same way as we took the rock from up and then down.
We're discussing the best way to potentially collect this sample. So this is one of the sort of living fossils that we're interested in collecting here.
I'm gonna get behind it like
Okay.
a
And so this sample will be doing double duty because we're going to get that chitin, and we'll also get a little bit more of this rock that our geology team wants.
Yeah, it's like break around a movable Tyler bike. A movable Tyler. I'm sure they love that. Ooh, I think that brittle star. Yeah, I don't want to crush it.
I was really surprised at the number of brittle stars I saw on this rock. But I think it's because it's a fairly large outcropping compared to what we've seen. Oh, that one broke off really beautifully.
Nice job.
Nice.
High stakes for you. Do you hear this? Oh, I hear it.
Maybe we can turn the 90 degrees.
Yeah.
Why do you want to turn the sample 90 degrees? For purposes of picture?
No, just so that we're
Just
not
so that
grabbing
we're not going
on.
on the
Oh.
Leptochiton
Is that
side.
another one on the?
I think
Okay, good. We'll grab one with the other one there.
Yeah.
More rocks outside.
I'm wondering if that isn't another one on the rocks directly in front.
And also the color of the sediments are very different to the other sides. So maybe any kind of bio is there, but
Oh.
microbiology.
Can we take a core?
I would like to.
No,
I'd
no.
like to take a micro core.
Okay, so
Real quick, you want this rock sample too before we grab the other piece?
Yes, please.
Yes. Yeah. Just take it.
okay. Okay, look at the rock.
Yeah, look at the
This rock.
Yes.
Okay.
yeah.
You have the black outside, a little pale brown, and the green. And maybe the green, I don't know, maybe it's some of the other sediments, but something maybe else.
Yeah.
Something else.
So there could be something bio here of interest.
It's very, very strange.
Did you say nougat or nugget?
Nougat?
Is that's like a combination? Alright. And that's going in bio box A 1A again? Okay. And so that should have the chitin and the rock, right? Or did we lose? We lost the chitin?
That's just
Okay.
the rock. I think there might be another chitin here. If we want to try to do
Yeah.
that as well after we've retrieved the first one. And I think some of the microbiologists would like to take a core here.
Is it possible to use a soil or something like that?
Like the scoop?
Yeah.
I don't know. I think they're going to try to use the other arm.
Because
Okay.
the rock or whatever it is not too hard to
They said
drop it.
a lot of stuff can come out of the scoop though, so I think
Okay.
with how this rock is, I think it's too loose.
I think it's there.
That's really cool.
That's very cool. So, are you saying you think that's the
That
chitin?
one, yeah. I think
That
there's
could
another
be.
one there. I think we should take it.
That could be a chitin, yeah. Let's see if we can get this one
I
though
think there's a
first.
couple on here. I think there's another one over there, maybe.
Oh, it could be, yeah.
It's hard to tell from this angle.
Yeah. Oh. Gosh. Every time I watch you guys work, it just reminds me of how many times I've failed at that crane game. Yay! So, for those of you just joining us, we've kind of landed on a really interesting spot here. There's some really interesting rock here, and we're seeing some processes in the rock that makes us think there could be some interesting microbiology as well. So, I think we were headed for a rock wall. I think we're gonna pause here for a little bit and collect some samples. I think. Are kind of interesting to us, so we may have a living fossil on our hands, which would be kind of cool. And definitely some interesting rock formations, and these colors are looking good from a biological perspective. All right. Can we confirm that the chitin is on that rock? Yeah, I see him.
Can we maybe zoom?
Yep.
To check
Yeah.
if it's not being
In good shape, yeah. It looks like the rock broke just right in between. So there we go. Beautiful. Looks like a...
Leptochiton, mostly okay.
Yeah. Guillermo's like beaming from ear to ear over there. Look at his face. He's like, you're super excited.
It's supposed to be the 1A, right?
Yes.
Wow, okay.
We should zoom in and see if we can see any more of those on the rocks
Wow.
there. I see a couple other white
Please.
spots, but I'm not sure.
And I think I know you want to take a core. I do too.
Yeah.
So
Yeah, I probably can take a biopsy.
Yeah.
it's core time. That'd be great. So I think we were thinking this guy right here on the left might be one right there. Oh. Oh, you mean this?
No, that's a sponge.
Yeah. How dare that sponge be there? Yeah, I think it's more of a yellow color, so I don't know that there are any more on this one. Was it
On the top down view, so you can
that? Maybe that one? Yeah. 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 Okay. I think.
Maybe, but if we can grab another piece of, if there's time to grab another piece of rock, it seems to be plenty.
Okay.
And I would also appreciate it having it.
Okay.
Another piece of rock and 2 quarters.
Okay. One more piece of rock, please, and oh, that is pretty.
So cute.
Are you happy with the first rock we took? We broke off some pebbles around it. You want to take a chunk of that?
Yeah, that'd be fine. Yeah.
It's just the logo.
Or you grab that.
What happened?
He's hearing music.
I'm not. I'm all absorbed in rocks at the moment. So hyper focused nerd at work in here. So. So for those of you just joining us, we have paused moving through the trench on our course for today to this really rock formation that had some really cool biology, perhaps a living fossil. But then there also looks like there's some interesting geology and microbiology here that has made everyone in the room here get excited. Yeah.
It does. It's got like layers.
We can maybe take the big piece of that.
Switch to the
Yeah.
It's got the jaws on it.
That should be a lot of it. So.
That looks great. Oh. Boo. Yeah.
Punk rock.
Yeah. All right. This is the challenging part. The rock is very soft. Yeah, this material is very soft, so it's been pretty hard to collect. Yeah. So Lauren in the group just made a really interesting remark that when we started off our expedition here by being in the desert, so trying to see about the terrain on the land and what that could teach us about what we were going to see in the water. And there too, we had a lot of the rock material there was very, very brittle. And so it's kind of interesting to see this kind of continuing. Right. Hopefully that will stay together enough, whereas we think in the bio box it should be stable enough that it won't break up too much. Right. I think.
You want that last little piece?
Do we want the last little piece, or do we think we're good?
I think we're good. That bit was pretty big. So.
Yeah, I think we have enough pieces at this point. So.
Can we take 2 cores here?
Yeah.
One for microbiology, one for Nick? Or
Yeah.
You want some? Okay.
Yeah.
Do you want
So.
to like bump up into the flats there where there's a little bit deeper sediment, or you want to?
I don't. Can we move a little bit forward? I don't want to move too far because it's the geology is the interesting part.
Right, okay.
yeah, I'd like to get close to this one.
I'd like to stay bio adjacent.
I could, like, if you look at our sit camera here to the right of the arm.
Like over there.
Yeah, we could try that.
Okay,
instead.
that'd be awesome. I have a weird pointer thing that I'm using right now, and it makes me feel very school teacherish.
I love that thing. I was really having fun with it.
So, this is probably what I should have when I teach in some of the lecture halls. I end up like jumping up to try and reach things. And I'm kind of a chubby guy, so it's kind of funny to watch.
At least you're tall.
So that is true. I have that going for me.
Let's try it and see. Yeah, maybe a little bit. Yeah.
So
Probably 7.
which cores do we have available? The 7, the next one.
8, 9, and 9 to 17.
Okay. So a lot. All right.
She told me to take them away from her, so they're yours. They're yours. They're yours. They're all yours. Oh, I don't know.
Lauren, I accept you.
If we're gonna grab one on like the right side, do you want to try and do one on the left side, or do we want to move forward a little bit and try not to disrupt the sediment?
I mean, I'd like to try to get as close to that one as I can.
Yeah. I mean, I think we're doing that.
I don't think it really matters which side.
Okay. I was wondering more about like if one did the right and one did the left, like on the other side. So.
Or if you want, well, I don't know if you want to try to get them more close together so they're more
Homogeneous. Yeah, that sounds good. All right.
Do you want me to lock this one as yours or yours?
Happy here?
Just
Yep.
Do you want to take this one, Drew, and then
Sure.
we'll pick it? Yeah. All right. This one will be
Okay.
7 is clear. Good.
My first core.
Want to do a 2 for 1 here?
Oh no, I'm not interested in that. Sorry. That's a big form of life. I don't like those.
Okay, come down.
If I can see it, I'm not interested.
Yeah. If I can see it, but it's not a plant, I'm not interested. Most of the work in my lab is involves plant microbial or plant bacteria associations. So, oh, that looks nice. It's going down.
Well, we have.
Ooh, 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 ooh. Ew. So this is the 7th core we've done, and there's a moment every time the core gets pulled up where I just kind of like hold my breath and go, please don't fall out. So
And you don't have to take another one afterwards, so 3 here in total.
here we do 3.
Another
Okay.
one, see one,
Okay.
one here as well.
Oh yeah, that's really sticky. That's interesting. What does that? Do we know? So what does anyone in the control room know what kind of properties, what reason do we have that would make the sediment sticky like this?
We can't hear you.
Oh, I was asking if you had any thoughts on what would make the sediment particularly sticky.
I mean, I could think of biological reasons.
Yeah, I can think of biological reasons too. I'm
Like-
wondering if there's a geological.
Yeah, if there are microbes that are producing a lot of exopolymeric.
Yeah, biofilm type
biofilm
material.
type stuff. But as far as the geology, like if there's a lot of clay, that would probably
Yeah.
stick together more. But I don't think.
So I'm gonna go with hoping there's a biological explanation.
Yeah. Of course, I'm going to blame bacteria. But.
Oh.
Just like
Yeah.
I was thinking maybe like the chemical structure.
Yeah.
Those things could kind of help
Mm.
those stick together. if that
That's
makes
true.
sense.
Yeah.
It's such a productive area that's.
Good.
Yeah. No, that makes sense.
Okay.
Probably.
Yep, I'm gonna use it.
Kind of gluing things together.
Okay. So in addition to core 8, they'd like to take a core 9. They'd like to take another one.
Another core.
And they're kind of hoping that 8 and 9 could be close together.
Okay.
Yeah.
Cool.
They want to be core buddies.
2 more for microbiology.
8 and 9. 8 is mine?
8 can be yours.
It can be yours.
Are we going for 10? Do I hear 10? 10 cores.
Andrew, you know that it was for chitons and brachiopods that we came from. We have a boss.
Yeah.
We can celebrate that.
Okay, we can all go home.
We found the living fossils we were looking for. Expedition over. Biobox 2A is containing, I believe, what is Biobox 2A, the sea cucumbers? Yeah, they've been trying to escape.
Okay, the 8 is Armando's.
8 is Armando's, and 9 will be mine and Nikolas.
And get that.
Where do you want this one?
Andrew?
Andrew.
Where do you want it?
I for mine, yeah, with foraminifera on the top as well, like a kick.
Okay. So we do want the twofer in this one. So I guess probably here if it's possible, because we still want to stay next to that formation. And then that would put the second one pretty close by too. Then I don't know if that's too close to the rock, although that rock breaks apart pretty easily.
Yeah, you get a good chance you can just get the rock.
Okay. We have some happy dances going on in the background here. So.
Apparently we want her push from here as well. So
You do? Okay.
another one.
All right. And we'll be taking another a fourth core here. So I was right. It is number 10. So for the geology, microbiology, and biosignature group, this has been pretty much the sort of the mother lode in terms of samples. Ooh, all the way down. I think there's actually 3 sea cucumbers in BB2A. Oh. Oh, that course, really good job.
Nice core.
I wonder what it's going to smell like.
That's for Armando.
Yeah.
What?
Turn your mic back on, Hannah.
Oh.
Say that again.
I wonder what all this sediment is going to smell like when it comes back.
Roses.
I'm very curious.
It's it's going to smell like discovery.
It smells like a science paper. Science smell.
Right.
I love the smell of discovery.
Yes.
I love reading description. I love reading the sediment next to rock macro with Leptochiton with foraminifera.
Yeah.
Delicious.
Yeah. Chef's kiss.
This was the 8th.
That was 8.
Yeah.
So we'll take
And-
9 and 10 and then
Yes.
we'll continue towards the waypoint.
Yeah, 9 will be Lawrence and Nice.
Okay.
How much did they mark yesterday? This area will not go to the rest of the
Andrew, could we get a big piece of rock? But the guys are looking for little animals inside.
We already grabbed a couple pieces. Big. We can ask. I'm not. The rock that we're looking at right now is very crumbly. Yeah. So I think.
I've been dying to make a stream dive finally because I can do beauty cream.
But.
rock samples from the rock because it's really sandy. So there
Can we
was
sit down and make this?
Yes. That's so one of the reasons why we stopped at the spot is because Oscar noticed the rocks.
And then another rock for the microbiologist.
Yeah.
no.
Yeah,
Then
that's
we'll have
that's
we'll have
yeah,
next.
we got
Yeah.
it. 4-9, that's mine.
Don't touch mine.
And
This
yes,
is higher Divestream.
mine it is.
Can I hear me?
I just I just like swimming by. Hey guys, what's up? Oh, it's the headless chicken.
Headless chicken monster.
This is where
Where would you like it?
As close to the rock as you can get.
Yeah.
You want this one right next to the other. Even maybe with a little core.
That yeah. This one could be next to the other core. That was where we wanted
We
it.
don't want foraminifera in this one.
Yeah,
Yeah,
That's good.
that could work.
No foraminifera.
I'm not sure.
But,
Let's see if it goes down. It might not
Yeah.
go down.
Yeah. Well, I think you're. I, do you think you might get some of the rock in there? I mean, it seems like it's crumbly enough that it'll.
Yeah, let's see.
It might break away.
Yeah.
try it. If it doesn't work, we'll try it.
Ooh,
Yeah.
I saw that.
So did we hit a hard spot? Okay.
Okay.
Move a little away.
We can do it.
Shake.
Give it, clean it off. Alright.
Maybe a bit
Yeah, maybe on
more
the other side.
outside around.
Yeah.
like 20-30 centimeters away from the other one. Might work.
Maybe,
Do we go in?
are we
Okay.
okay if we bump up a little bit?
Yeah, I think so.
Reach the-
Yeah.
Yeah. All right, we're gonna have to move forward a little bit.
Pivot to the left, and we'll come right over the rock and try to do like over by your left rail somewhere there. That way, yeah. Is that what you know?
You just thinking like
That works good. I was thinking we just move it this.
like so sampling over in this side. Okay, we're currently discussing how to try and get some of this material. I think we need to. All right, we're going to briefly touch off and then touch back down and just rotate the RV just a little bit. This should give us access to that same sediment.
How do you like the bottom? Makes it real.
Yeah, it's alright. Gives you the
So this is sort of an example of what makes this expedition very interesting, is that this is a site that geologists and microbiologists get excited about, and then astrobiologists get excited about it too. So it's a diverse crew in here. All right, we're going to see if we have better luck in this part here. Yep. Oh, looks good. All right. All right, now we hold our breath for a second as we pull the core out, and fingers crossed. And again, we've been seeing that this particular sediment is rather sticky. Awesome. Okay. And that is core number 9. Done. 17. All right, we've got one more core to go at this site. This is the most popular site in the Atacama Trench. It's Club Atacama. This happening trench site has it all. Right. So we're collecting a record 4 cores at this site. That's the highest so far on the expedition. Which, since this is the first day we've been down here, it's not bad.
Same area?
Yeah, please. Yes, please. Sorry.
We still make 1 core for you, 1 core for fauna, 2 for microbiology, and we still have 3 extra ones.
I might collect my water. I might collect the 2 liters of water here too. I might collect the water here. It seems kind of interesting.
All whiskey bottles are empty, so it's
No.
and activated super fast is just quick as well. If you want you to wait a bit until everything that's going around the sediments. Otherwise, you might get cloudier water than it's
That's a good idea. That's a good point. I might pick a different site. This place can be pretty subtle.
And if you want it, just do it the first push for
Yes. All right. This is push core for Perna, who is a scientist here in the university system in Chile. Perna is studying biofilms, and I think she was the dive watch in the time right before mine. Oh, I think that's the best one yet. Man. Any core that's Miguel's core is the best core. Miguel's core is the hard core.
And you said my pants were back.
Very nice. Awesome. All right. So, I think, all right, real quick to the control, everybody in the room, are there any more requests to sample from this site? Or do we feel that we have?
I think we're good.
Okay.
I think he took a cursory look around the rocks for any other leptochitons and didn't see any, so.
I think this stay that was supposed to be 5 minutes and turned into 40 or so. I think we're ready to move on finally. Awesome. Thanks, guys. So that was a really interesting spot there. You can see how everybody kind of aggregates together really quickly, and like we all found something we wanted there. So in terms of the geology and microbial groups, that is the most sampling we've done since about 9 a.m. So for those of you just joining us, we just moved away from a pretty interesting geologically, microbiologically speaking site, and we are moving forward. We are moving back towards our waypoint and trying to find that slight rise that we had talked about in the trench. We're hoping to find some additional geological features. Some of these critters that you're seeing here on the screen, we've already sampled one of these interesting creatures, and is in one of the bio boxes that we'll be taking a look at when we get it back on deck.
That's the first thing I would do before you just from the camera. I know you're one of the people.
Do you want to take over, or are you good? I can take over. If I keep going, you can take over.
A quick outro and then maybe a quick outro here. I'm on the phone.
Right. Do we have to slow down a little bit? All right. So for everybody watching around the world, I have been Drew Palmer. I am a professor at the Florida Institute of Technology. I'm going to sign off from my dive shift here. I'm going to turn it over to another member of the team, Antonio. So I will be back on tomorrow. Everybody have a wonderful day. You're up.
Thank you,
So, hello everyone. I'm the next in the shift for narrating this exciting dive. My name is Antonio Molina. I'm a planetary scientist. I've been studying the surface of Mars for more than 10 years, participating in several different missions from NASA and ESA. And as the people who were already following the dive are aware that we took very nice samples from both biology, microbiology, and geological importance. And I don't know if anyone in the chat speaks Spanish, but I would like to mix both Spanish and English from now on. And I will have a little chat with Miguel in here. That is. So.
We just came from a very interesting outcrop in which we found Leptochiton, an ancient mollusk. But we also got several sediment samples, and as soon as we can analyze them in the next few months, I'm sure we'll have great results. And it's great to hear, Antonio.
Paso al español para la gente que se conecte a partir de ahora. Estamos en la expedición oceanográfica Atacama Trench. Nuestra finalidad es encontrar fósiles vivientes en esta zona de la costa de Chile. Y ahora mismo nos encontramos casi a 3 kilómetros de profundidad en una zona que es realmente interesante porque encontramos donde se unen dos de las placas tectónicas, la placa oceánica de Nazca y la placa continental sudamericana. He pasado al español, Miguel. Si quieres también comentar algo sobre ti y sobre lo que estamos haciendo en español.
Muy bien, pues aquí en esta expedición de 21 científicos que somos, estamos 7 del Centro de Astrobiología, si no mal no recuerdo, de una amplia variedad de disciplinas. Probando. Ok. Perdón que ha habido un pequeño problema con el micrófono. Gracias, Antonio, por darme la palabra. Estamos aquí en esta expedición de 21 científicos que hemos acabado por venir. 7 somos del Centro de Astrobiología. No llegamos a ser mayoría, pero somos una parte razonable. Y venimos... el Centro de Astrobiología es un centro extremadamente multidisciplinar y, como no podía ser de otra forma, nosotros también lo somos. Tenemos desde microbiólogos, como es mi caso o el de Armando, hasta geólogos, como pueden ser los casos de Óscar, Javier y Antonio, hasta físicos, como puede ser el caso de María Isabel. Hay una amplia variedad de disciplinas y por lo tanto la variedad de muestreos que estamos llevando a cabo es también muy diversa. Habréis visto que hemos tomado muestras tanto de macroorganismos, que de eso se están ocupando nuestros compañeros en buena parte de universidades chilenas, como de muestras de sedimento, que ya analizaremos más adelante para entender cómo funciona la microbiología y los microorganismos que habitan estos sedimentos, como muestras de rocas para nuestros colegas geólogos y muestras de agua que también hemos ido tomando no a través del ROV, pero sí a través de una roseta que tenemos que nos permite tomar muestras de agua. Es decir, el estudio que estamos llevando a cabo, que no deja de ser un intento de determinar si efectivamente esta zona alberga modos de vida y metabolismos y organismos antiguos, no deja de llevarse a cabo, como no podía ser de otra forma, a través de una amplísima variedad de técnicas de muchísimo interés. Y estos transectos que estamos haciendo ahora a través del fondo marino, pues así intentamos hacerlos un poco más entretenidos para todos los que nos estáis escuchando. Muchas gracias. Te devuelvo la palabra, Antonio.
Muchas gracias, Miguel, por la explicación. Aunque puede que haya gente que nos esté escuchando que no sepa lo que es la astrobiología. No sé si... puede que haya gente que nos está escuchando que no sepa lo que es la astrobiología. So many people here may not know what astrobiology is.
¿En español? Both. Vale. La cuestión esencial es que la astrobiología es un concepto bastante difícil, como bien... Mira, lo voy a contestar en español porque Andrew Palmer ha contestado muy bien esta pregunta ahora y yo me voy a dedicar a parafrasearlo.
Perfecto.
se suele agrupar dentro de la astrobiología dos ramas distintas que son la biología espacial y la astrobiología per se. La biología espacial, que es una rama concreta que es a la que se dedica nuestro compañero Andrew, no tiene tanto interés en entender si hay vida más allá de la Tierra o entender formas de existencia distintas a la nuestra, como puede ser la astrobiología, sino entender cómo funciona la vida en el espacio. Entonces, experimentación en ese aspecto puede darse por una amplísima gama de distintas formas, desde determinar si es posible el cultivo de plantas en lugares como Marte, hasta entender cómo funciona la biología de los seres humanos en el espacio, y en buena parte de los experimentos que se realizan en la Estación Espacial Internacional quedaría dentro de lo que sería la biología espacial. La astrobiología, por otro lado, es una ciencia mucho más difícil por la razón tan sencilla de que no hemos encontrado vida en otros planetas. Entonces, tratar de trabajar es complejo, y es siempre a través de lo que, no siempre, pero en buena parte a través de lo que llamamos los análogos. Los análogos no dejan de ser lugares en nuestro planeta Tierra que son parecidos a lo que podemos esperar encontrar en otros lugares de tanto el sistema solar como el universo.
¿Y qué es lo que buscamos cuando buscamos estos análogos? ¿Son características físicas, por ejemplo, no?
Pues esto dependerá mucho de qué análogo estés buscando. Por ejemplo, si quieres buscar un análogo de los océanos que hay debajo de las capas de hielo de lunas como Europa y Encélado, pues no vas a buscar en el desierto del Sáhara. Probablemente vayas a lugares como donde estamos ahora mismo, a grandes profundidades, temperaturas bajas. Ahora mismo estamos a 1,82 grados, temperatura muy estable también, que se ha mantenido en las últimas 5 horas exactamente. Si por otro lado estuviéramos buscando análogos marcianos, pues quizá podríamos ir a lugares como el desierto de Atacama, donde ya hemos estado, otros desiertos como desiertos de Utah, el desierto del Namib, zonas áridas, pero también zonas frías, porque Marte es un planeta extremadamente frío. Entonces muchas veces condiciones muy calientes pueden ser difíciles. Y pasamos ahora que estamos pasando a muestreo, que es lo importante.
Muchas gracias, Miguel. Ahora mismo estamos buscando si hay otros elementos en el fondo que puedan ser de interés para su muestreo. You want to get a closer look to the green ball? Okay. So if you don't mind, we can go back to the green ball, yes, and maybe a closer look. If you can try that, yes.
Biology observation.
That's true, yeah.
Anyone know what that may be?
This one's better.
Una idea de qué estás pasando. Lo pongo como
Hay bastante movimiento en la habitación, estamos viendo hacia dónde movernos a continuación y qué muestras tomar. Y vamos a intentar hacer una aproximación a esa especie de bola de color verdoso. So we are approaching this green structure. To see if we can see it closer.
Oh, what's that? I'm just looking at everything under a known organism because I have no idea about biology. Actually, what you can comment on that? Yeah, that's what I used to do. A couple hours. I think I'm probably going to be for 4 hours.
So we are looking to some of rock outcrops over here. It's interesting for many factors because we are looking not just for the materials on the geology but also on the animals and microorganisms that may be related to those. Estamos buscando no solamente sedimentos, sino también estos afloramientos de rocas que pueden tener importancia no solo por su composición como por los microorganismos que están asociados a ellas.
Did that hide? Did we move sediment or did it hide?
This is with more lights on, so we should be able to see more of it, but it's darker.
So do you want to move on or to keep looking at this?
Take it.
You want to take it?
You want to take it? Okay, is it possible?
Do we have
If it's not too complicated, we can try.
suction sample?
Okay, we are going to try to retrieve the structure, maybe organism. Es esa esfera con forma esférica que encontramos verdes. Vamos a ver si hay alguna forma que podamos extraer una muestra de la misma.
Abajo a la izquierda, hemos visto varios ya.
Como podéis imaginar, esto es un trabajo de alta presión, no solo por encontrarnos a esa profundidad, sino porque estamos trabajando con una tecnología bastante compleja y a unas condiciones físicas complicadas. So this is not an easy maneuver. We need to go steady and very delicate. Nos vamos a acercar con este succionador con el que vamos a intentar absorber. Does anyone knows what could be that green structure? Have any idea on that?
No.
So in the room we are not sure what could be, so that's why we are trying to retrieve that, and hopefully we can get it with us and look at it carefully in the surface when we retrieve the ROV. No sabemos aún en la habitación con las imágenes qué tipo de estructura o organismo estamos viendo. Esperemos que podamos muestrearlo y analizarlo en detalle en la superficie cuando supamos el ROV. Vamos a empezar a absorber ese sedimento. It seems like we couldn't get it. It went away. Parece que no pudimos obtener la muestra. We're gonna give it another try.
Sorry, Zach.
So my colleague Oscar, that is next to me, is pointing to possible place that the structure will slip off. Let's see. Mi compañero Oscar ha identificado que se encontraba por esa zona. Esperemos que podamos obtenerla.
Can we try to lift the rock with the hand thingy?
We got the wrong rock.
Yes, this one.
That one, that's it.
So it looks like is that the crevice of the one right here?
Oh, my collapsed.
Let's go on.
Yeah,
I think we can go on.
we can continue, yeah.
Alright, let's section off.
Vamos a continuar moviéndonos para ver qué más encontramos. De nuevo estamos a casi 3 kilómetros de profundidad. Podéis ver que la diversidad y la cantidad de animales y organismos que encontramos es bastante abundante. Esto es debido a que estas aguas son muy ricas en materia orgánica. Yeah. Life, it's everywhere. That's why we try to look for life outside, even from Earth, because in here you can find Earth in the hardest conditions. So why not in other planets? Aquí podemos encontrar vida hasta lo más profundo del océano en condiciones tan complejas como estas. Ahora mismo estamos a un poco menos de 2 grados centígrados de temperatura. Ahora mismo tenemos luz gracias a los focos del ROV, pero habitualmente estaría completamente oscuro. So we are in about 2 Celsius degrees temperature, and it would be completely pitch black if not for the ROV lights. So the kind of light that we can find in here is quite peculiar. It needs to live in these extreme environments. Es unas formas de vida bastante complejas. ¿No ves eso? ¿Eso crees que puede ser sedimentos claros? Bueno, ya los hemos pasado, pero vamos a buscar. Yeah, okay. So we would try to go slower, just in case we are able to identify
anything.
Anything?
This is place, just in case.
Okay.
So maybe we can zoom in specific parts to see if we can see anything better and then go on.
Okay. Vamos a intentar ir despacio para encontrar nuevos organismos. Aquí como esa zona tiene como los sedimentos más claros, pero bueno, vamos a ver si más arriba.
What's the white thing?
Sí. Podemos. Can we get closer to that white? I mean, sorry. In here. So let's see what's in there. Vamos a ver qué encontramos. Do you know what?
Most likely noise.
It's a sponge. Okay. This is a sponge.
If they move, it's fish.
I think we can keep going then if you want to.
Okay. We can keep moving because it's nothing that we haven't seen before.
We can try over if you want to.
Maybe
But Oscar, we may try. Can you see this kind of sediments in here?
Oh, yeah.
Is this any way to get some sample on this? Maybe in the rock box, or I don't know what is the best way to.
Just depends on how fragile it is, because if it's in the
Okay.
rock box, if it's loose, it'll.
Yeah, we can go slowly and test it.
If that ground there or above right there that we're heading to, if that's good, if soft enough, I'd like to get our second pour.
Okay.
There's
Okay.
those-
In here.
Those green things there.
In here.
Where?
Yep.
Next to that rock.
Oh yeah.
Yes, now it's open. Yes.
I mean, we can go see, I guess.
Alright, the green thing first.
Yeah. Again, we found another of those green bubbly organism or structure. Hemos encontrado otra de esas estructuras
And there's another one
verdosas.
on the middle of the rock.
Where?
There's one here and there's
Oh,
one
in here. Can you see it? So either here or here.
La lengua
de un equiuran. ¿Cómo se llama? El proboscis. El proboscis
On the left, we have the proboscis of an equiuran.
of an equiuran.
In here?
Yeah, that thing.
That's the proboscis.
So,
Yeah.
Maybe that's priority to the green.
which one is more priority?
The curious.
The problem is the worm is inside, and what you see is the proboscis. I don't know. I was not here, so I don't know what I was going to
Okay,
do.
okay.
I just have to tell you that
Okay.
this is the proboscis of an equiuran. That's all.
Do you want to try to take a sample? Or do you rather not?
But can it go out?
He probably has to break the
Yeah,
rock.
we can just take a picture.
Well, it's the thing is, the animal itself is in a crevice. That's yeah.
Ujú. So, estamos viendo la probóscide
Anyway, I just have to tell you this.
de un organismo. I don't know if retrieving it is possible. We may just give a good look at it
Yeah.
and then try to scare it or something.
We can maybe try the sediment thing before, because we're not going to
Because it
live
was
here.
open, otherwise it will be again hiding.
No, there it is.
This is the similar to the organism that we
I don't know what is it. I can tell you what's the other one.
were looking at before.
I think it's similar, isn't it?
No. The proboscis does. These are here.
Is it another
It's very,
Is it a green
very
chicken
characteristic.
monster?
No, that's only like an acorn worm.
I think like those
But
were
the other one
big
is
parts.
different phylum, like you
I think this is similar to the one that we saw before. It's
I would
Yes.
prefer the
instead of being red, it's
Okay,
green.
Antonio,
So we call those chicken monsters.
hemos pillado ya los blancos.
¿Seguro que lo tenemos?
I'm just trying to, this is
Are we sure we go there the other?
I had not seen this before.
Okay,
I haven't
okay.
seen this. I just saw the other one.
So we maybe can just have a closer look to the other specimen that was in the upper part, just to have a an image.
Antonio,
Yeah, this one. So maybe we can get a closer look, so just to have the record.
we don't know.
I think it's the
Crustacean.
the headless chicken monster is just green.
Do you want to try to suck it?
Yes. Okay. Yeah.
Okay. Is it possible to try to suction?
No matter what, if we see the whole animal, maybe.
And then you have another one down there with some nice sediments.
Yeah, I was thinking.
The one that you think is easier. The one that you think is easier. Vamos a intentar
The green tongue is over there.
tomarlo con la aspiradora. We are going to try to.
So we're still on jar 6. Now we're on jar 5.
Yeah, it's very curious why is it green? Because in here there shouldn't be any light. So, ¿por qué es verde si no debería haber luz solar? It may be some other kind of pigments, not just chlorophyll or derivatives. So that's why we are here. Ooh.
Almost got him.
As I said before, it's super tricky
Luckily, we have another green thing.
to maneuver with all this technology. So we need to be gentle and go slowly. Each time we move, all the sediment also moves and makes everything difficult to see. Cada vez que nos movemos hay que tener cuidado porque todo se vuelve turbio y es complicado de observar. Pero vamos allá, vamos con este primer intento para obtener la muestra. Let's go with the first try, retrieving the sample. Hopefully we get it. Difficult to see with all the dust that's just lifted. difícil de observar con todo el polvo en suspensión o partículas en suspensión. Estamos intentando succionarlo y según parece, creo que está dentro. I think it's in, yeah. Yeah.
Do we not have
Good work.
on the suction boxes, suction places? No. Okay. Oh yeah, that's it.
Yeah, I think I got it.
Okay, so we have it. Yeah.
The guy in the rock thing?
Yeah.
Yes.
And we are going to take also maybe it moved. I cannot see it either. Does anyone is able to see it?
I cannot see it.
Green spoonworm. Okay.
Might be.
Yeah. It's possible that this green organism is a green spoonworm.
Oh, yeah, there it is.
Es una especie de gusano verdoso que podría... Su nombre científico puede ser Bonellia viridis, pero tenemos que confirmarlo, lo veremos cuando esté en superficie. And afterwards, we are trying to take some push course, right? For the sediment.
For geology team, if it
And
goes
for-
well,
or for Drew.
Drew will get one.
Okay, you want to do one for Geo first?
No, Geo wants one. Yeah,
If it works, I'll
Geo
take
wants,
one off.
It's an expert Geo push core, okay? So they're on 3 nights.
Yeah.
So
Oh.
if the next push course go well, take a push core
Oh,
here.
a piece of the claw.
So it should be basically. Anyways, we should get like 4 pushers here.
8 there.
We will still have 1 actual
All the way.
and then 2 extra bags because the other extra one will not be really.
Alright, you can set that back. Ram coming out.
Antonio, se ha caído antes un trozo de plástico amarillo. Entiendo que lo sabe, pero si es que se van a avisar. ¿Vale? Están con. Lo van a hacer. Antonio, tienen que recoger el plástico.
Yes, yes. So we are going to retrieve the little piece of plastic that just fall off. Vamos a tomar la parte de la garra que se ha desprendido para no dejar un residuo en estas aguas. So we're just retrieving the plastic.
You want? What's that? From 5 bucks.
It could go to a rock box if it's okay for you. I don't know, wherever it fits better.
Set that one, because that was found me. Antonio, don't close one because of the way they want to escape.
Yeah.
Put it in the big guy.
BB2 we cannot open.
No, I say rock box.
Okay,
One.
that's great. We have no this one. Yeah.
Okay.
Oh, you got it.
So if you guys think it's okay, we may try to take a couple of push cores in the sediments, in the white color sediments over here. Is it possible?
We're going to see how this right now look
Yeah, we may go first.
If it's just
Start with
before
geocore and then we'll
I think we can maybe sacrifice the geocore for it. ¿Tú vas a buscar la manita?
We are on
No,
11. Around.
Do you know which is the next score?
11.
11th, yeah.
We'll score 11, this will go to the geo team, which is the extra score.
So now we are sampling these light color sediments that needs to have seems to have some possible layering. Vamos a muestrear estos sedimentos de color claro que parecen tener ciertos. Yeah. Let's see how hard it is. Yeah.
Sí, para la próxima hay que ver si sale de uno que tenga una
So maybe it's too hard or.
Let's go, let's just bring our load to it.
So it seems like we are able to penetrate the rock or the sediment. Se ha conseguido obtener la muestra de sedimento.
Okay, who wants another postcard? What is the white stuff?
White is white. Maybe a carbonate.
Antonio, ¿tú va a querer el podcast?
Yes.
Seguro. ¿Estamos confirmando? Y Lore y yo también. Sí, a Parna te confirmaba, ¿vale?
So it's possible if we can take another two of the same kind of
Otros
material.
tres.
Another two, yeah.
Yo también. ¿Tú también? No, cuatro. Yo tengo una. Otros cuatro.
Okay, not two but four.
Four more or?
Four more.
Yeah, in the white part, Antonio.
3 or 4?
One, Parna wants one, Lore and I want one, another wants one. Yeah, we have
4 in
we have
total?
like 1 hour left.
4 in total or 3 in total?
4 in total.
Okay.
No, four. We just took the geology one, we need another 4.
Another 4?
Five.
Compartimos Lore, tú y yo y ya está. 3 more. 3 more. Antonio.
Yeah, okay.
3 more, okay?
Taking also the white part.
4 en total para de geología, 3 más.
Mhm.
This one goes to the second at the very side where.
So we are going to try to take this
We don't need that much.
these sediments more because we are, as we just said before, we are many different teams working in with different interests. So when we find something interesting, we need to retrieve and now sample for everyone. So somos varios equipos con diferentes intereses y es necesario que tomemos a veces muestras distintas para todos. Sometimes you reach the hard rock and you cannot go further, so we can get whatever it's inside. A veces este tipo de muestreos, este tipo de cores, solo penetran hasta cierto punto y no puede obtenerse toda la profundidad.
Yeah.
Yeah.
I'll take it.
Some people only needs the first
I only really
5
top 5
centimeters.
centimeters, but this is, and that top 5 centimeters looks neat. So.
Yeah, I mean, if someone else needs just the upper part, it could be, this one could be for other people, so we can then. So Romina asked, how are the samples disposed inside the road? So we have several boxes and this kind of cores and also others. And then everything is, as you can see, handled with these clothes. We have two clothes, this one that is made of metal, the other made of plastic that is a little bit more gentle. And afterwards, everything is, this part of the rover where everything is disposed gets inside the box, and we can go to the surface safely and then retrieve it.
Okay. More?
Yes. Did you? Hemos tomado dos, ¿cierto? Y otros dos más. So another two, please. Estaba comentando que, como podéis ver, todos estos recipientes se encuentran dispuestos en una caja con capacidad de movimiento. Tenemos varias cajas, varios tubos de este tipo que obtienen muestra a través de mecánicamente introducirse en el sedimento. Y también hay otras cajas con algunas rejillas que permiten obtener ese sedimento. I don't know. Let's see how large is it is. Yeah. The people who's driving this is the great crew of the Falco 2. They have lots of experience.
Okay.
Okay, they rather have it taken from close to the others. So,
next to the white
so we are able to take this kind of whitish material, right?
Yeah,
I think it's under all this.
it should be, but just in case.
Okay, over here?
Yep. Yeah, let's try that. Yep. Let's try there. Yeah. Thank you.
Yeah, there's a rock over here, that's what I was trying to get over.
Oh, okay.
It may not get so deep, you know?
Push it down.
Push it?
Push it down.
I mean, if it doesn't work, we can go to the place before. Which one should we go to?
Channel.
Just trying to match it.
So.
Okay, maybe in the place you said. You might be right.
Okay. Then we're back out.
Yeah.
We are going to be diving each day from the next 6 days.
We'd rather not, I think.
No?
If it's too hard to take it out, maybe we can just try another one. For the reason.
It seems that we wasn't able to take enough samples, so we need to remove. So okay, we can take it and just we have enough left, so yeah. No, don't worry. Vamos a recogerlo aunque no haya salido perfectamente. We are going to take it, even though it's not perfect, because it should be easier than empty.
Que en realidad tampoco está fallido, porque tiene ahí un estado bueno, pero por si acaso. Si conseguimos ese material blanco ese, curioso, que sea igual. bloquea. Okay, so Antonio,
Mhm.
if we can try another one for us, you can try the next one for us as well. For Mandalorian and I, we still count this as ours and failed, okay?
You still sorry?
We will still count this one as one of ours that failed, okay? Doesn't matter. Just another one.
Ese podría ser distinto para la vida.
So just another one? Or?
Another one for us and then a partner.
Oh,
So two more because this one
Okay.
this one Daniel will use, but for us it's not enough.
Okay, so let's try to take a couple more. Just let's see if we can get it with more material. Probably what you said before was a better place. Es complicado tomar estos sedimentos porque no sabemos qué hay debajo, y en el momento en que el tubo choque con la roca, pues es hasta ahí hasta donde podemos obtener la muestra. So, could it be closer to the...
Pero es lo mismo que lo que hay abajo.
Sebastian? Over there.
This area?
Yeah, that's right.
Okay.
Okay. There's a rock right below, so maybe we'll mark it too. It's good. Hay una roca abajo. No, está abajo. Tenemos ahí de nuevo. Descansar un poco.
And you left.
Yeah, understand.
That's all she got.
Do you know if Andrew already took water samples?
No, but he left his hoodie here, so he will come.
Okay. So
Okay.
everyone.
That's great. Sí.
Yeah. I'm just saying afterwards. It's okay for everyone if we take water samples.
For you or for you?
For Javier and.
Okay.
Yeah. I see you later.
Okay. Thank you so much.
Para verlo.
Vale. Sí, cuando termine.
Ahora, cuando meta ahí.
Sí, me parece buena idea.
Okay, so next one will be Aparna's. Aparna's second call.
Okay.
Okay. Another one?
Yes, another one for
Yes.
Aparna.
This
Yeah.
last row is when you ride in a sit camp, so I'm gonna try to get the porch all the way out.
Exactly, vete todo eso bien.
Call.
¿Cuál sería el número, Miguel?
Oh yeah, those are nice. That's a
the
little bit better, huh?
Way better. Like you're messing with me now.
Right, exact thing, I'm making everything a little bit harder. Alright, porch coming in.
Aquí lo bueno es que se ven bien dos capas diferentes de material.
Mhm.
El sedimento que hemos estado viendo durante todo el día, así con ese color así
So.
marrón, claro, verdoso.
Es-
Hay una capa blanca, hay una capa.
So Oscar, it's a college of mine in the Centro de Astrología. Oscar, un compañero del Centro de Astrología, está aquí con nosotros. No sé si quieres hacer algunos comentarios.
Sí, hemos llegado a una zona de topográficamente más inclinada.
Everybody happy?
¿Está bien?
Yeah.
Yes. Y aquí lo que vemos es, aparte del sedimento que llevamos viendo durante prácticamente todo el día, con ese color pardo, marrón, un poco verdoso, por debajo vemos que hay una capa blanca que podría ser de carbonatos. Y que en los testigos que hemos sacado, en los cores, hemos visto como unos 4 o 5 centímetros de ese material de sedimento superior, y en la zona inclinada junto a las rocas negras que se ven ahora en la imagen arriba, está ese material blanco por debajo que
So it's okay, yeah.
Que podría ser un carbonato o quizá una arcilla.
Maribel quiere muestras
Sí,
de agua.
lo sé.
¿Vale? Quiere 25 litros, que es el volumen total. Va a haber que decirle que no, porque Drew quiere también.
Claro, por eso quiero saber qué quiere
Le
decir.
pillamos con solo 5 y ya.
Pero para filtrar o
Cosas de Maribel.
Ready?
I'm ready.
Okay, we are done with the sediment sampling. Hemos acabado tomando las muestras de este sedimento. So now we may wait a little bit for the sediment to settle and maybe take a closer look with the cameras where we took it, so we can see how the sediment looks like. Vamos a esperar unos momentos a que se depositen las partículas en suspensión y poder observar cómo es el material debajo de donde hemos tomado estas muestras. So we are waiting for the sediment to settle so we can look at the place where we just took it, so we can see in the fresh how are the layering and the different colors and the texture. That will help us when we study the sediment in the surface.
Alright, we'll pull the porch in and then we'll wait for the viz to clear.
That's it, huh?
Mm-hmm. Yes, thank you. Okay, can we get closer to this area? Perfect. And then to the left to here. Like, yes, over here. Okay, thank you. So we would like to take water samples, but in a place that where the sediment is not, lifted yet. So we may be better go a bit farther away, right? Or waiting here. What do you think is?
Probably just move. I think if you don't want this disturbance, let's just move ahead about 20 to 30 meters.
Okay. So let's do that.
Anything just setting auto altitude patterns out?
Yeah, or depth. Probably depth.
Mm-hmm.
Yeah.
Station keep and all that.
So now we are taking some water samples.
Ask how
So
high he wants them off bottom.
we will need to move a little bit away. So the sediment is settled. Vamos a tomar unas muestras del agua que hay en esta zona, para lo cual vamos a desplazarnos un poco a los alrededores. Y de paso continuar explorando este fondo marino de la costa de Chile. Seguimos en esta campaña de exploración del Atacama Trench. A casi 3 kilómetros de profundidad, buscando en estas aguas organismos que se crean, quizá algunos extintos y que sigan vivos. Yeah, I think it is. So, how much water do we need?
5 liters for Javier. Who will
So-
start with that?
One bottle, 5 liters.
Okay, one of the 5-liter napkin.
How high above bottom do you want it?
Huh?
How high above bottom?
How?
Javier?
Is it clean? I mean, is it, we don't. We moved away from the area where the.
Okay, so it's not, there's no turbidity from the working, okay? So, as far as possible, I mean, as close as possible. If
As close
it's
as possible.
2 meters.
You want us to sit down?
If you think you can do it without because otherwise,
No.
between 2 meters and 3 meters, for us it's exactly the same.
Yeah,
3 meters would be fine.
We'll just 3 meters.
Yes.
We are going to want to sample water close to here after as well, so
I
try to
prefer
avoid.
3 or 4 meters above the ground and clean than 2 meters and a half and dirty.
That's perfect for us.
Thank you. Vamos a aprovechar que hemos alcanzado esta zona donde el agua está bastante más clara para abrir cerrar una de las botellas que toman muestras del líquido alrededor. So, do we know if Andrew wants also from here? Can anyone text him?
I'm trying to.
And does anyone else need water? Because maybe this is a good time.
Oh, can you look over the port arm? I think I got the port arm.
We can take maybe another bottle just in
Take
case.
another bottle because we think Lauren doesn't want it, but just in case we can take
Okay.
another 5-liter water for Lauren.
So can well,
Those rock. Did you guys, have we done any Niskins yet?
No,
not yet.
Okay.
all of them are free.
We got a cloud coming.
Oh, yeah, maybe we should,
This can
move ahead.
We can move a little bit more.
Can we
Yeah.
do it while we're moving?
Eh?
Can we do it while we're moving?
Yeah.
There's some sediment down there, so maybe we can try to go again the other way. So we have 4 5-liter bottles and 2 2.5 liters ones, right?
Yeah, they just want undisturbed water from.
Sit, I think I can rock it with this.
Alright, let me get you over here.
Which bottle? We need a 5-liter bottle, Antonio.
2.
Sorry?
Yeah,
One
that's
right
it.
now.
Oh, yeah.
For
You
Javier,
got your sit,
and
you
one
right?
for Lauren.
I got my
2
sip, you have a
5-liters.
Santa Cruz.
Alright, so I'm just gonna hit, am I hitting them all, or how many?
Hit 2 5-liter bottles for now, please.
2 5 liters for now, I guess.
1 and 2, right? Okay.
Mm-hmm.
Alright, that's it?
That's 1 and 2.
Just
Just
2.
yeah.
Okay.
Actually,
Yeah, do 3 as well.
3 as well?
Yeah, for Andrew, just in case. And then we can keep going and just wait until he comes back. Which is right now. Andrew, we have some drinking water. I think we'll just hit 5-liter whiskey bottle for you here.
Okay.
Is it
One
okay?
more. We still have two 2.5s and 1 5-liter bottles. And I don't think anyone else wants. So.
I mean, if we use any more, it'll just be for Mara. So.
Mm-hmm. Okay.
I would say if you don't need them, it's a good idea to pop them, just in case anything didn't work.
Yeah, if
Yeah.
you can pop everything, I think that will be.
Will work?
Okay.
Antonio, Javier y Maribel son la misma botella.
Vale.
Es una pregunta.
Sí.
Okay. Javier, Lauren, and Andrew.
Where's that coming from?
And then 4, 5, and 6, they are everyone.
So Armando,
I can't tell that.
do you want to keep moving?
Yeah,
Okay.
we can win. Team. Javier.
What are we missing? I mean,
We still have 2 cores to fill.
I'm not sure if 4 went.
And that's basically, we have both rock bottles.
okay,
are
thank you.
empty.
So all the water.
I mean, we are mostly done. We can maybe walk around, see if we can find a nice structure to put the last 2 cores in, and be done with it.
Okay.
Oh, yeah.
Okay.
It's a good test anyways.
Yeah.
All of them fired correctly, right?
One of them we think maybe didn't.
Which one?
Number 4.
We don't
Okay.
care about that. So I'll
It's-
just put an asterisk.
Yeah, I think we have more than enough. So after this, I think we are about to wrap up. So we can. I don't know, move maybe a little bit more to take.
I think we can spend the next hour just moving around, see what we find.
But whatever is which direction is better for you guys. I don't know if you rather to keep going or you need to come back at some point or.
We can go. I mean, we can follow the line you guys put in.
Okay, we can keep going.
Yeah, let's follow the line.
You guys doing alright? What's up, man? You guys doing alright?
Vamos a seguir hacia adelante. We are keep going,
You want to keep playing?
exploring the bottom of the Chilean coast. About 3 kilometers deep. Seguimos en estas aguas de la costa de Chile.
You want to go out? I can tell.
We're just flying in a line now. So.
I can see the smile on Tyler's face. Okay, I'll just text me if you want on break.
All right.
¿Qué es esa? ¿No tenemos biólogo marino?
Oh, se ve que tiene.
Can we zoom a bit closer to that if possible?
Yeah, a glam
We're basically finished,
video
so we
maybe,
can
yeah.
get some nice pictures. I think that's it.
¿Un pepino? Sí. Sí, cucumber.
Oh, he's fast. He's faster than my kind of dog.
Oh, that's
Sí,
beautiful.
su branquia.
Yes, like everyone in the chat must be like.
So take the time you need for filming it.
Es como el otro que
Floating guys.
Oh, he left.
Okay. We can maybe grab some more rocks.
Pero ese género que se vio, sí que era para los dinosaurios. Era una especie.
Okay.
Y esta se parece.
Yeah, we can keep going.
Sí, eso es una especie de
Es una especie de fruta.
¿Te parece si narro un poco?
Sí, claro.
Hoy hemos terminado ya buena parte de los objetivos del día y tenemos ya prácticamente todas las muestras, tanto biológicas como de sedimento, rocas y agua que estábamos buscando. Así que la hora y media que nos quede hasta que empecemos a volver a la superficie la vamos a dedicar a hacer una serie de transectos, ver si encontramos algo
Hello, this is Zach.
muy llamativo y así estaremos, ¿vale? Cualquier
Hey,
pregunta nos decís.
He is. Sure, and I'm sure she can go in there too, you know. Did she already go in there? Yeah, she can totally go in there and check. I'll tell Adam as well. I'll come back.
Thank you.
So we're mostly done. We're just
Explore the ocean.
exploring a little bit more, yeah. Estamos casi acabando. Vamos a seguir explorando un poco más. Pero hemos encontrado parte de lo que nos interesa. Yeah. Do you think that we can take a closer look just for looking? I mean, just for the video?
Oh yeah.
Come back a little bit.
And we may.
Yeah, we can't forget about the sea urchin.
Yes.
Forget the sea urchin?
We can't forget about it.
Okay.
Oh, we can forget. And what about this?
Coming back a little bit.
Do you want to go back, Armando?
There's a white brachiopods we just
Can you come and
No, and let me know where.
Yeah, when I see it, yes.
Okay. Next side. So maybe if you can go stay out here. We have to focus on something.
Yeah.
So we can focus on just
here somewhere.
I will leave it until an hour and a half. Okay. And then we can try to super surface.
Yeah, the bad thing to going back is just got all the dust.
Yeah, better head forward. It's very dusty bottom here.
Yeah, don't worry, let's keep going. If we see it again, we're going to try to.
Sorry about that. So we are going to continue. The soon you go back, all the dust that you leave when the ROV is moving is there, so we cannot see very clearly. Vamos a
What time are we supposed to come off bottom? What time are we supposed to come off bottom?
I think it was 5:30 more or less. 5 is the strict time. So we have like an hour to square around.
So you don't want to get any certain distance, just keep going, see what we see?
That's it.
Is that something, Andrew? Is that something?
Can we zoom on the little
Wait.
what?
Atrás.
Back?
Come back.
We cannot go back because when you go back, you have all the dust, so you cannot clearly see
It's
anything.
like 10 centimeters to the back.
Okay.
We just need to go slightly back. There it is. Okay, so on that formation,
Okay.
you can try to zoom on it.
This, maybe this one, or maybe this one actually.
No.
But the dust.
You can just wait until it clears.
Just a little bit.
What do you think it is?
Maybe. I will have a brachiopods.
So it could be a brachiopod.
Yeah.
Let's try to look at it closer. And wait until.
I feel I'm driving somewhere in Antofagasta.
Try to clear it out.
Okay, so
Great reveal.
there we have it. Yeah, we are zooming in.
Let's see spider.
No, esponja. No.
This is a sponge. This is the thing that they
we need
thought.
this anyway.
Yeah.
Oh, picnogonid.
Yeah.
Oh,
We got amphipods.
let's get the picnogonid.
What should I
Arthropoda.
So,
All of it together.
Como araña.
okay.
Parece una
I
araña.
didn't even see the picnogonid.
Okay, how do we get this guy? Slurp?
Yeah.
That's great.
So we are going to take these specimens.
No, it's not gonna. They're very, but they're not gonna run. But you may lose a leg here and there. They're very brittle.
Yeah. They are super fragile, so we need to be very careful. Son
I couldn't see the picnogonid. No way.
unos organismos muy frágiles y puede que se vean
Picnogonid.
afectados por la toma de muestra.
You're still on jar 5.
Una araña marina.
We're going to go to a new one, right?
Es una araña marina, así spider.
Sí. Pero
Four is empty, yeah?
en todo el mundo. En Caribbean, Antártica.
Suction sample jar 4. You guys have that empty?
Sí.
Suction jar 4?
4 is empty. We have 3, 4, and 5 empty.
Y,
Okay.
pequeños hasta.
So it's on 4.
Sí.
So let's try to get them. Vamos a intentar acercarnos para tomar esas muestras de organismos. Again, those are real brittle, so we need to be very gentle, but let's see how it goes.
Give me suction.
Suction, full.
Hold on, buddy, don't run.
It's pretty good.
Max suck.
Let's see, it's moving. It's going away, but we got it. Okay.
So,
Great.
Suction place 4, right? Yes.
Yeah.
Guillermo, ¿quiere la esponja también? No? Just leg. Oh, yes, okay.
Can we also try to get the sponge over here?
Does that suction into the, it's in the jar.
Is it there?
Yeah.
Same jar, yes.
Same jar?
Yes.
Same jar? Yes.
Yes.
Yes, they say so. Yeah.
Suction's still on.
Suction 4.
Yeah, you might need a little torch on this one though.
Okay.
Oh. Let's see how it goes. This small sponge seems to be very stick to the rock, and also it's very flat and not. Yeah, the pilots are doing a great work in here.
What's here?
Yeah, he's kind of stuck on there. I don't know if I'm gonna
Okay,
get it.
we can forget about this point.
That one.
Ah, okay. Yeah.
Right?
Yeah.
We can forget about the sponge. It's very difficult. It's an indentation of the rock.
The sponge is really difficult to catch, but we have those small organisms.
Yeah, I think it went in.
I think it went, yeah.
Yeah, I think it went in. Yes.
Hopefully it will in, yeah.
How do I assign the chiton to?
Con suerte hemos podido tomar una muestra de ese organismo. Uno de esos living fossils.
All right, we done?
To which section sampler did this go to? 4 as well?
4.
Okay.
Any more? Or are you guys good?
Is everyone happy? Can we keep going?
Suction off.
So, la hija de Armando pregunta si podemos ver algún pez. We are going to be able to see any fish, so con un poco de suerte podremos ver alguno pronto y hacer un zoom para verlo en detalle. Son rápidos, pero seguro que podemos verlos antes de que se marchen. ¿Continuamos?
I think we can go, yes. We can go.
So
Good?
let's move. Yeah. Oh.
Buen día.
Let's keep moving.
No, plastic. Antonio,
Mm-hmm.
tenemos que recuperar el plástico.
Yeah, can
Ya lo
we?
sabe, pero bueno.
Yeah, you probably already know the purple.
What's that?
A little bit of electrical.
You have to recover the purple plastic. It can go to rock box 1 or 2, which the other one went as well.
So we are retrieving the purple piece of plastic that fall. Again, we don't want to keep anything in here that wasn't there before. So estamos obteniendo esa parte de plástico que ha salido del brazo aspirador y lo vamos a guardar para recuperarlo. Okay.
That thing pops out again, we'll just leave it.
Okay.
What's coming in?
That's good.
So, vamos a seguir moviéndonos.
Antonio, Armando quiere mirar el antiguo. Este no hagas caso, no hagas caso.
Vale, vamos a continuar. Let's continue. Just in case we see anything more.
¿Hemos encontrado algo antropogénico?
No, no se puede.
Eso pregunto. Pero en realidad está bien. Porque aquí no pasaba mucho. Iban hasta México, cruzaban México y salían ahí a la anomalía. Por aquí no pasaba. Lección de historia.
Sí, sí, Miguel, si quieres contarnos un poco.
Estoy inventando todo.
Yes, Miguel was just saying some.
Esto es una expedición astrobiológica, no historiográfica.
Exactly, we are not aware of any.
¿Qué? Ah, sí, lo tenía con eso. Vale, vale. Antonio, ¿cuál es?
Do you want to get close to here?
That one. Sí, sí, eso. Si se puede, we have 2 empty suction boxes. So
Okay.
I don't know if it fits.
Maybe we can first take an image of
Puede
it.
que no quepa. Bien. ¿Cómo se llama?
Es una holoturia. ¿Lo puedes repetir, Miguel?
Holoturia. No hay opción de Cnidarians.
Sea cucumber, maybe?
Sí, cucumber, yeah.
I think that you have sea cucumber as an option.
Yes, yes. Holoturia. Qué bonita, voy a hacer otra porque ya te vamos a ver.
It should be on, it's just terrible when there's any dust.
Okay. Yeah, I'm still on.
Margarita says that if we can catch that.
And we need to readjust it. What?
Margarita
Sorry,
says,
you're on
my daughter,
three.
we can catch that.
You were on 4, so both of those should be okay.
We are going to try, but
Okay.
it may not be suctionable.
You want to suction this?
I think Guillermo wants to try it. It may not be possible.
Margarita will talk.
Okay.
Sí, Margarita pregunta si podemos tomar esta muestra. Vamos a ver si sería posible.
Que separar la vida personal.
We are going to try to catch
You can hop a little closer to that.
this cucumber, but we are not sure if we're going to be able to take it without damaging it.
Alright. Up.
They do look like a vacuum, yeah.
Quizá quieres contar algo sobre los pepinos. No son muy interesantes. Son morados. Tienen un cuerno. Tienen unos caracoles parásitos. Tienen unos caracoles parásitos. Estos pepinos de mar tienen al parecer unos caracoles parásitos que tienen algo interesante.
So these cucumbers usually have some parasites that may be interesting to study. Yeah.
We're on jar 3
We
right now.
have 3 on 5.
We're on
Ready,
3.
so this is in 3. Okay.
3's good?
Let's see if it works. I'm so sorry for this poor picture.
That's Max.
Is he stuck though?
No. Oh no, it's stuck.
Just keep suctioning him for a minute. Oh, maybe that'll get him.
Okay. I mean,
Alright, you can kill the suction.
Suction dead.
And that's the only thing I don't like about these new jaws is it's got this thing so
Slippery,
easy.
right?
Gotta give him some more texture or something.
So
Yeah, it's still on there.
there are some new pieces of equipment that they need to be
At least
get
you're
used
gonna
to
get
use
him either
them.
way.
yeah, if it blocks it, you can unblock it here, right?
Hay piezas nuevas de equipamiento que son complicadas de utilizar, todavía no están suficientemente testeadas.
Sí, quizá para otros días los super más los dejamos para como hemos hecho.
Alright, now you can, is he suctioning?
Suction back on. That's max. I think he took the suction off.
So it seems that it got stuck in the suction, but let's see if we can
No, actually they suffer a lot.
take it off. Okay,
Microstomes
it's in there.
are very closely related to us.
So hemos sido capaces ya de tomar esa
Developed
esa muestra.
nervous system. I am certain they suffer a lot. Ellos tienen
Can't tell if it's in there.
deuterostomes.
It looks like
nosotros.
looks like something's in there.
So yeah, they're cousins.
Alright, suction coming off.
¿Puedes preguntar si ha entrado? Bueno, si lo has visto. ¿Ha
And
entrado
away.
ha entrado al SS3? Ok.
Sinceramente, Miguel, y los primos.
Tengo
Yeah.
primo más feo.
Just on jar 2.
So is it, where was his store?
That's in jar 3.
So, jar 3, okay.
Yeah.
Okay, that's good. We have jar 5 empty, and that's it.
Okay, so
Which one's empty?
According to this, 5.
5, that's the only one?
yeah.
So there's stuff in 2?
Oh no, there's something but it's scratched out, so I think there may not be anything.
Okay. I just want to make sure we stay on empty jars so nothing swims out.
Okay, no, I think there's nothing on 2, but
Okay.
someone wrote, but then they crossed it, so. Okay. We have 2 more jars empty.
Alright, I'm just gonna hold this thing for now.
Para chupar cosas.
Alright, froze, locked.
Alright. And let's continue on our way.
Yes, we are continue moving.
Now to where it's like a park. So they don't actually close anymore.
That's good. That's good. Initial run, huh? Thing about these feet too is when we get down in there, we get anchored. Good. There we go. I'm gonna reel me in a little bit, Zach.
Yep.
So let's see if we see something new.
Antonio, vamos a ver la cosa verde esta, ¿no?
Do you want to have a look? Maybe we can
Maybe we can zoom in on the
just to
green thing,
take a look to this
just to see how the structure is.
green sediments or greenish olives maybe. Vamos a parar a ver de cerca esos sedimentos de color verdoso.
Thanks, Zach.
Isabel me está llamando, dile
No hagas caso.
que necesita. And there, okay. Yeah, we can pan a little bit through the full outcrop.
Yep. Antonio, quizá podrían probar a romper un poquito la piedra y tener un trozo verde y guardarlo con las demás. Si pueden.
So,
O en un coral cuello o algo así. Quiero un trozo de la verde.
is it possible to pick a little bit on the rock to see how consistent it is, to see how it breaks or it's super hard?
Y pichar un trocito verde en cualquier caso. Si
And
se
if
puede.
possible, retrieve a little bit even with the coral rivers?
Give it a shot. I got my professional rock guy here.
I don't know about a quiver though, you have to open, I'll do the port arm to break it, the port arm can't reach the quivers.
We can put it with the other rocks.
Can go in a rock box?
Yeah, in any of the rock boxes.
¿Interesante para esto? Ya no, yo sé. Antonio, ¿podemos mirar esto un poquito más?
Antes de romper la roca.
Yes. Before breaking the rock, can we zoom in this? Zoom in with the camera? Just
This little guy.
Yeah.
That little guy. Zoom in on that little guy.
So nicking here was.
So this is not attached. I don't think it's attached to the substrate. Como un reducto pequeño.
Okay.
So this is not attached to the substrate. The suction is not working anymore, right?
Yeah, we have one space left.
But there's
No, but it went into the.
Yeah, we think it's working.
Then with a very light suction, you can get the whole animal.
We'll have one space. So.
It's not attached to the surface. It's just
Okay.
loosely.
Okay. We can try to suction it before breaking the rock.
Just put Cnidaria, or you can put if it's Medusa, you can put Skyphozoa.
Alright, and we are currently on jar 2.
Yeah.
Port check.
Yeah. Just put Skyphozoa or Cnidaria.
Leave my arm out,
Yes,
right?
put Cnidaria beside
I'm just going to shuffle to the left.
So we have confirmation that the sucking pump is still working, and we should be able to retrieve that. Creemos que la extracción anterior fue exitosa y que vamos a poder obtener esta especie de pequeña medusa. Let's see.
You needed water from the Niscan bottles as well, right?
I didn't want to, but my name is there, so I'll just sing it, yeah.
That's us.
Good, give me a suck.
Max suck.
So no, that's.
You have 5 liters of water, okay?
Okay, I'll work on it very fast.
No, 5? Yeah.
So let's see if it goes in gently.
Max suck.
It's coming.
There it is.
Yeah, it's in.
Okay, we have it. We got it. La hemos conseguido obtener. And I think we don't have any.
We still have one left. We have
One space
SS5.
left. Okay.
You can just leave it running if you want.
But we should try with the rock, right?
Huh?
We should try with the rock.
Yeah, please. If you can break the rock and give me a decently big green part, that's great.
Mhm.
That's cool, right? You see the spikes?
Esto. Esto.
Esto.
This.
Otra esponja más.
Dejamos. Okay, the one.
Can we first take the green rock and then go there?
Yes. Yeah, of course. Yeah.
Okay.
Yeah, we got most of them. I can't say all.
Yeah. Can we just? Do you want to take a closer look to that before?
No, take the rock first. Rock first.
Okay.
Yeah.
Okay.
All right, so for the rock, can you kind of hop back where you were? Because I had my eye on one of them.
Yep.
So let's see if this rock is
I think I see a loose one.
very hard or if we can rock it. Vamos a ver si esa roca es suficientemente blanda para que podamos extraer una parte.
Oh, I'll save it, yes, okay.
The green color doesn't need to be anything related to life. It could be just the mineral. We just need to know the composition of it. In here, mostly we have volcanic rocks and also sediments that have been metamorphosized, so affected by pressure and temperature because we are in between these two states.
Yes.
Yeah, it should work, yeah.
I mean, it's huge, but I can work
Nice,
with it.
it's moved.
We can share it. I don't want this
It
gravel is work.
seems a little bit, yeah,
Yeah, that's great. Thank you.
perfect.
Alright.
Perfect catch.
Very crumbly.
Let's throw this other ram off for you.
Sure.
El color, como digo, puede verse más bien a su composición, pero puede que no, porque ahí vemos que no parece que llegue hasta el fondo. No lo sabremos hasta que lo analicemos en superficie.
One or two. Antonio, quiero preguntarle qué caja es esa.
Which box is this one?
One. Okay.
RB1. Thank you. And then I think they want to have a closer look to where is it, Nick?
Ahead.
Over here?
That one, yes. Okay.
So let's take a look to there. Vamos despacio, intentando que cuando aterricemos no se levante demasiado polvo.
No metes a pepinos de mar.
So we are trying to get down slowly, yeah. Perfect. And let's see what's there. So Nick, what we are looking at?
We're concentrating in those two esponja,
A sponge?
sponge,
Yeah, in here.
Sponge as well.
and then sponge as well.
In here, right? These two.
Wow, so many
And we got maybe another picnogon, I don't know, I cannot tell right now because it's full of detritus.
No, ese es un Cnidaria.
Cnidarians quiere, but
O un luna.
this is completely covered with detritus.
Es posible sacarla bien.
Un tubo anemone aquí.
No,
No, prefiere. Is it possible
To get the whole rock.
to use the suction to get a small crustacean?
What? Quiero say whether it would take the whole rock. Everything.
So either try to suction this small crustacean in here, or they are also yes.
No, sabemos de los tamaños por el pilar de láser.
Small crustacean. Antonio, could we put the laser to know the size of that? Poner un láser para ver el tamaño.
Okay. Can we point to this direction so we know the distance? In here?
Size.
Yes.
The laser, yeah.
Yeah. There you have it.
This is like a couple centimeters, 1 centimeter.
So the crustacean must be 1 centimeter large. Parece que el crustáceo tiene un tamaño de aproximadamente 1 centímetro.
Antonio,
let's try to get the whole thing.
Do you want to?
Like this.
Okay.
Se va a desnegar.
Let's try.
Armando said that could it be possible to retrieve the full rock? I don't know if it's loose enough, but we may try.
Okay. I don't know if it's part of a bigger one, you know, but.
Yeah.
Probably it's like a rock attached. But if not, let's try it.
You know, that's the door to the underworld.
Like if it moves.
Vamos a intentar tomar la roca, aunque seguramente esté fija. Yeah, it seems to be attached, so yeah, maybe it's better just to try to gather the crustacean, yeah.
If it's a bigger rock, forget it.
Parece que ese afloramiento rocoso es suficientemente grande como para que no podamos tomarlo, así que simplemente vamos a intentar absorber el pequeño crustáceo que hay en la superficie. You don't want to take it? Okay. It seems like we abort the mission and keep going.
Let's do it. Well, that's good.
Can any of you text the others if they have any specific needs before we leave? Just
I will
make
tell
sure
them that this is the last half an hour. If they want
Yeah,
anything,
that's
it's
it.
time.
20 minutes we have.
Sí, os digo que quedan 20 minutos y que si quieren algo, pues también.
Let's go for drinks.
So it's been a very long first dive for the expedition. We were able to retrieve many exciting samples, both for organisms and rocks and sediments. So we have a lot of work ahead of us, but it's good to have it. Llevamos una primera inmersión bastante larga, unas cuantas horas desde que empezamos, pero hemos sido capaces de tomar muestras bastante interesantes tanto de diferentes organismos como de materiales, tanto rocas como sedimento. Aún queda un montón de trabajo por delante. Una vez el ROV, el vehículo, alcance la superficie, tendremos que almacenar.
Antonio.
Yes.
Loren va a querer un core más para nematodos, ¿vale? Aquí mismo.
Okay, so maybe we can
Que lo confirme, pero vamos parando.
we can start stopping because we want to retrieve a final push score. Let me just they are
One push
checking.
for wherever, around here.
Okay,
So as deep as possible.
maybe far away from this creature so we don't disturb it, but anywhere when you find sediments that would be easy to sample. So, as I was saying, this is just a part of the hard work, because when the ROV gets to the surface, we need to take all the retrieved samples and prepare them, analyze some of them, others we will just storage them for analyze later. So lots of hours of science ahead of us, and tomorrow hopefully we will keep exploring again. Como decía, llevamos un día largo de muestreo. Hemos empezado esta mañana tomando muestras de agua a lo largo de toda la profundidad, unos 3 kilómetros, y ahora hemos estado explorando el fondo marino de esta zona. Sin embargo, luego todavía queda un montón de trabajo analizando todas estas muestras de sedimentos, de rocas y de otros y de organismos. Parte de ellas haremos parte de sus análisis aquí en el Falcortu. Otra parte la almacenaremos para llevarlos a nuestros centros de trabajo y estudiar, pues, o bien su composición, sus morfologías, hacer diferentes cultivos para analizar los microorganismos que habitan en esos sedimentos. Yeah.
Yeah.
Looks good, yeah. Hopefully we get the full thing. We are taking another core sample, just trying to find, I think, little crustaceans for some of the teams that are working here in this campaign. Estamos tomando otra muestra de core para analizar si contiene pequeños crustáceos que estudian uno de los equipos que trabaja en esta expedición. Parece que hemos sido capaces de obtener una muestra casi completa, faltan algunos centímetros, pero creo que será suficiente. We are missing some part, but it's more than okay. Did anyone say anything else?
I think plans to keep moving for like 10 minutes. Then wherever we are in 10 minutes, get the last score.
Okay.
And go through that.
So a few minutes more work, and that's all. Yeah.
Cool. We have for these 10 minutes one last suction chamber, and that's basically it.
Okay.
And the last score, which remember.
Let's keep moving.
Yo este último no voy a tomar más.
Vamos a continuar. Solo nos quedan unos 10 minutos.
Lo subimos y si no lo tiramos. Okay. Lo pongo como general o para quien quiera.
O si ves alguna esponja, la tomas así como...
También.
Esa esponja
Espera, me escapa.
que tenía un camarón dentro.
On the left up the hill, there are some rocks.
Yeah, we may go to that direction when we find some rocks. Parece que encontramos algunos afloramientos de roca hacia la izquierda. Bueno, ahora just straight ahead, right?
No, to the left. To the left of the ROV.
Oh, yeah.
But,
Go straight ahead or furthest to the left?
So Nick, where? Nick?
Son igual. Ahora son iguales.
Okay, we can.
Yeah, we can go straight ahead.
Okay. We're going to keep moving. When we get all the samples in the surface, we are going to take a very careful look to, different layering, how the materials, and also if it's some organisms in there. Una vez que alcancemos a obtener esos push cores, vamos a analizarlos detenidamente haciendo un análisis de su granulometría. Vamos a analizar con lupa cómo son esos materiales y los diferentes grupos tomarán muestras según sus intereses. Algunos les interesan las partes más superficiales de esos push cores, que es donde se encuentra la mayor parte de la vida. Otros buscamos entender cuál es la composición completa, así que iremos tomando muestras a lo largo de la profundidad de esos push cores. So we are going to analyze them very carefully, sectioning them, trying to see if there are any organisms living in the spaces in between the little grains, or also analyzing the grains themselves very carefully through an augmented lens. And then the different teams will take different portions, subsamples of each course, because some of them are more interested in the upper parts where most of the life is expected to be. Others want to analyze the full thing just to understand what's the variability on materials and also textures on the sediments.
Bridge, ROV, got a new waypoint?
Copy that, RV.
It's a nice outcrop.
Ujú. ¿Bahía hacia dónde? ¿Hacia la izquierda?
La entrada que hay a la derecha.
¿Acá?
Ahí,
¿Acá?
ahí.
Okay. So if we can get closer to this area and to see in detail, would be nice. We can see many different organisms living in this area, usually associated to rocks. Vemos un montón de organismos distintos asociados a rocas. So is there anything specific that you want to...
Let's go to the last big rock because there's a lot of things.
Yeah, let's go to the big rock ahead.
Okay, so in here. We're going to the large rock ahead of us. Vamos hacia esa gran roca que está enfrente de nosotros. Sí. And can we get a pan view, a close-up pan view of the front of the rock? Vamos a tomar una panorámica de detalle de todo este frontal de roca donde vemos diferentes organismos. Super beautiful view. Muy hermoso. It's incredible how this deep we are able to see this variety and rich life. Oh, sorry. Okay, so they want to try to retrieve this rim in here with the suction. Just in the last available, the chamber 5, yeah.
We're on 5, so that works.
Vamos a intentar tomar esta pequeña gamba, small shrimp. We are going to trying to keep it.
The suction wasn't working super great last time. I think it probably has a little bit of that polyethylene in there. So I don't know if it's going to work, but I'll try.
Okay, perfect. Yeah, that's all that we can ask. Thank you. So it seems like one of the samples that we retrieved may have partially
Antonio, tenemos solamente 10 minutos. Vámonos para recoger el coral, por favor.
Pero no el sí. Ven.
Are they going to try for the camarón?
Probably should try.
Because they told me that the section was limited.
Yeah, but they will try either way.
If it doesn't work,
Okay.
next to the bumble, there's a little coral. Next to the big bumble, on the right, there's a little coral.
You can.
Well, okay.
Later, Jerry. Yes. Stay close and.
Not to. That is full suck.
We're lucky they'll shoot backwards into there, but I don't know.
So let's see if we can take the shrimp. Probably not, but yeah, they are super fast.
It's not sucking at all.
Okay.
Too much cucumber there.
I think there's too much cucumber inside that.
Alright.
I mean, okay. ¿Llevamos el coral y tiramos?
Esponja de coral. Okay.
Arriba.
But the suction is not working, so.
Si quieres el último core, lo quieres. Si no, no vas a tirar. Ok, hacemos that.
Yes.
Para la última,
Yeah.
let's get a little coral next to the bubble.
But how with
It's going to be easy to go like this.
Okay, so we might just to finish, right?
Yeah.
Yes.
We finish. It's okay. If we don't get it, that's fine.
Okay.
Yeah.
Yeah, I think it's enough.
Yeah. Mañana otra día.
Oh yeah. So thanks everyone who was following this full dive, and we will tell you tomorrow more about what we have already found when we get the samples to the surface, and what is the next goal. We are probably going to be closer to the fault that we have to the east on here. Vamos mañana a seguir muestreando. Gracias por seguirnos durante esta inmersión, y mañana continuaremos más hacia el este donde hay una pequeña falla. Así que muchas gracias. See you all tomorrow. Nos vemos mañana. Muchas gracias. Hasta luego.
Alright. Thank you, Antonio.
Phew, phew.
Okay.
Hey everyone, good evening from Falcor 2, where local time is about 18:48. Thank you so much for joining us for our first dive of the living fossils of the Atacama Trench expedition. We've got excited scientists hanging out in the hangar, waiting for the vehicle to be recovered. We're all very excited to see what sample processing will bring us tonight. As always, thank you so much for watching. Don't forget to like, subscribe, share with a friend. We're always happy to have you back. And tomorrow we will be launching ROV SuBastian for dive 679. Shortly, early in the morning, we're going to try and keep a consistent schedule this expedition, so we will all see you back tomorrow. Thank you for joining us for this little midwater ascent, and we'll see you tomorrow. Take care.